Hi everyone, good morning. Hi everyone, a very good morning to all of you. Good morning, I hope you all are doing good. Am I perfectly audible and visible? Am I perfectly audible and visible? I hope I'm perfectly audible. Yes, please do let me know in the chat section. Do let me know in the chat section. Good morning, I hope you all are doing good. Am I perfectly audible and visible? I am audible. Just a minute. Am I? So great guys, welcome to the session. So today this marathon is for the class 11th syllabus, and you know that we are going to target the unit diversity, and along with that we are also going to target the chapter cell, right? So in the next marathon we will go for the structural organization, we will go for in the plants as well as in the animals, plus cell cycle will be done there, and then human physiology and plant physiology. Okay, so class 11th portion is very important, and these marathons, they will definitely help those who are right now giving the board exams, right, for the CBSE. So these marathons are going to be very helpful for them, because here in detail we'll be covering the class 11th syllabus, right? Yeah, I'll start from the basics. And there is one more good news for you. So you people can see here, on 7th of March, there is something very important for you. Let me show you this one. So what you have to do, just go to this link, click on this link, right? Click on the notify me so that you will get the update. So here you will see the amazing plans for NEET 2026 as well as NEET 2027 and 2028 aspirants as well. Okay. Yes, I will start from the basics, but please do not spam here. And if you are going to spam here, I don't care. Don't worry. Okay, so let's get started. So the chapter that we are going to start is Animal Kingdom, right? This is what we are going to start first. Animal Kingdom. And yes, we will stick to the NCERT for this particular marathon so that you can get best out of it. So let's start the Animal Kingdom. Okay, ready? Ready? So let's start the Animal Kingdom. Okay, so you know that when we talk about these kingdoms, yeah, when we talk about the classification, how do we start? In our syllabus, we talk about the R.H. Whitaker, right? R.H. Whitaker gave the 5-kingdom classification system, right? R.H. Whitaker gave the 5-kingdom classification system. I hope you all are well aware of it. And even we talk about Carl Woese as well, who gave 6-kingdom classification system. Yes, please. Who gave 6-kingdom classification system as well. But mainly in our syllabus in detail, what do we discuss? We talk about the 5-kingdom classification system, isn't it? So when you talk about these 5 kingdoms, because these are the basics here, right? So when we talk about these 5 kingdoms, which kingdoms do we have? We start with Kingdom Monera. Yes, please. We start with Kingdom Monera. Then there is Kingdom Protista, then Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia is there. Yes or no? And Kingdom Animalia is there. Yes or no? Yes or no? So these are the 5 kingdoms that we have. right? These are the 5 kingdoms that we have. So when you talk about kingdom Monera, here you talk about the prokaryotic organism, and in rest of these kingdoms we have eukaryotic organism, right? What do we have here? We have eukaryotic organisms here. Now, the point here is, see, we are going to discuss this part. So when we talk about the kingdom Animalia, so here you will see all multicellular. There is no exception here. We will see multicellular eukaryotes, and we know it very well, right? Here we have to study the different phylums. Yes or no? So overall, you know that there is a trick, right, for these taxonomic hierarchy, that is keep pot clean or family gets sick. Yes or no? Keep pot clean or family gets sick. Yes or no? Yes or no? So it's keep pot clean or family get sick. Yes or no? This is what you have to remember. So when I'm saying keep pot clean or family get sick, so here you will see kingdom, phylum, class, order, family, genus, and species. These are the 7 broad taxonomic categories. Are you getting my point? These are the 7 broad taxonomic categories that we have to study. So it's keep pot clean or family gets sick. That is your kingdom, phylum, class, order, family, genus, and species. That is something that you have to remember. Is that clear? Is that clear? So that is something that you have to remember. Okay. So the next part is what? Here in this topic, we will study the kingdom and the phylums. Only in the case of Phylum Chordata, we will study it up to the class level. Otherwise, kingdom and the phylum. So when it comes to the animal kingdom, right? So which phylums are we going to study in kingdom Animalia? Which phylums are we going to study? So here there is the list. So in this sequence, you should remember it. And if you are able to remember it, guys, trust me, other basis of classification will be easy for you then. Okay. So I'm writing the name of all the phylums here. Please note down or do revise it. Just have a look. So firstly, there is Porifera, then there is Cnidaria, Ctenophora is there, Platyhelminthes is there, Aschelminthes is there, Annelida is there, Arthropoda is there, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and then finally Chordata. So it is the Phylum Chordata in which you will study it up to the class level as per your syllabus. Otherwise, this is what you have to study. So Porifera, Cnidaria, Ctenophora, Platyhelminth, Aschelminth, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata. These are the phylums that you have to study. Is that clear? Right? These are the phylums that you have to study. Yes? Yes? Done? So that is what we have, right? That is what we have. So before starting this part, we will talk about the basis of classification. Why is it so? See, when it comes to the animal kingdom, we humans belong to the animal kingdom. Even a rabbit belongs to the animal kingdom. Squirrel belongs to the animal kingdom. Bird belongs to the animal kingdom. Snake, lizard, spider, mosquito, flies, right? Even a jellyfish, they all belong to the animal kingdom. So we are so different from each other, but still, if we belong to the same particular kingdom, right? Same kingdom, if we belong to the same kingdom, so what does it mean? It means that we have certain common characters. Yes or no? Yes or no? That we have certain common characters. So that is why, right? That is why we belong to the same particular kingdom. So this is what we have to study now. We will talk about the basis of classification. Are you getting my point? So the very first thing we will talk about is basis of classification, where we will talk about the level of body organization. Are you getting my point? Where we talk about the level of body organization. Yes or no? Yes or no? Right? Level of body organization. No doubt we start the things from the protoplasmic level, right? Protoplasmic level, when we are saying, then you are actually talking about the protozoan group of kingdom Protista, because that is compared to the animals. But here what are we going to start? We will talk about the cellular level, tissue level, organ system level. So we know this hierarchy, that cells together they form tissue, tissues together they form organ, organs together they form organ system, and organ system together is going to form what? It is going to form organism. This is what we study. So same way here also, very first thing that you have to remember is ki cellular level of organization is there, right? In which we talk about the phylum Porifera. Then comes the Cnidaria, or also known as Coelenterata, right? It is even the Coelenterata and the Ctenophora, next phylum. So firstly, try to remember the sequence here. So they both will have, just a minute. So they both will have tissue level. Okay, so when I'm talking about the tissue level, so it is going to be Cnidaria, it is going to be Ctenophora. So Cnidaria is also known as Coelenterata. Then comes the organ level. Under the organ level category, you will put what? Platyhelminthes. What do we have then? Platyhelminthes. Then comes the organ system level, right? So here from Aschelminthes to Chordata. Is that clear? Yes, everybody. So if you remember the sequence of the phylums, then these things are going to be easy for you. Is that clear? Then these things are going to be easy for you. So that's how you have to remember this. So this is the level of body organization. Any doubts here? Sure. Any doubts? Okay. So the next part that we have to discuss is the germ layers. So germ layers, right? They are going to form the body organs. So basically, you know that we all are developed from a single cell known as zygote. Yes or no? We all are developed from a single cell which is known as zygote. We all are well aware of it. So when that zygote grow, you know that the 2 cells blastomere, the 4 cell, and then morula, blastula, all that stages will come, right? Then gastrula formation is there. What is there? Gastrula formation is there. This is, let's suppose I said zygote forms, right? Then we talk about the morula, we talk about the blastula, we will see the blastocyst, right? We will see the gastrula then. So this gastrula is actually having what? This is having the germ layers, right? Germ layer, right? To germinate, yeah, germ layers here. So what these germ layers are going to do, my dear students? So germ layers are going to form the body organs. Is that clear? Now, what's the point here? When we talk about these germ layers, right, on the basis of that, we have 2 types of organisms. We have diploblastic organisms and we also have triploblastic organisms. When we say diploblastic organism, di means 2. Here you will talk about 2 germ layers, ectoderm and innermost is endoderm. These are the 2 germ layers that we discuss here. Triploblastic organisms will have 3 germ layers. They will have ectoderm, they will have mesoderm, and then they will have endoderm. That's what you have to remember. Are you getting my point? So here you have ectoderm and endoderm. There you have ectoderm, mesoderm, and endoderm is there. Now, when you talk about the diploblastic organism, right? Just a minute. Okay, so when you talk about these diploblastic organism, let's suppose here So basically, what am I trying to tell you? That here, outermost layer is what? This outermost layer that you people will see here is going to be ectoderm. Then this innermost is the endoderm. As I said, in between you will see mesoglia. What will you see? Mesoglia. Like these are two layers. In between, what will you see? You will see mesoglia. So it is an undifferentiated layer. It is not made up of proper cells, right? It is undifferentiated, right? Undifferentiated layer it is, which is present in two. So if you have ever seen a jellyfish, right? If you have ever seen a jellyfish, then you should know this thing, right? In the case of jellyfish also, that jelly wala part is there. Like we say, na, that is the movable form medusa. Basically that part, jelly-like part, it is mesoglia, right? It is mesoglia. Done, beta? So that is how it is. So ectoderm is there, endoderm is there, then in between you will see mesoglia. But there, ectoderm, then mesoderm, then endoderm is there. Now what's the next point here? Yes, what's the next point here? Next point here is very simple. See, when we talk about the porifera, even in NCERT, they didn't mention it. They didn't even mention it, right? They are not talking about it there. But actually in porifera, there is no germ layer, right? There is no germ layer. Then next to it is your Cnidaria and Ctenophora. In their case, what will you see? You will see that these are the diploblastic organisms. What are they? They are diploblastic organisms. So from Platyhelminthes, yes please, from Platyhelminthes to Chordata. From Platyhelminthes to Chordata, they are triploblastic. They have 3 germ layers. Is that clear? What do they have? They have 3 germ layers. So if I have to show you the diagram, this is the diagram, right? So diagram based questions are also there in your exam. So outermost is ecto, endo, and in between this is the mesoglia, diploblastic organism, and that is going to be the triploblastic organism. So now if you see that what kind of diagram it is, basically let's suppose if I am an organism, you are taking my transverse section, right? And then you are observing it. So outermost layer is basically ectoderm. So ectoderm in our case, like it forms the central nervous system, if I have to give you the example. Mesoderm form the skeletal system, endoderm form the gut lining. So this part here inside, it is the lumen. It is the lumen. Actually, this endoderm inside, right? This endoderm inside, it forms the digestive system. So it is that, you know, alimentary canal. That alimentary canal is having that empty space. No, that lumen. This is what you are talking. Is that clear? Is that clear? Okay. So diploblastic, and then it comes what? Then it comes the triploblastic. Understood? So next to it is what? Next to it is the symmetry. Next to it we will discuss is symmetry, right? So symmetry basically is what? Like how that organism is arranged or something. Yes or no? Yes or no? Next topic that we have to discuss is what? We have to discuss the symmetry, right? The arrangement, that how our body organs are also arranged. Is that clear? Yes, please. Is that clear? So that is what we have to discuss here, right? The symmetry. So it is basically the arrangement of body parts, or I would say if an organism is symmetrical, it is having a balanced arrangement of the body organs. Is that clear? So when it comes to the symmetry, right, though there are many other things as well, right, but as I said, we will be basically focused on the NCERT. So when you are talking about the symmetry, first of all, on the, if we consider the symmetry as the criteria, so yes, there are organisms which are asymmetrical, there are organisms which are bilaterally symmetrical, yeah, bilateral symmetry or the radial symmetry. So that is what we have to discuss. Is that clear? Right, just a minute. So it is a kind of balanced arrangement of body parts in an organism, isn't it? Balanced arrangement of body parts in an organism, that is what symmetry is. So firstly I told you, asymmetrical organism, asymmetrical, like they do not have the symmetry, right? You cannot divide them into equal or the identical halves. That is what it means. So basically what will you say when any plane, be it like this, whatsoever it is, when any plane passing through the center cannot divide the body into the equal halves, then you will say that the organism is what? Organism is asymmetrical. Yes or no? Yes or no? Yes or no? Isn't it? So that's what you have to discuss here. Are you getting my point? Are you getting my point? So when any plane that passes through the center cannot divide the body into equal halves, or I would say identical halves, then that organism is asymmetrical. So again, as I said, examples are important. So example is Porifera here. But yes, when it comes to the Porifera, exceptions are there, like Cyclostomata, Leucosolania. These are the examples of Porifera, right, which shows radial symmetry. So exceptions are important. And this exception is very important. Why? Because it is the example given in our NCERT. Understood? Understood? So that's what you have to remember. Next is your radial symmetry. So when we talk about the radial symmetry here, the best example that we consider is of starfish. The best example that we consider is of starfish. Just allow me a minute. Yes, it is of starfish. So when you talk about this particular example, any plane that is passing through the center, be it like this, right, or like this. So any plane which is passing through the center can divide it into equal halves. That's the meaning of radial symmetry, right, beta? When any plane which is passing through the central axis of the body can divide body into equal halves, yeah, two identical halves. So I'm writing it here. When any plane that passes through the central axis can divide body into identical halves, then obviously organism is what? Organism is basically having the radial symmetry. So obviously you know Porifera, next to it is your Cnidaria, right? Next to it is your Ctenophora. No doubt here in some members you also study biradial and all the biradial symmetry as well. But as I said that here one thing is clear, right? Here one thing is clear that we have to go as per the NCERT. So yes, they show the radial symmetry. And adult echinoderms, जैसे your starfish, it basically belongs to the phylum Echinodermata. To which phylum it belongs? It belongs to the phylum Echinodermata, starfish. So adult echinoderms. Are you getting my point? Adult echinoderms, they show what? They show the radial symmetry. They show what? They show the radial symmetry. So this is a very important point, but their larvae show bilateral symmetry. So overall, in the case of a phylum, which symmetry are you going to consider? Right, we consider the, like if somebody is asking you that where do you put the phylum Echinodermata, obviously we will put it under the radial symmetry. Why is it so? Because adults are showing it. But yeah, this is a fact regarding it which you really need to know, right, that the adult echinoderms, right, they have the radial symmetry. But their larvae, they show what? They show the bilateral symmetry. So that's what you have to remember. Is that clear? Is that clear? So next is bilateral symmetry. Let's talk about it. Next is bilateral symmetry. So bilateral symmetry is the word. Yes, when the body can be, because bi means two, lateral means side. Like let's suppose if there is any plane that is passing from the body, any one plane, right, which is passing, can divide body into, I'm giving you an example here. Like let's suppose one plane can divide the body into two identical right and left halves. So when the body can be divided into identical, when the body can be divided into identical right and left halves by, right, identical right and left halves, right, identical right and left halves in only one plane. By only one plane, yeah, in only one plane. Are you getting my point? In only what? In only one plane. Then it is the bilateral symmetry. It is the bilateral symmetry. So that is what you have to remember. So example is what? Platyhelminthes. They are having this bilateral symmetry, yes or no? So from platyhelminthes to chordata, right? Chordata, we will have this particular symmetry. So that's what you have to remember. So if I have to ask you a question from this particular part, right, and even it is a PYQ without options, I'm asking you, like let's suppose give the example in which organisms have, the organisms are bilaterally symmetrical and triploblastic. There are many examples, right? There are many examples. So I can just pick up two particular characters and on the basis of that, I can frame the questions, right? I can frame the questions. Is that clear? Is that clear? Any doubts? Sure? Sure? Done? So that is how you have to remember, okay? So next to it is the coelom that we have to discuss. Next to it is what? It is the coelom that you have to talk about. Okay, so what do you understand with this particular word? Coelom. Anybody? What do you understand with this particular word that is coelom? Anybody? Basically, when you are saying coelom, you are talking about the body cavity. What are you discussing? You are talking about the body cavity, right? Cavity means a space. Actually, what is happening, right? In general, what is happening? Just say, let's suppose you have ectoderm. Let's take the example of a triploblastic organism, of course. Ectoderm is there. Endoderm is there. And when in between this ectoderm and endoderm, in simple way, firstly I'm explaining it, in between this ectoderm and endoderm, you will have the space, plus when that space is lined by mesoderm, then you are saying that here in between, you are saying that this is the true coelom. What's that? This is the true coelom. This is what you can consider. Now, because this ectoderm is also, right, making your outer body wall, in another way if I have to define it, in all the possible ways I'm defining it. So outer body wall, outer covering of our body. Now you know that alimentary canal is there, right? The gut wall. So outer body wall and the gut wall and the space in between. Plus the condition is that space is lined with mesoderm. You can see it here. That space is lined with what? That space is lined with mesoderm. Just see, here we are making this. That space is lined with what? It is lined with mesoderm. So it is actually what? It is the true coelom. It is the body cavity in which body viscera, body organs will be there. So even if in this way you are looking at it or you are taking a TS of it, right, you can see that. So when there is a space in between the outer body wall and the inner gut wall, who's making your gut wall? Endoderm, as I said. So the space in between, plus that space is lined with mesoderm. It's not just the space, plus I'm saying that that space is lined with mesoderm, right? That space is lined with what? That space is lined with mesoderm, and in between you have the body organs, you have the viscera. So that's how you get it, right? This is what you will see. This is basically the coelom. Yes or no? Yes or no? So presence or absence of cavity between the body wall and gut wall will decide the kind of organism. Is that clear? Is that clear? So coelom is the body cavity, right? So coelom is the body cavity. So here, what will you see? You'll see, let's suppose the organism. If an organism is acoelomate means it is without body cavity, right? It is what? It is without body cavity. That organism is not having the body cavity. Is that clear? Right? Right? So where do you see that body cavity? Body cavity basically we are saying the space between body wall and gut wall, which is lined by mesoderm. The space between body wall and gut wall, right? Space between body wall and gut wall, right? Which is lined by mesoderm. If it is available, then you are saying that body cavity, or I would say coelom, is present. Okay, that coelom is present. So that's what you have to remember. So here the organisms are without body cavity. So from Porifera to Platyhelminthes, you will see there is no body cavity. Next is pseudocoelom. Pseudocoelomates. So pseudo means what? Pseudo means false. So this is actually not the true coelom. So what is happening here in this case? So in this case, you will see, yes. So in this case, you will see that in between body wall and gut wall, there are mesodermal pouches. Like this mesoderm is not completely lining the body wall or the gut wall, right? No, it is not like that. But here you will see that mesodermal pouches are present. What is there? Mesodermal pouches are present. So this is the pseudocoelomate. Example here is Aschelminthes, your roundworms, right? Roundworms are the example here. So here what will you see, right? You will see that in some organisms, body cavity is not lined by mesoderm. Instead, the mesodermal pouches are scattered between ectoderm and endoderm, right? So in some organisms, body cavity is not lined by mesoderm. Instead, mesoderm is present or scattered between ectoderm and endoderm as mesodermal pouches. That's how it is. Okay, so mesodermal pouches are there. Is that clear? Right? Mesodermal pouches are there. Okay? Done. So next is what? Next is eucoelomates. So U means true. U means what? U means true. Eucoelomates. So in this case, you will say that body cavity is lined by mesoderm from all the sides. Is that clear? So eucoelomates are of two types on the basis of, yes please, embryonic development of this coelom. So one is schizocoelomates and the second one is enterocoelomates. The schizocoelomates and enterocoelomates. That's what you have to discuss. Schizocoelomates and enterocoelomates. That's what you have to discuss. So I'll write the examples here, but before that, so see, up to Aschelminthes you have read here. So your Annelida, Arthropoda, Mollusca, they show schizocoelom, right? That is arm. Okay, right? So you can remember it as arm or ma, M-A-A. So schizocoelomates. Rest. This is enterocoelomate. So obviously we start with Echinodermata, then comes Hemichordata, and then there is Chordata. So they are basically what? Enterocoelomates. I'll explain you the meaning here, but that's how you have to remember. So last time in NEET 2025, there was a question from this part, and that was not a direct question, right? Right? Like they are basically a kind of, they have given you a case, then they were saying, just say, let's suppose if I have to give you an example of the question, just say you are going to a, going to a forest, you find an organism, in that organism you are noticing that mesodermal pouches are there. So what type of organism it can be? So questions can also come in this way. Right? So you have to prepare accordingly, and you can only answer such questions when your concepts are clear, right? So it is all about the concepts. Okay. So eucoelomates body cavity is lined by the mesoderm, but there are of two types, right? That here schizocoelomates are there, enterocoelomates are there. So from this schizo, from this S, I just remember one thing, the split, right? I just remember one thing, the split, right? Split means, so coelom is formed by splitting mesodermal mass. Like there is a kind of split in the mesoderm, right? That split will result in the development of the cavity. That's the point here. But in the case of enterocoelomates, from the gut wall, a cavity will start forming like in the form of a pouch. Is that clear? Is that clear? So coelom is formed by, here in the case of enterocoelom, coelom is formed by fusion of gut pouches during embryonic stages. Formed by fusion of gut pouches during embryonic stages. So it is just the matter of development, that how this coelom has developed. Okay, that how this coelom has developed. So before going forward, look at the radial symmetry, look at the bilateral symmetry example. So this is the one which is given in NCERT. So yes, examples are important. So let's suppose if this example is given to you, let's suppose if this diagram is given to you in the exam, right? They're simply giving you the diagram, and then they are asking you, right? And then they are asking you that what type of symmetry this will show? Radial symmetry. This one will show bilateral symmetry, right? So next is coelom. So you can see when you are talking about the acoelomates. So yes, acoelomates and this triploblastic diagram, it is same. Here, what are they trying to show you? See, this innermost is endoderm, you know it. This one is ectoderm, you know it. And this one is mesoderm. So there is no space in mesoderm. There is no space in mesoderm. That's how you can remember. This is pseudocoelomates. So where do we see the pseudocoelomates? We see that in the case of Aschelminthes, that is roundworms. That is roundworms. And then here you people can see, right here, you people can see, just have a look. Just have a look, guys. Just have a look, right? So ectoderm inside endoderm, this is actually the lumen, the gut, lumen here, and in between this mesoderm you have the space, you have the cavity. So it can develop by basically it can be schizocoelomate in nature or enterocoelomate in nature. So that's what you have to remember. Now if I have to quote one more PYQ here, give me the example of an organism which is triploblastic plus acoelomate. Which is triploblastic plus acoelomate. So what should be the example here? What should be the example here? Triploblastic plus acoelomate. Yes. Obviously, Platyhelminthes. That's the example. What's the example? It's Platyhelminthes. Am I clear? Platyhelminthes. They are triploblastic plus they are acoelomates as well. Right? So the example here is Platyhelminthes. That's how you have to remember the example. So here you can see on the basis of development as well, people. So this is endoderm, early mesodermal cells. There is a split in that mesoderm, right? There is a split in that mesoderm that is resulting in the form of cavity. So there you can see the development. Now here, these are the early mesodermal cells. So as I said, because endoderm form the gut, so there is a fusion of this gut wall, right? Fusion of this gut wall. So first of all, from this gut, there is the pouches. Pouches will get separated from this gut wall. Just I told you, in the case of enterocoelom, right, it will develop from what? It will develop from what? It will develop basically from the gut wall. Yes or no? It will basically develop from what? It will basically develop from the gut wall. So here, the word is given, this enteron. It means gut itself. That's how you can remember. Because this word itself means gut. Are you getting my point? This word itself means gut. So coelom is basically the body cavity. So here what is the thing, beta? Right here, from the pocketings of, or from the, like in the pouch-like structures are coming out from the primitive gut, right? This is further developing. This is not developed yet. So from this primitive gut, these pouch-like structures are coming out. Later on, if you will see, they are basically growing and they are forming the coelom. So it is like how the coelom develops, okay? So small pouches, they are arising from this gut wall, right? From this primitive gut wall, they will separate from the gut and the cavity will develop inside them. So that's how, right? So this wall of this pouch is basically made up of what? Mesoderm. So that is how it works. So that is how you have to remember it as well, okay? Okay, so next is, yes. So when we study this part, okay, right, so next is segmentation, but before segmentation, few other terms that we will discuss. Okay. So first of all, ये देखो, जैसे open circulatory system is there, closed circulatory system is there. But before starting this open and close, firstly, let's talk about the body plan, right? In general, what sort of body plan do we have in organisms? Okay, let's discuss about what? Let's discuss about the body plan first. Okay, let's talk about the body plan. So number one is cell aggregate body plan. Number one is what? Cell aggregate body plan. So cell aggregate, like it is made up of cells, like you know that the tissue is still not there, only cells are together. So in cell aggregate body plan, right, where do we see this? We see this in porifera, that is your sponges. B is blind sac body plan. Blind sac body plan, let's talk about it first. So blind sac, it doesn't mean the sac is not able to see. Blind mane, basically here you are saying, let's suppose there is a tube-like structure. That tube-like structure is having only one opening, right? Only one opening is there. Only one opening. Now, if let's suppose if any organism is having such type of body plan where there is only one opening, so don't you think this opening will be used for ingestion as well as egestion? Only this opening will be used for ingestion as well as egestion, right? And in such organisms, even the incomplete digestive system, yeah, incomplete alimentary canal will be there, right? They will have incomplete alimentary canal, yeah, incomplete digestive system, means they have only one opening which serves as both, mouth as well as anus, right? Mouth as well as anus. So both the things are there. Are you getting my point? Both the things are there. Are you getting my point? So incomplete alimentary canal. So only one opening which serves as both mouth as well as anus. So such organisms are having blind sac body plan. So digestive system is also incomplete here. Is that clear? So from coelenterate to or Cnidaria to Platyhelminthes. And yes, do not forget that it is biology's science of exceptions. So exceptions will always be there. Next comes the tube within tube body plan. Tube within tube body plan. This is the next part. Okay, it is tube within tube body plan. So here what is happening, let's suppose this is your outer body wall. Inside you have gut wall, and this gut is complete, this digestive system is complete. It is having a separate mouth, it is having an anus, right? So basically here you will see two different openings are present. Yes or no? Two different openings are there, right? One will be your mouth, the another one will be anus. So such organisms will have complete digestive system. What will you see in such organisms? You'll see complete digestive system. What will you see? You people will see complete digestive system. So when such type of body plan is there, just say outside is basically what? It is the body wall. Inside you have what? You have the gut wall. So this is actually tube within tube body plan. Am I clear? Right? So it is found in those organisms having complete digestive system. Okay, found in those organisms having complete digestive system. Okay. So this is the tube within tube body plan. So obviously you'll start from the Aschelminthes here to Chordata. Okay, Aschelminthes to Chordata will have this. Is that clear? Right. So with it we will also discuss this part also. Just say open circulatory system, closed circulatory system. These are also the terms which are given in your book. So in open circulatory system, you know that there will be a heart. The large blood vessels are associated with this heart, but it is not like that to each and every body organ the blood will pass through these blood vessels. No, it's not like that. Let's suppose there is a heart, right? With that blood vessels are associated. Heart will pump the blood will pass here in this sinuses or in this cavity or in this coelom. So body organs, they will basically bathe in that blood. So here cells and tissue receives directly the blood pumping out of the heart. There is no closed blood vessel here, that there is no specific blood vessel here which is going to innervate each and every body organ. Right? So this is the open circulatory system. You see that in the arthropoda, like other arthropods, right? The cockroach, you'll see it there. Then closed circulatory system, blood is circulated through the arteries and veins and the capillaries. Through in these blood vessels, blood will pass. Is that clear? So Annelida is the example for the closed circulatory system. So basically in open circulatory system, you cannot say that there is no heart, there is no blood vessel at all. Heart is associated with some large blood vessels, but they are not passing that blood to the respective organ. Like further, there is no from arteries, yeah, any arterioles or something are developing from it. No, it's not like that, right? That heart will pump the blood from that blood vessel. It will pass to the body cavity there. Body organs will bathe there. That's it. So this is open and the closed circulatory system. So next is the segmentation, which we also call as metamerism, right? Segmentation and metamerism. In which phylum you will see it for the first time? Phylum Annelida. In Phylum Annelida, we will see that for the first time. So Phylum Annelida. The best example that I can give you here is your earthworm. Are you getting my point? Earthworm. That is the best example. So what exactly is this segmentation actually? So here basically beta, body is internally and externally. Matlab here you say that body is externally and internally divided into segments. For that we also use the word metamers with a serial repetition of at least some organ. That's the definition. Are you getting my point? So here body is divided into a series of repeated segments which are metamers, or right, which are metamers basically beta. Which are what? Which are metamers, right? They can also be referred to as somites along a longitudinal axis. This is what we see, like body externally or internally. In the case of your earthworm, yes, in the case of earthworm, we see that external segmentation is also there. We also see internal segmentation. Just for this internal segmentation, you are using the word septum for singular, septa for plural. So here outside, you will see the grooves there. You will see the annuli there, the grooves there. It is like that. So external, internal segments are there. So there is the body is externally and internally divided into segments with a repetition of at least some organs. Is that clear? With at least the repetition of some organs basically. Okay? Okay? Sure? So in the case of this earthworm, or I would say in the case of Phylum Annelida, in the case of Phylum Annelida, yes, there is metamerism, right? So segmentation, I told you, it is also known as metamerism. So this is the true segmentation, right? This is the true segmentation in their case. But yes, we also talk about the pseudo-metamerism, right? Pseudo-metamerism is also there. So the example for pseudo-metamerism is tapeworm. Tapeworm. In the case of a tapeworm, which is a platyhelminthes, right, you will see that this is how the body is. These are the segments that we see in their case, right? But these segments are not embryonic. They are basically coming out from the neck. They are not even embryonic. From this neck region, they will form. Are you getting my point? So this is the pseudo segmentation only, right? This is the pseudo segmentation. Is that clear? Is that clear? Right? So external segmentation is there, right? Here you will see, we will be able to see some external segmentation. It's not embryonic. From neck, basically, these segments are forming. And moreover, there is no repetition of internal body organs here. Done. So tapeworm is the example, okay? Tapeworm is the example, okay? So in Phylum Annelida, for the first time, you will see this. So Annelida may we see the segmentation even in, as per NCERT, if I have to mention, Arthropoda, they show segmentation. Chordata, they show segmentation. Right? They show segmentation. Now in Mollusca, you will see the true segmentation is not there. In their case, we'll talk about the torsion, right? In the case of Mollusca. Then other than that, again, power issues. So other than that, right, so in the case of Chordata, yes, we do see the segmentation. So we belong to the phylum Chordata, even we show the segmentation. Yes or no? Yes or no? Yes or no? We have the beta in our embryonic stages, the mesoderm, it develops into the somites, right? In our case, right, the mesoderm, it develops into what? It develops into the somites, right? That segments. So basically, they are forming that vertebrae, ribs are there, these muscles are there, right? And even in the case of adults also, you know that we have the vertebrates. Yes or no? We have the vertebrates. So vertebral column, vertebrae, what are they? They are the repeated. That in vertebral column, that vertebrae, basically they are repeated. So this is basically the true segmentation. You have the spinal nerves as well, etc., etc. So yes, we also show the metamerism, we also show the segmentation. Is that clear? Yes, everybody. Done. So next is the notochord that we have to discuss, right? So notochord is exactly what? Notochord is basically, just this diagram is also given in the NCERT. Question will definitely come from this part this year. So please pay attention. When you look at the notochord, it is actually a mesodermally derived, very important, that is, it is made up of mesoderm. It's a rod-like structure plus it is flexible as well. So it's a rod-like structure, students. It is present on the dorsal side during embryonic development. So let's suppose if an organism is having notochord, they will be chordates. Absence of notochord, non-chordates. So from Porifera to Echinodermata, right? From Porifera to Echinodermata, what do you see? You see that they are non-chordates. They don't have the chordate. They are non-chordates. Then even where do we consider this hemichordata? They are believed to be the connecting link between chordates and non-chordates. Chordates and non-chordates. So they are what? They are the connecting links. Then after that, there is Phylum Chordata. So that Phylum Chordata, right here we know that the notochord is present. Okay. So it is not just the only criteria. There are 4 basic criteria that we say when we say that an organism is a chordate. Just we have to talk about the presence of notochord, presence of dorsal hollow nerve cord, presence of pharyngeal gill slits, presence of the postanal tail. These are the main 4 basic characters: notochord, dorsal hollow nerve cord, your pharyngeal gill slits, postanal tail, that will make a chordate. Just here in this diagram, this rod-like structure is your notochord, and here in this diagram itself, this part is basically dorsal hollow nerve cord, right? That in our case we have this, right? Nerve cord is on the dorsal side and it is hollow. Then this part is basically paired pharyngeal gill slits, right? They are paired. And this part is what? This part is the post-anal tail. So these are the 4 basic characters for somebody to be a chordate. So that's what you have to remember, right, students? So the next thing is basically, so ye dekho, chordates, non-chordates. So even the phylum name I have mentioned. So next is basically on the basis of development we are going to see, right? So we will, circulatory system is already done. So embryonic development we will study the next. Right? So here you will talk about 2 types of organism. I'll discuss that first. जैसे protostomus या protostomes, deuterostomes. Protostomes, deuterostomes. So here what's the trick? You have to remember bad. Bad is the trick here. Bad is the trick here. That is, B stands for blastopore, A stands for anus, D stands for deuterostomes. Is that clear? So that is what you have to remember. Is that clear? That is what you have to remember. So what does it mean? That during embryonic development, it's the blastopore, है ना, that will form anus first. During embryonic development, it's the blastopore that will form the anus first. So then obviously, in the case of protostomes, the blastopore will form the mouth, right? Will form the mouth first. So mouth will form, right? The blastopore is going to form it. So yes, these are the terms which you have to remember. So you can just focus on one thing. You can just focus on this development part here. Is that clear? Done? Any doubts? Any doubt? Sure. So mouth is formed first here. So the trick you can remember, so it's bad. Is that clear? So proto means first, the stome means mouth. So it is the mouth that develops first. And from where? From the blastopore. Is that clear? Is that clear? Right? So in such organisms, just say they are mostly invertebrates. Okay, they are mostly invertebrates. I'm not saying all the invertebrates, mostly invertebrates. And here mostly you will see if they are true coelomates, they will have the schizocoelom. Is that clear? So your Annelida, Arthropoda, Mollusca will come there, right? Annelida, Arthropoda, Mollusca will be the part of this protostome part. Is that clear? Deuterostome mein deutero mane second, right? So here, what's the meaning of deutero? Deutero mane second. So it's not the mouth that form, and stome here is, as I told you, proto mane first, right? And stome mane mouth here. So it is the mouth that will form first, but here mouth will form second, because what will first form? It is the anus that will form first. Is that clear? So your echinoderms and your right chordates, they show this. And this development, mollusca, you will see this is what they have, right? Right? Any doubts? Sure? Sure? So this is all about the basis of classification. So if you guys are having any doubt, do let me know. Done? So yes, oh, how can I miss? So this is hemichordates as well. They will also come in this category. I hope it's done. So this is all about the basis of classification, students. Here you can see what will form first. So next is the classification. Now next part is the porifera, okay? Next is the porifera that we are going to discuss. So you can see this chart as well. So Animalia, multicellular eukaryotes. Multicellular eukaryotes they are, right? So cellular level mein porifera, if it is tissue organ or organ system. When it is tissue plus radial, you know these are the two names. When it is organ plus bilateral plus without body cavity, platyhelminthes. Then organ system is there, right? Bilateral symmetry is there with false cavity, Aschelminthes. Organ system, bilateral symmetry with the true coelom, right? And here what you have to remember when it comes to the symmetry, right? Even if you will check the NCERT, they have clearly mentioned that in Echinodermata, where do you keep it? Because adults are having radial symmetry. Larvae, they show what? Bilateral symmetry. So that's what you have to remember, an important question. Understood? An important question. Okay, so let's start. So diagrams done, tables done. So let's start the porifera. So as I said, beta, I will stick to the NCERT portion here. Okay, there are extra things also that we regularly discuss, even in our batches, or even in the marathon. I have discussed the extra points here, but today we will stick to the NCERT, because this marathon is for the quick revision of all the students, and for them, right, it's better that they understand NCERT properly, right? Okay, done. So porifera mane pore bearing organism. What are they? They are pore bearing organisms, right? You know that initially they were not even placed in kingdom Animalia. They were believed to be the plants. Why? Because they are in the water. They were sedentary, right? Sessile. They are what? They are sedentary. They are sessile. Sessile mane attached to a substratum. Okay, attached to a substratum, mane fixed to a substratum. Are you getting my point? So they were believed to be the plants, but later when their development was studied, right? Later when their development was studied, then we got to know that they are what? They are the animals. Is that clear? Is that clear? So that's what you have to remember. So these are the organisms which are having pores in their body. Understood? These are the organisms which are having pores in their body. So they have different shapes. Firstly, their body is porous, right? Pores they have. So these small pores on the body, the singular is ostium. The singular is ostium. The plural is ostia. Then there is a cavity in between. That's not a true cavity. In the case of true cavity, I'm saying that body wall and gut wall is having a space in between which is lined by mesoderm. There is nothing like that, but they have the spongiocele, and this large pore here is osculum, right? So that will take out water exteriorly, outside. So this is how they look. They have the they have different examples here. So 3 examples are given in the NCERT. So Cycon is given in your book, and I told you that it doesn't show the asymmetrical, it is having the radial symmetry. Then you have this Euspongia, right? And then this is the Spongilla, which is a fresh water sponge. So yes, diagram examples are very important here. So this is Euspongia, which is the fresh water sponge. Okay. So in their case, if you will ask me, firstly, so I told you this, in their case, then you talk about the choanocyte, right? Collar cells. I'm writing the peculiar features here, beta. Water canal system. Water canal system. Very important here. Very important here. So let me show you this, because I have added the diagram. So this is how it looks like. This is how it looks like. Porous body they have. Through ostia, water will come. It will be in the spongiocele, and then it will go out. So as I said here, the very important thing is what? Choanocyte. So look at the diagram here, guys, right? Look at the diagrams. Dekho, I'll be very clear, right? If you will check my marathon, you will see that there are many extra points which are beyond NCERT we have discussed, right? So that you have the better concept clarity. But at this point of time, I'll be very clear that we just have 2 months, right? Literally, we have 2 months. It's 1st of March today, right? And on 3rd of May, you people are going to write your exam, right? So one thing is crystal clear here. One thing is crystal clear here. What is that? That it is the NCERT. If you are struggling with the syllabus completion, especially in the biology, just stick to the NCERT, just cover that. So I'll give you some extra points as well here, but ultimately what you have to revise, just revise the NCERT. That's what you have to do. So just look at the diagram here. Like when I'm taking its section, right? When I'm taking its section, this is how it looks. So outermost layer is there, right, beta? There is an outermost layer, because here you will see this outermost layer is made up of certain cells. You are calling them as pinacocytes, right? In between, you will have some porocytes where you have these ostia, yeah, ostium, right? Inside, you will see these cells, which is a peculiar feature here. These type of cells you are going to see. So what these cells are, these are the choanocytes or the collar cells. They are like this. They are flagellated cells. So do not mention that it is a cilia, it's the flagella. So what is the role of this flagella? This flagella is lining its innermost layer. Just say here, if I am showing you this part, outermost is your layer which is made up of these sort of flat cells. So this layer we call it as pinacoderm. Then innermost layer, students, this is basically your choanoderm. Why choanoderm? Because it is lined with these choanocytes. So both the sides are having this. Look at this. Just say this is the innermost layer which is made up of your choanocytes. This is that outer layer which is made up of your pinacocyte. Now in between this, they have a space. They have this jelly-like part. This space in between these 2 layers, you are calling it as mesenchyme or mesohyl. Right? This is what you are calling it. So look at both the sides. Can you see the cavity in between? So it is not a true coelom. It is not a true coelom. So this cavity actually, students, it is the spongiocele. So yes, it is a question in the paper that it is lined by what? It is lined by choanocytes. It is lined by what? It is lined by choanocytes. So that's what you have to remember. So actually this flagella here is beating, right? This flagella here is beating. This flagella here is beating. This flagella here. So this flagella will keep on beating, So from ostia, the water will enter. This flagella will beat here. They will maintain the water current here. That's how the water will pass outside through this osculum. And this type of setup here, you are using the word water canal system here. What do you call it? You are calling it as water canal system. Water canal system you people will call here. So this is the water canal system that you guys are having here, right? This is the water canal system that you people are having here, and that's what you have to remember, that this water canal system is having different grade, different level in different organisms. This is the one that will maintain water current, and this water current actually is keeping it alive. Through this water itself, the food is also entering, right? The gaseous exchange, the excretion is also taking place. And here you will see these are the choanocytes. They are beating their flagella, maintaining the water flow, and if there are minute particles in that water, that will also enter here. So intracellular digestion is there, then the digestive food will further pass. And in this mesenchyme here, right, in this mesenchyme, mesoglia here, you will see there are some amoebocytes as well, right? Some archaeocytes are also there, which will further form the gametes. And here you will have the spicules also. So that spicules are providing it the protection. So on the basis of the spicule and the fiber, we divide it into we divide porifera into different classes. So if you don't remember the tricks, you can remember pore all your sides. But I believe that in simple ways also you can remember the examples. Is that clear? Is that clear? So this is the diagram of that choanocyte, the collar cell, also the flagellated cell. Do not confuse it with cilia. So it will maintain the water current. So here the food will be digested within the cell. So yes, here I'm going to talk about the intracellular digestion, students. And at its base, what do you have? You have amoebocytes, right? At its base, what do you have? You have amoebocyte as well. Is that clear? Bit electricity issue is there, so please manage for some time. Done. Okay, so that's what you have to remember here. Got it? So these are the real pictures here. So euspongia, bath sponge, freshwater sponge, right? This is the cycon. So it shows the cycon and what is the another one? Leucosolenia. They show radial symmetry. What do they show? They show radial symmetry. Understood? So let's mark it from the NCERT. Let's mark it from the NCERT. So see this. Water is entering, different grades of like how this water canal system is developed, but ultimately you will see the spongiocele is lined by choanocytes. So these are believed to be the filtered chamber. Flagella is beating, the water current is maintained. That's how it will pass further. Okay, chaliye. So let's start. Let's decode the NCERT here. So members of this phylum are commonly known as sponges. We know that, right? They can absorb, even they have that pores in them. So they are generally marine. So if they are generally marine, means not all of them are marine, and they are mostly asymmetrical. So generally marine means we should know the example of freshwater sponge as well. So freshwater sponge is your spongilla. Am I right? Freshwater sponge is your spongilla. That's what you have to remember. Mostly asymmetrical. So yes, where do you see the symmetry? At least one example is there in your syllabus, that is cycon. But other than that, there is one more, leucosolenia. So they have the symmetry. Which symmetry do they have? They have the radial symmetry. Then already you know that from the basis of classification that they are primitive multicellular animals. What are they? They are primitive multicellular animals. Cellular level of organization they have. water transport or water canal system is there, right? I'm writing it here so that you do not confuse it. Water vascular system, it is a different thing. That is in the case of echinoderms. That is in the case of echinoderms. But when I'm saying water canal system, then where do we discuss it? We talk about it in Porifera. So these are two different things. Do not confuse it, right? So water will enter through the minute pores ostia in the body wall. So central cavity here is spongiocele. So water will go out through osculum. Told you. Next is this pathway will help in food gathering. How do we gather the food? Even in the respiratory exchange, even in the removal of waste. So these cells here are very important, which we are calling as choanocytes or the collar cells. They are the one that line the spongioceles. Do solve the PYQs. You will find this as a question. Digestion here is intracellular. Remember that choanocytes, right? With the help of that flagella, even if there is any food that will enter in them. So within the cell, the digestion will be there. So that's why I'm calling it as intracellular. So skeleton is there. It is made up of spicules or spongin fibers. On the basis of that, beta, they are classified into different classes. Is that clear? Is that clear? So on the basis of that, they are classified. Understood? They are classified. Done, beta? So classes of, this is how the classes are also present. Is that clear? Is that clear? Done. So जैसे we have different classes, जैसे if they have the skeleton made up of calcium, then they have different name. If there's spicules, if they are made up of silica, then there will be different class. If they are made up of fiber, then there will be different class. So this is also the basis of classification. So sexes are not separate, so it is hermaphrodite. So multiple times I have repeated the trick BMH. From BMW, you can remember BMH, that is bisexual, that is monoecious, that is hermaphrodite. Hermaphrodite. So they have same meanings. Are you getting my point? They have same meaning. Bisexual, monoecious, hermaphrodite. Okay. So here you will see eggs and sperms are produced by the same individual. That's the meaning here. So they reproduce asexually. Right? Asexual reproduction is also there by fragmentation and sexually also by gamete formation. So in that mesenchyme, you will see the cells which will develop into the gametes. So yes, they show this. So fertilization is internal here, right, within the body, and development is indirect. So indirect development means, what is the meaning of indirect development? That larval stages are present in between. It's not like zygote is not directly developing into adult. Zygote is not directly developing into adults. Got that? Larval stages are present in between. Done, beta? Done? But this larval stages are morphologically distinct, mane different from the adult. So you have cycons, kypha, spongilla, euspongia. One more example I told you, leucosolania. So that's how I'm going to simplify this NCERT for you. So any doubts here? Any doubts here? Yes, please. Any doubts here? Done? So that is what you have to remember here in the case of Porifera. Okay. So next phylum that we have to discuss is your Phylum Cnidaria. Okay. So I'll start with the example here. I'll start with the Hydra. Example of Hydra. Okay, beta. Here there are many important points which you need to keep in your mind. Like I would say the unique points also. There are many, but I'll write the few here. So if they are in your mind, you can easily manage to answer it. But for examples, you literally need to put the efforts. I don't know whether you have seen it or not. Do check this. Sorry. Do check this video, this particular video. Okay, so in this particular video, I have discussed the strategy in detail, right? Here, see, this diversity unit is mentioned, the date is mentioned, right? Even I told you how to remember these examples. So do watch it. Okay, so Phylum Cnidaria, Coelentrata. Here you talk about stinging cells, which are nidoblast या फिर nematoblast या फिर nidocytes. You talk about the alternation of generation here, that is metagenesis. That is what? That is metagenesis, right? So these are two very important points here, right? So here, obviously, if alternation of generation is there, we talk about polyp and medusa. That's how you have to remember it, okay? That is how you have to remember it. Got it? Done, beta? Done? Any doubts here? Okay, sure. So I'm just writing the main points here, right? Here you will also talk about the corals. So let's discuss that, okay? And overall, you know it. Overall, you know it that there will be tissue level of organization, diploblastic they are, right? We discuss about the radial symmetry, even radial symmetries, acoelomates. So these are the points that we already know. So I'll start with the example here. Jaise here, this is the example of Hydra, right? This is the example of Hydra. Actually, here what is happening in this particular phylum, like when I talk about the layers, right? Layers. Obviously, outermost epidermis is there. They are diploblastic. Here you will see in between mesoglia. Now there is the innermost layer. Actually, this phylum will have, it is having a primitive network of, in this particular phylum, you will see primitive network of nerve cells, a kind of diffused network. This is the phylum where for the first time you will see this. This example is very common. We are talking about the Hydra here. You can see that it is sedentary. It is having the tentacles. Their tentacles are actually having that special cells which will help in their defense. These are the stinging cells, right? These are the stinging cells. So these stinging cells are also called as nematoblast, nidoblast, or nidocytes, right? Right, they help in food capturing and all. Now if you look at this, this is the mouth here, right? Below mouth, obviously you will have hypostome. Hypo mane below, stome mane mouth. Then we can see the tentacle here. Actually this form, right, this form here, just let's suppose if I am standing like this, these are my tentacles. So I am the polyp. How do I remember this? This type of form is polyp. How do I remember this? P for polyp, P for plant. This is my way to remember it. P for plants, and plants are fixed. They are attached to a substratum, right? That's how I remember. Now, if instead of it, if I will do like this, right? My mouth, which is here, it will come down. If I will do like this, then it will be medusa, right? This is what you have to remember. So if you look at this diagram, nerve net is present, mesoglia is there. This cavity in between, you can call it as gastrodermis and even the coelentrin. So you can see this epitheliomuscular cells are there outside, even longitudinal muscle fibers are there. See, nidocytes are also there. Some other sensory cells are also there, right? I'm not asking you to remember it, but at least you will get to know the arrangement. Okay, so outermost layer will have epitheliomuscular cells because there are certain muscle cells also which are present there. This is the nidocyte, guys. So in between mesoglia is there, right? Just ectoderm, endoderm, mesoglia. So here you'll see some gland cells and all. So here intra as well as intercellular digestion is there. These are the tentacles which are having this. So this is hypostome, right? Hypostome. This is your mouth. Below mouth, you'll say hypostome is there. Now, the cell that I am talking about, why this cell is that important? This cell is actually helping in their defense mechanism. This cell is helping them to capture the prey, to capture the food as well as to kill the prey as well. That's how it is helping. That's how it is helping. Actually, in this particular cell, you will see, right, there is a stinging capsule. This one is actually the stinging capsule. This one is actually what? This one is the stinging capsule. So this is a stinging cell having what? Having a stinging capsule. So do remember the terminology here, right? Do remember the terminology here. So for stinging capsule, you are using the word nematocyst, nidocyst. Are you getting my point? Nematocyst. Cyst is the word for this capsule. If nematoblast is the word, you're talking about the cell. If nidocyst, yeah, nematocyst is the word, you're talking about the stinging capsule. It is having a thread. So basically, it is going to release what? It is going to release the poison. Is that clear? It is going to release what? It is going to release the poison. It is basically for capturing the prey. Got it? Yes, please. Yes, please. Sure. Right. So this one is the discharged nematocyst. This is discharged. This one is undischarged. So in our NCERT, they have given us the diagram of the discharged nematocyst. And once they are used, they cannot be reused. Further, that cell will be replaced. That's what you have to remember, okay? So this capsule-like structure inside is nematocyst. There is a thread, right? There is a hair on the surface. Okay, so whenever there is any trigger, right, the capsule will burst. The thread will shoot out, will pass the toxin into the prey. Okay, okay. Done? Done, beta? So that toxin is a neurotoxic. This is having a neurotoxic effect, this toxin, and it can paralyze that prey. So that's how it is. These are the important points here. So let's discuss it one by one. Okay, so look at the example here. Just say the inverted structure, right? That umbrella shaped structure is, you know, that medusa, free floating. So your jellyfish is the example there. This is your polyp. So here you will see that some forms are having, just say, this is medusa. So mouth is on the lower side here. This is here, the mouth is like this. So you can imagine this part here, just a mouth. These are the tentacles. If I'll go down, then obviously the position will change, okay? Accordingly. So on the basis of this polyp and medusa, they basically divide the organism. So polyp is what? Polyp is the sessile and the cylindrical form. So here you can see that two types of forms are there, polyp and medusa. So in this phylum, we say there is the alternation of generation, which we are saying metagenesis, which we are calling as metagenesis. So PAM is the trick here. What's the trick here? PAM is the trick here. What is the meaning of PAM? That polyp, it should not be map. If you will call it map, it will be incorrect. So polyp asexually form medusa. Polyp asexually form medusa. Is that clear? Polyp asexually form medusa. Is that clear? This is what PAM is. This is what PAM is. Polyp asexually form medusa. So polyp is the sessile form, students. Medusa is what? Medusa is the mobile form. So asexually it is forming like this, and obviously then medusa will form polyp sexually. So that's what you have to remember here. So alternation of generation is there because sexual and asexual forms are present here. Is that clear? Is that clear? Sexual and asexual forms are present here. So polyp is sessile. It is cylindrical form like Hydra, Adamsia, which is your sea anemone. Your medusa is umbrella shaped. It is free swimming. If it is free swimming, means mobile it is, like your Aurelia or jellyfish. Right? So if both are there, alternation of generation, metagenesis is there. So polyp produce medusae asexually and medusae form polyps sexually. So obelia, right? Obelia, it shows us both the forms. Obelia. Understood? It will show us both the forms. Understood? Any doubts? Done? Any doubts? It will show us both the forms. So this obelia is also referred to as sea fur. So let's understand this part from the NCERT. I hope up to this part all clear. So these are the diagrams. This is Physalia, Portuguese man of war, and this is the trick here. Physics of, okay, this is in Hindi, you'll get confused. Just allow me 1 minute. Okay, so diagram done, and see, this is given in NCERT. So in NCERT, we have the diagram of discharged nematocyst. Means here that capsule is, the stinging capsule is discharged. Is discharged. Right? Is discharged. Okay? So the name is from the stinging cell. So let's understand this part from the NCERT, because the important points I have already discussed with you all. So let's see this. So these are aquatic, mostly marine, sessile or free swimming, radially symmetrical, right? So mostly they are also what? They are marine. Mostly they are what? They are marine. Okay, just a hydra, the example of hydra we have discussed now. So hydra is basically in polyp form, it lives in fresh water. That's how you can remember this. Now, the name Cnidaria is derived from the word nidoblast or nidocytes, right? So they contain the stinging capsules or the nematocyst. So stinging capsule is the nematocyst, right? Stinging capsule is the nematocyst here. So they are present on the tentacles as well as on the body. So they are used for anchorage, defense, and for capturing the prey. So they have tissue level of organization. They are diploblastic. They have central gastrovascular cavity, which is also the coelentrin, with a single opening, mouth on hypostome. Right? So below mouth, what will be there? Hypostome. So obviously in this way also I can explain it, that mouth is on hypostome. Understood? So digestion is extracellular and intracellular both, right? And some of the cnidarians, right, just like corals, they have skeleton which is made up of calcium carbonate. So two basic types are there, polyp and medusa. So polyp is sessile, it is cylindrical like Hydra adamsia, and medusa is umbrella shaped, free swimming like Aurelia or jellyfish. So they exist in both the forms. So alternation of generation is there, beta. So examples and the common name should be clear. Portuguese man of war, Adamsia sea anemone, right? The Penatula sea pen, Gorgonia sea fan, Meandrina brain coral. And Obelia is the sea fur, right? Both forms. Understood? They have both the forms. Got it? Right? Just read it quickly. Yes, please. Yes, please. Quick. Done? Okay. So, beta, you have the different classes on the basis of this. Just I'll give you the example. Class Hydrozoa. It is having both your Obelia as well as Physalia, which is the Portuguese man of war. So, yes, they both have polyp plus medusa in their life cycle. Understood? They have both the things. Yes, please. They have both the things. Is that clear? Right, in this particular class. So we have Scyphozoa as well, where both stages present, but medusa is dominant. Okay. Aurelia is the case. So do remember it. Any doubts? Any doubts? So that jellyfish mein basically that is the medusa form which is dominating. Okay. Okay. Done? Sure? Noted? So let's move to the next phylum. Oh, finally, electricity is here. Thank God. So, just a minute. Okay, so finally, electricity is there. It's done. So, I thought the marathon will be ended. Okay, so let's have a break for 5 minutes and then let's continue. Okay, let's have a break for 5 minutes only. Okay, guys. So let's start the next phylum. Am I audible? Okay, so let's start the next phylum, that is Phylum Ctenophora. Okay, let's start the phylum Ctenophora. So here, beta, if I have to specify the characters, bioluminescence we are going to see. We'll talk about the coloblast or the lasso cells here. Here you will talk about the cilia also, which is used for locomotion by many. And we will see that they are also exclusively marine. That's what we have to discuss here mainly. Is that clear? Yes. That is what we have to discuss here mainly. Okay? Done, beta? Chaliye, let's continue this part, the phylum Ctenophora. So in their case, what sort of organism you are going to see? See, this is what we have here. Just have a look. So ctenophores, they are commonly known as sea walnuts or the comb jellies. Sea walnuts, we also call them as sea walnuts or the comb jellies, and they are exclusively marine. So this data is literally important when we say exclusively marine, right? When we say exclusively marine means no organism will be there in the fresh water. They will only in marine water. They are present only in marine water. Yes or no? So you have phylum Ctenophora, right? You have phylum Echinodermata and the Hemichordata. These are the three phylums, students, right? These are the three phylums in which you people will see that the organisms are exclusively marine. Is that clear? Organisms are exclusively marine. Okay, so radially symmetrical they are, right? Radially symmetrical they are, diploblastic they have tissue level of organization. That's what you have to remember. Now in their case, yes, they do have tentacles. Some organisms, they do have tentacles, they do not have, but it is not the tentacles that are helping them for the movement, right? So even in the lower organism we see that thing. In their case also, you people will see that their body will have 8 rows of cilia. That cilia are arranged like a comb, right? So that's why the body bears, right? The body, this is important, bears eight external rows of ciliated comb plates. 8 external rows. So from this, you have to remember this 8 external rows of ciliated comb plates that will help in locomotion. So yes, on the basis of the tentacle, the presence or the absence, we divide them into different classes, but here for the movement, just like any lower organism, it is the cilia which they are considering. Is that clear? So they are sea walnuts, they are comb jellies, they are even referred to as sea gooseberries. Is that clear? We also call them as sea gooseberries. Understood? So digestion is again both extracellular as well as intracellular. So bioluminescence means when the living organism, they emit light. Okay, so why do they emit light? This is due to the protein luciferin. Luciferin helps in that. Do you know that? Luciferin protein helps in that, right? Yes or no? It is the luciferin protein that helps in that. Got it? Got it? Yes or no? Yes or no? Sure? Done, beta? Done. So it is the luciferin protein that helps in the This is a luciferin protein that helps in the bioluminescence, so it will react with oxygen. So there is a presence of an enzyme luciferase, so that helps in emitting the light. So that's what you can remember. Okay, so sexes are not separate here, only sexual reproduction is there. So this is what you need to highlight, where they have specified the thing. So fertilization again external, in Porifera it was internal, indirect development is still there. So pleurobrachia and Ctenoplana is the example. Got it? There are other examples also, but as I said that let's stick to the NCERT part here. Got it? Right? Read it. If there is any doubt, then do let me know. Done? Any doubts? Sure? Okay, so this one, this pleurobrachia is actually referred to as your sea gooseberry because it looks like, its structure looks like that. Do remember it. Chale, next, so this is the tentacle. So it is not the tentacle that is helping in the movement. So Ctenoplana, pleurobrachia is there. And the next thing that we have to discuss is the platyhelminthes. So here they are the flatworms. Right, here they are what? They are the flatworms. So here we will discuss about the flame cells, very important, right? Very important. We have to talk about the flame cells, also known as solenocytes, also known as protonephridia. Okay, protonephridia. And yes, how can I forget? Just here I told you about the coloblast cells, right? Coloblast cells. Ya fir they are lasso cells. Coloblast cells, or what else do we call them? We call them as the lasso cells, beta. So they are present in their case, yes or no? So they are basically, they are present on the tentacles that help in capturing the prey, right? They are present on tentacles and help in capturing of prey. Okay, so yeah, here also we see the example in which you will also find the nidoblast. Okay, there is one example there. But okay, now come to this. So here you'll talk about the flame cells, solenocytes or protonephridia. So first of all, have a look. So their body is going to be dorsoventrally flat. What sort of body do they have? Dorsoventrally flat body do they do have, and they are mostly endoparasite. I'm not saying all of them are endoparasite, but mostly they are, right? Mostly they are. So let's talk about it. So as I said, they have what sort of body? That's why you are calling them as flatworms as well. Right, that is why you are also calling them as what? You're calling them as flatworm as well, because their body is dorsoventrally flat, right? And they are mostly endoparasite. So if they are mostly endoparasite, it means they are not, all of them are not the endoparasite, right? All of them are not the endoparasite. Mostly they are endoparasite. So that's what you have to remember. Some are free living as well. Is that clear? Some are what? Some are free living as well. Not all of them. Not all of them are what? Not all of them are basically your endoparasite. So endo means inside a living body, inside a host they are going to live. Yes or no? Inside the host they are going to live. So if I talk about the free living one, I have the example of planaria, which is also known as Dagesia. So this one is believed to be the free living one. It lives in the fresh water. It is even the omnivorous, right? Right, so here you will see in it, there is great power of regeneration as well. In its case, you will also see there is great power of regeneration as well, right? So they are bilaterally symmetrical, triploblastic, acoelomate animals. Organ level of organization is there. So now because they have to live within the host body, so you know that a parasite is having the parasitic adaptation. Just they have hooks, they have suckers. They don't have digestive system because they are getting the pre-digested food. They just have to make sure that they are able to stick to the host system there. So they have hooks and suckers, absorb nutrients directly from the host. So that is why they do not have the respiratory system. So flame cells are present here, right? Flame cells are present here, which is very important thing. I told you already. So the another names are also important, which are solenocytes and Protonephridia. You cannot miss these names as well. So it helps in what? You can see that. So it will help in the excretion as well as osmoregulation. So osmoregulation, you know that. What is osmoregulation? To maintain the salt and water balance, right? Osmoregulation is what? To maintain salt and water balance. Is that clear? To maintain salt and water balance. So that is how it is helping. This is important part. Again, they are also the hermaphrodite. Internal fertilization is there. So while writing the short notes, you make sure, right, you write the phylum name, the internal external fertilization, even digestion is given. You have to remember this. So planaria, which is also referred to as Dagasia, which is having the ladder-like nervous system. They have high regeneration capacity. Taenia, tapeworm, right, where we have seen pseudometamerism. Right, where what do we have seen? What we have seen? Pseudometamerism. So firstly, I'll give you the example here. Then one extra point is there. So just look at this. So it is the planaria. Even in the chapter living word, it is given. So planaria is having great power of regeneration, right? It is having great power of regeneration. And in its case, the pharynx can be everted. Are you getting my point? Pharynx can be everted in its case. Look at it. Right, they don't have, like here also, for the very first time in Cnidaria, we have seen the diffuse network of nerve cell. Here also, if you will notice beta, you will see these are the longitudinal. These are the longitudinal nerve cords are there. In between, you have transverse nerve cords. Can you see that? Right, in between, you have transverse nerve cords. Right, you have transverse nerve cords, right? So this is the phylum cell. This is the phylum cell which is serving the purpose here. Got it? This is the phylum cell which is serving the purpose here. Is that clear? Sure? Okay. So in this case, pharynx can be everted, which means it can be turned inside out or turned outward. Understood? Right? Inside out or turn outward. This is what we will see in this case. Understood? Yes? Sure? Sure? Right? So another example is of schistosoma. Here, this example is not given, but I want you people to know this. Schistosoma, which is also known as blood fluke, right? Which is also known as blood fluke. Beta, in this case, though here we see the sexual dimorphism, we don't see the sexual dimorphism, we see the hermaphrodite body, but what you have to remember that here they are unisexual. There is a large male, beta, right? That large male which carry the female in its canal. Okay, so schistosoma blood fluke is the example which is unisexual here. This is what you should remember. So here male, large male, here male is not small, male is large, large male carries female, large male carries female in its gynecophorial canal. Understood? Understood? Any doubts? So gynecophoric canal it is going to carry the female, so sexual dimorphism is seen here. Is that clear? So the examples should be clear. When you look at the example, you should know which character is it showing. So you already know that, the pseudometamerism. That pseudometamerism. Okay. So next is the Aschelminthes. So next is the Aschelminthes, but so any doubts so far? Any doubts so far? So nervous system is ladder-like here. Nervous system is ladder-like here. Okay. Okay. So next is what? Next is the Aschelminthes that we have to discuss. Here you talk about the roundworms, and in the case of Aschelminthes, right, what extra do we need to study? Like what are the points which we need to focus? Let me tell you the unique points here. So here, first of all, they are the roundworms. Okay? When we get their cross section, we see the round structure here. In their case, you will talk about the, it is not given in NCERT, you should know this. H-shaped rennet cells are present here for the excretion. Here you will see the first phylum which shows the sexual dimorphism, right? It's the first phylum which shows the sexual dimorphism. Keep the exception of Aschelminthes in your mind, right? So H-shaped rennet cells are there. You'll see the large female and small male here, right? Usually small male here. And there is also here, we will also talk about the hydrostatic skeleton. Like there is a fluid in the body that is going to provide the structural stability to that. Is that clear? Is that clear? Sure. So they are also referred to as nemathelminths. So they even infect plants. Okay, they even infect plants. So body is circular in cross section. That's why the name roundworm is there. Maybe free living, aquatic and terrestrial, or they are parasitic in plants as well as in animals. Is that clear? Parasitic in plants as well as in animal. Is that clear? Is that clear? Yes. So if I have to talk about their body wall, in their case, students, yes, you will see the cuticle is present and epidermis is there, right? And below epidermis, longitudinal muscles, right? Longitudinal muscles are also present. Longitudinal muscles are also present. Only longitudinal, you're not talking about the circular here. This one is non-living. This one is going to be rigid. So it will protect them from the host immune system. Got it? So outermost is non-living. It is thick. It will protect them from the host system, right? When they are even in the digestive system, they are going to, they will be protected from that. So epidermis is also there. And other than that, what do they have? They have the longitudinal muscles. Is that clear? Is that clear? And overall, we have already discussed that they are the pseudocoelomates. They have the mesodermal pouches. What do they have, students? They have the mesodermal pouches. So yes, they have organ system, bilateral symmetry, triploblastic, pseudocoelomates. Alimentary canal is complete. So if it is complete, so here you will see tube within tube body plan, right? What will you see? Tube within tube body plan, and here you will also see there is a muscular pharynx. That is also you have to remember. What is there? There is a muscular pharynx in their case. Is that clear? So an excretory tube removes the body, remove the waste from the body cavity through the excretory pore. So sexual dimorphism is there. They are unisexual. They are dioecious. Males and females are distinct. Often females are longer than the male. Fertilization here is also indirect, and internal fertilization is there. Is that clear? Is that clear? Right? So here in this case, the skeleton is absent, of course, right? Skeleton is absent, but a fluid is there in pseudocoelom that provide a shape. So that's why I told you they will have this hydroskeleton. They will have what? They will have this hydroskeleton. Just ascaris, roundworm. Even in the chapter Human Health and Disease, we have covered this ascariasis, it will call. And it is monogenetic, means it is having the single host. Beta Wicheria vencrofte. We have discussed the Wicheria as well, filarial worm. We say it is viviparous, it will give birth to the young ones. And it is the female female Culex mosquito, that is the vector here. Then comes the hookworm. So it is not given in NCERT, but yeah, one should know this, right? So this part is clear. So they have H-shaped rennet cells. They have H-shaped rennet cells. We'll show you the picture as well, which are meant for this excretion and osmoregulation. Am I clear? Which are meant for this excretion and osmoregulation. Understood? Which are meant for the excretion and this osmoregulation. So they have, so actually this H-shaped rennet cells are having this excretory tube, right beta? They have the excretory canals which will remove the waste through that excretory pore. Is that clear? Sure? Sure. So nervous system mein nerve ring and longitudinal nerve cords will be there. Longitudinal cords are present. Is that clear? So other sensory organs, right? Other sensory organs are also there in their case. Jaise ki you will see papilla, right? Which are basically the tango receptors. Are you getting my point? Papilla are there, which are basically the tango receptors for touch. You will have the amphids there, which are the chemoreceptors. Got that, beta? And other than that, you will also see the phasmids, which are again the chemoreceptors, right, beta? So this one is basically on the lip, right? They are on the tail. So yes, they do have these receptors, right? They do have these receptors. Got it? So amphids are there, phasmids are there. Got it? Yes. Sure. So which area is the filarial worm, which is viviparous, having two hosts, so it is digenetic. Okay. Okay. So this filarial worm, even in that chapter also we have discussed it. So this which area? It causes filariasis or elephantiasis. Filariasis or elephantiasis. Understood? Filariasis or elephantiasis. Understood? Sure? Done, beta? Okay, so this is Cyclostoma is the hookworm. This one lives in small intestine. Cyclostomiasis. Bas, theek hai, done. Next is the phylum. So this is the diagram. You should know about it. Next is Annelida. Annulus, mane rings. We know that this is the phylum where for the very first time you will see metamerism. And here we have open circulatory system. And here they are, right, eucoelomates. That is, they are true coelomates, right? And in their case, you will see this is schizocoelom. This is schizocoelom. Is that clear? Is that clear? So they live in marine and the fresh water. They are free living, sometimes parasitic as well. They exhibit organ system level, bilateral triploblastic and this. And here for excretion, we will talk about the nephridia, not protonephridia. This is nephridia that you are going to discuss here, right? That you are going to discuss here. So three examples we discuss in their case. We talk about the leech, we talk about the earthworm, and Nereis. This is a scientific name, of course. So here, these two, they are again hermaphrodite. These two are again hermaphrodite, and this one will show sexual dimorphism, and here you will see that it is dioecious. So yes, they can ask you this example. So hirudin leech is hirudinaria because from it we get hirudin. This is the natural anti-blood coagulant, blood coagulant. Is that clear? So it is having suckers to suck the blood. So this hirudinaria that we have to discuss, it feeds on blood meal. Do you know that? They have the suckers. It feeds on blood meal. So if any organisms, it feeds on blood. If any organisms, it feeds on blood. So it's sanguivorous. What's that? It's sanguivorous. Understood? It's sanguivorous. Just have a look. Okay. Done? So true segmentation we will see here, right? So externally you will see that they have the annulus, right? They have the annulus, yeah, grooves, right? And internal segmentation, septum is there. Septum is there. And nephridia, obviously there is the repetition of that, right? There is the repetition of that. Firstly, look at the diagrams and then I will discuss it in detail. So this part is clear. This part is clear. Sure, beta? This part is clear. So you have this earthworm, right? Do not focus on other examples. Hiriodinaria, Nereis is there. So this Nereis that you are talking, it is also referred to as sandworm, right? It is also referred to as sandworm. And here, beta, syphilization is present, right? This is again the another term which you have to understand. Syphilization is present. What is the meaning of syphilization? Syphil is the word, mane, associated with head. So if I am saying syphilization, mane head formation. What do we have here? Head formation is there. Got it? Right? So in the phylum Annelida, you will see both longitudinal as well as circulatory muscle. We also discuss the setae here, chitinous S-shaped setae here, and the muscles in locomotion, which we will cover one by one. So this is your Nereis. If you will read its reproductive cycle, you will be amazed to see that, right? They have parapodia, they are the unisexual one, right? So yes, stick to the diagrams, which stick to the examples which are given in NCERT. So this is what you know, right? And then the Hiriodinaria is there, freshwater leech it is, right? It is bisexual, okay? Right, firstly, I'll tell you what parapodia and all is if I have added the diagram. So earthworm and leeches, they are monoecious, reproduction is sexual. Nereis is the dioecious. So Nereis mein na beta, here you will see your body is having na parapodia. This is the structure which is meant for locomotion, in their case. Earthworm mein you will see S-shaped chitinous setae. In leeches, both are absent. Muscles will help in the movement here. And in this earthworm, this diagram is clitellum. Right? First, just have a look. Done? So even in your NCERT, please write it, please mark it. Quick. Please write it, please mark it. Quick. Do mark it, students. Done, beta? So this is the clitellum, if you will see, right? It is basically the glandular part, okay? It is somewhere around segment 12 to 16, okay? Present in region 12 to 16. So help in even the cocoon formation for egg, and during reproduction it also forms the mucus. So these are some special points which people should remember, okay? So glandular 12, I think 14 to 16 segment. 14 to 16 segment. I'll cross check it again. So glandular 14 to 16 segment, help in cocoon formation, releases the mucus during the sexual intercourse. So that's how it helps. So in the clitellum, basically earthworm, it is monoecious, but generally they prefer the this cross fertilization, okay? Right? Oh, okay. So in their case, the in their case, they have the closed circulatory system. I think initially I made a mistake here. Let me check this, because open is in the Arthropoda. Arthropoda, they do not have, just 1 minute. Arthropoda, they do not have this thing, beta. They do not have, without any exception, they have the open circulatory system. They don't have the closed blood vessels. Let me check. I think here I have coelomates. Let me check. Triploblastic, body plan, tube within tube. Yes, open here. So closed will be there in your Annelida. So for the first time you will see it in Annelida, closed, and here again in Arthropoda, we see the open circulatory system. I thought I have mentioned, I didn't mention it, but nahi, I have mentioned it. Okay, so this, so I hope you have done this correction. So let's move to the next part. And see this excretory pore thing in the case of Annelida, terminal duct, right? And then H-shaped rennet cells, done. Phylum Annelida, yes. So let's discuss it. Look at their bodies. So they have the closed circulatory system. Okay, so in Nereis you will see proper segmentation. Even in the case of Nereis you will see cephalization, that is head formation is there. Okay, and they have, what do they have for locomotion, for movement? It is parapodia. This is the lateral appendage. Very important point. And here you will see, in the case of leech, in the case of Hemichordata, we are going to see the suckers. Okay, right? Yes, please. Understood? Done, beta. So they have two suckers. One is interior, another is posterior. Okay, right? So two suckers are present here. So interior and posterior. So they may be aquatic, that is marine and freshwater, or terrestrial, right? Or free living, and sometimes parasitic. You know the example of parasitic. So organ system level of organization, bilateral symmetry, triploblastic, metamerically segmented, and coelomate animals, right? And coelomate animals. So their body surface is distinctly marked into segments or metamers. So as the name, Annelida. So Latin means, here, what is the meaning of this? So in Latin, annulus means little rings. That's what you have to remember. So they have longitudinal as well as circular muscles. So I told you in Phylum Aschelminthes, we just talked about the longitudinal muscles, but here you have longitudinal as well as circular muscle. Longitudinal as well as circular muscles that will help in locomotion. But in Nereis, we will have, you know, oar-like structure, parapodia, oar-like structure, parapodia, that will help in swimming. So they have closed circulatory system. Nephridia is meant for the osmoregulation and excretion. But a neural system will have paired ganglia, right, connected by the lateral nerves. Now see, they are chordates or non-chordates? They are non-chordates. So in their case, what will you see? You will see that the nerve cord will not be on the dorsal side, rather it will be on the ventral side. So first of all, in their neural system, what have you seen? You have seen paired ganglia, right? What have you seen? You have seen paired ganglia. That's the number one thing that you have to remember. And this is going to be ventral. Just we have single dorsal hollow nerve cord. They will have double, right? This is not single, so they will have double ventral, right? Solid nerve cord. So if in any non-chordate, if there is nerve cord, that's going to be the condition. Is that clear? Is that clear? So that's what you have to remember here, right? That's what you have to remember here. It is important. Got it? Right? Yes, please. Is that clear? Right? Sure? Okay, so ganglia, you know that it is the group of nerve cells, है ना? मतलब basically, especially the cell body together is forming the ganglia here. So do remember this. So parapodia, lateral appendages, they are on the sides, they are fleshy appendages, they are in Nereis. So in the earthworm, what do you see for the movement? You talk about the S-shaped chitinous setae. Do write it. S-shaped chitinous setae. Got it? So cuticle, epidermis, muscle layer, both are there, right? So, but here you know that which muscles do we have? Okay? Okay? Guys, any doubt here? Sure? Right? So this is what they have. Any doubts? Any doubts? So you should know which one is dioecious, which one is monoecious. Okay, so muscles, which muscles do they have? So overall, yes, here you will see outer cuticle is there, you will see epidermis is there, and you are going to see the muscles as well. But I would like to specify one thing here, students, when it comes to muscles, right? Just say we have inner circular, outer longitudinal. In their case, outer one will be circular, inner one will be longitudinal. जैसे in our case we have ICOL, that is inner is circular, outer is longitudinal, but in their case outer is going to be circular and inner is going to be longitudinal. So do write it, okay? Do write it. So this is all about it. So I believe up to this part all clear, है ना? So sexual reproduction is there. Okay, laterally parapodias are present. 2 suckers they have. They don't have 1 sucker in Hemichordata. 2 suckers are there. Okay, so next is the phylum Arthropoda. There are many examples that we have to discuss there, right? So in the case of phylum Arthropoda mane jointed appendages, right? What's that? It means jointed appendages are present. Yes or no? Jointed appendages are present. Yes or no? Yes or no? Right? So Arthropoda, you know that out of 10 animals on this planet Earth, 7 are insect. They are that widespread. In their case, there are many points that we will see, but here mainly you will see external segmentation. Here you will see chitinous exoskeleton. That is meant for what? That is meant for protection, right? Chitinous exoskeleton is there. Without any exception, we will see open circulatory system, right? Right? We literally should know the, just in cockroach, you remember? Cockroach is the example of arthropoda only. We have what? We have compound eyes, है ना? Compound eye, and even the simple eye are there. Compound eye plus simple eye, which is also known as ocelli, ocellus, plural. So compound eye gives the mosaic vision. So here in this particular phylum, you will see, I'm just writing the main points here. You will see this is the phylum where you will see a proper respiratory system. Just even in the Annelida, in Arthropoda, right? If I talk about this particular phylum, that is Annelida. So in Annelida, right, we will see that respiration is basically through what? Through skin also. Moist skin, like an earthworm. So skin is there. We can say it is the cutaneous respiration, yes or no? What can we say? We can say that there is the cutaneous respiration, yes or no? Right, even gills are also present. But here you will see a proper, this is the phylum where we are going to see a proper respiratory system for the first time. Are you getting my point? Right, so here you will see that there will be the network of tubes, that will make tracheal system, that we will see in the insects, right? Tracheal system we will see in the insect. Network of tubes, basically tubes and tubules, basically, right? Even other than that, you will see gills, right? I would say book gills, book lungs. These are the names that You have to remember. So yeah, this is the phylum where for the very first time you are going to see what? Where for the very first time you are going to see the respiratory, proper respiratory organ. So gills we will see in the case of prawn. This one you will see in the case of limulus, that is king crab, which is even a living fossil, right? Which is even a living fossil. And here, book lungs you will see in the case of scorpions, okay? So this is what we see. Even in the case of excretory system here, I'm just writing the main features. Excretory system, you'll see Malpighian tubules, coxal glands, green glands. So yes, we do have the variety here. That's why it is the like degree of protection and all. You know that it is the largest phylum of animal kingdom. There are more points related to it, right? There are more related points as well. I'm just writing the points from where you can get a direct question. Okay, done, beta? So green glands, coxal glands, spiders, scorpions. So Malpighian tubules are also there, okay, which are in the insects. Got that? Right? Yes or no? So in their case, we will see there is high degree of syphilization. High degree of syphilization, that is the head formation. Okay, high degree of syphilization will be there. Got it? Done, bache? Right? So segmentation, of course, we'll see their digestive system, of course, will be complete. So body cavity, in their case, जैसे हम देखो heart, if present, we have on the ventral side. Their case, it will be on the dorsal side, right? Nerve cord once again, it is going to be ventral, double, paired. Done? That's how you will see it. They have the different functions. So circulatory system, as I told you, this is going to be open. So here you will talk about the hemolymph. Just in insects, we talk about the hemolymph, right? So in their case, let's say the heart is on the dorsal side, right? And the open circulatory system is there. Heart is on the dorsal side. It is going to pump the blood. So this is the hemocoel, right? This is the hemocoel. Body organs are going to bathe there, right? So here you will see in the body there are different types of cavities. We use the word sinuses for that. Just pericardial, perineural sinuses are there. So this cavity here is, right, hemocoel having, just in insects and cockroach we discuss about the hemolymph, right, which is white, yeah, colorless. It doesn't have the RBC, play no role in oxygen transport. So in the case of insects as well, students, you will see, just if I take the example of cockroach, you know that the body is divided into head. Body is divided into segments, head, thorax, and abdomen we see. And in some cases, you will see if head and thorax is fused, right, then what is the word that we have to use, beta? We will call it as cephalothorax, because head and thorax is fused. Okay, so in the case of cockroach, if I have to give you the example, you'll see that thorax, right, even in the abdomen, 2 pairs in thorax plus 8 pairs in abdomen, total 10 pairs spiracles are present. Matlab here in the case of spiracles are present. So basically these are what? These are compared to our these nostrils through which the air will enter. So yes, in the case of insect, that's how you will see the air will enter, right? Then air will pass through the network of tubes, tubules, and finally the oxygen will reach there and the exchange will be there. So yeah, it is diffusion only. There is no doubt in that. There is diffusion only. The blood is playing no role in the transport of oxygen. So that is how it is present here. Is that clear? Is that clear, beta? right? So blood is playing no role here in the gaseous exchange mostly, and they are oviparous. So these are the points. So baki beta, in their case, you will see the vectors, they have the economic importance as well. So if we will dive deep into the discussion, there are many things that we can discuss. But yeah, I would like to specify a few points here. Just I told you, you will see the compound eyes, right, which are made up of, because compounding word is there, which are made up of around 2000 hexagonal ommatidia, single subunit. So each subunit will make the image here, right? So resolution is not that good, but yeah, they'll have the mosaic vision. So they have simple eyes, ocelli as well. That is just like a photoreceptor that will tell dark or light, but otherwise image formation will be done by this. Okay, so they are the gregarious also. So we will discuss each and every point here. So let's start directly from the NCERT. So as I told you already that it is the largest phylum, right? And the largest class here is Insecta. Insecta, right? Having 3 paired legs, hai na? Yes or no? Yes or no? So next is the over 2/3 of all named species on Earth are arthropods. So they have organ system level of organization, right? They are bilaterally symmetrical, triploblastic coelomates. So chitinous exoskeleton they have. So they consist of head, thorax and abdomen. Head, thorax and abdomen. Jointed appendages are present here. Respiratory organs में gills, book lungs or tracheal system is there, right? So again, once again, let me mention the examples here. Just again, though already I told you, but still I'm mentioning it here. So book gills, you know that. Limulus, that is your king crab, that is your living fossil. So when we are saying something is a living fossil, means like it shows the character. Like in 2 ways you can define it. It shows the character. It shows the character just like the fossilized organism, or you are saying that they are not evolving. They have, right, primitive characters. That's how you can define them. So you have book lungs as well. And tracheal system. You see in the insects, told you. So circulatory system, open type. Yes, they do have antennae, sense organ. Eyes are there, compound and simple eye. So simple eye will just detect the light, nothing else. So simple eye, ocellus. We also use the word ocellus here, ocelli for that, right? Right. So compound eye is basically for what? This compound eye is actually for the mosaic vision. Am I clear? This compound eye is actually for what? It is basically for the mosaic vision. Am I clear? So excretion is, and yeah, statocysts. It is a balancing organ. Do underline it. So excretion is through Malpighian tubules. So this Malpighian tubules, where do you see it? So other examples are not mentioned. They are important. They are in the insects at the junction of midgut and hindgut. And hindgut. Got that? Fertilization, usually internal. Sexual dimorphism is there, mostly oviparous, direct or indirect development. So you have apis, honeybee, right? Apiculturing, silkworm, right? Lessiferous, which will give you lac. So anopheles that spread diseases, your malaria, culex, which area causes filariasis, Aedes is also there, right? Then gregarious pest. Gregarious in Hindi means milinsar. Basically, these pests, right, they show the swarms. They are in groups. They say locust is there, locusta. The living fossil is limulus. That is your king crab. Is that clear? Right? So they didn't mention the other excretory organs, but yes, I have already mentioned. They say you have coxal glands, green glands, which are also referred to as antennary glands. In the case of prawn, you should know this. Okay? You should know this. Okay? Yes, please. So, yes, in arthropoda, जैसे we used to read na cockroach. So, cockroach में beta, in digestive system, you will see crop and gizzard, which is also known as proventriculus. You also see that in the case of your birds. So gizzard is G for grinder. So this is for the storage of the food and this is for its grinding. Okay, so yes, once there was a question that out of the following, which one will show crop and gizzard in their digestive system? So that is how it is, okay? Done, beta? So look at the diagrams here, right? And then you should know about it, okay? Done? Have a look. Sure. So this is Arthropoda. Okay. Okay. Okay, so let's talk about the next phylum, which is your phylum Mollusca. Got it? The second largest phylum, okay? So again, you have the trick here that people of Paris save light current on all days. So you can see the different examples here. So before going forward, let me write the characters here also. So phylum is Mollusca, which is the second largest phylum of animal kingdom, right? What are the special things that you will see here? You are going to talk about the shells here. You're going to talk about the mantle here. You're going to talk about the foot, muscular foot, viscera. Chalo, already told you about the mantle here. And here we will talk about, these are the terms, these are the words that you should know. Cnidia. You'll talk about radula there. Hai Cnidia is there, radula is there. So these are the terms, these are the words that we will discuss. Or right, so these are the terms, these are the words that we are going to discuss here. Is that clear? Is that clear? So I'm sticking to the NCERT wherever it is required. There only I'm just discussing the extra points. So I hope you people are clear here, okay? Right? So that's what you have to remember, phylum Mollusca, which is the second largest phylum, beta. So ultimately, these organisms are what? They are soft-bodied and shelled organism. They have the shells and their bodies are soft, okay? Their bodies are soft, okay? Done, beta? So in the cases of mollusca, and yeah, how can I forget to mention torsion? This is also the word that we need to know in their case. I'll add it if there will be anything except anything extra. So overall, your mollusca are what? Overall, when you talk about the mollusca, they show bilateral symmetry. They show bilateral symmetry, but in their case, there is one more point. Just say in some classes, you will see what will happen. In mollusca, normally they show the bilateral symmetry, but some secondarily they become asymmetrical. Do you know that? Right? Few are secondarily asymmetrical. So what is the meaning of this particular line? Few are secondarily asymmetrical, means they are not asymmetrical actually. They were showing the symmetry, but in their case, torsion will occur, right, beta? Right? Just say you have the example of snail here. So why is it so? Due to torsion. right? I'll show you the example and the diagram as well. So just have a look. This is the snail, okay? In this particular snail, this is the shell that they have. We'll discuss each and everything. This is the shell they have. Now they have to fit in all the body organs here, right? They have to do what? They have to do what? They have to fit in all the body organ there. Yes or no? Yes or no? Yes or no? Done. So they have to fit in. So in their case, there is a twisting, that is torsion. It is a twisting actually of the body organs. So it is it is at 180-degree anti-clockwise twisting of visceral organs, shell, mantle, right? So it is the twisting. Are you getting my point? So it's that 180-degree anti-clockwise twisting of that visceral organs, yeah, visceral mass, the mantle, the shell, right? So that will result in being asymmetrical. So when will it occur? During embryonic stages, right? During embryonic stages of gastropods. So when I'm saying gastropods, so gastropods, this is actually, right, gastropoda. Just say insecta is the largest class in the phylum Arthropoda. So this is the largest class of mollusca beta. Here you will see this torsion. Are you getting my point? So these are the gastropods. So here you will see torsion. Here you have the example, you have the pila, which is also referred to as apple snail, and that's what you have to remember. And overall, they are also the opens, they have the open circulatory system, but there is one more class here, class Cephalopoda, where octopus is also there. There you will see closed circulatory system, a very important point. Otherwise, all will have open circulatory system. Is that clear? Done, beta? So visceral mass, mantle, and the shell will rotate, right? So mantle will, so here mantle, ultimately this will shift. This will shift basically. So its shifting is done. So that's why they will become asymmetrical. So it will shift basically from posterior to anterior, right? Posterior to anterior, it will shift. So that is how it works. Is that clear? Is that clear? So you will see the torsion, right? But so in the largest class here, okay, so it will be there in the during the embryonic development, yeah, during the larval stages basically. Okay, okay. Done, beta? So you look at the organs here, look at the arrangement. Just here they are showing the Cnidia, you know, thin filamentous gills-like structure, which are along with respiration playing role in the excretion. Right here, you will see the rasping radula organ, which is just like a grater here in their case, right, which is having the row of teeth on it. So look at the arrangement here. You will see the pulmonary sac-like structure also for the exchange. Overall, you will see the muscular foot and the mantle. So this is what we have to see. So class Cephalopoda is the class with closed circulatory system. It is the most advanced class in their case, beta. And Gastropoda will show you the torsion. And moreover, you will see that some organisms will have two shells externally. So two shells wale, where will you put them? They are bivalvia because there are two shells, and the example is pink tada. The pink tada is having this, so it is the pearl oyster. It will give us the pearl. That is again a PYQ, okay? So now let's discuss it. So you can just have a look at the arrangement of the soft body organs, posterior side, dorsal side, interior side. Look at the gills there, muscular foot, adductor muscle, there is a hinge. So it is showing that bivalve structure. So let's discuss about it. Is that clear? Is that clear? Right? So hey, here, can I say that body is unsegmented? Can I say so? Body is unsegmented. What kind of body is there? Body is unsegmented with varied shapes and is with varied shapes and is covered by shells. So body is unsegmented here. What sort of body is there? Unsegmented. But yes, here we have an example of Neopilina, which is a segmented mollusk. Which is basically considered as a connecting link between Annelida and Mollusca. Is that clear? Which is considered as a connecting link between Annelida and Mollusca, and also a living fossil. Okay. Also a living fossil. So in their case, so body is divisible into, right? How their body is divisible. So you will see that their body is divisible into head, dorsal visceral hump या mass, and muscular foot. This is what they have. And muscular foot. Is that clear? So head will have obviously sensory organs. Just you will see sensory tentacles here, right? Yes, they do have. If you have, see, if you notice the snail, and even you will also see the eyes here, right? You will also see the eyes here. Is that clear? Is that clear? Sure? Sure. So visceral hump mane contain all visceral organs, all body organs. And when it is the muscular foot, so you know that it is present on the ventral side, and it is meant for what? It is meant for locomotion. Am I clear? It is meant for what? It is meant for locomotion. Got it? Got it? Right? So there is a soft and spongy layer of skin which will form a, right? There is a soft or spongy layer of skin that forms mantle, which is very important here. And it will be over the visceral organs. Is that clear? So this is the mantle, which is basically where it is over visceral mass, your visceral hump. Okay, so this is the mantle, and this is basically, you can also call it as pallium. So that's what you have to see here. Any doubts? That's what you have to see here. Yeah, it is quite easy to understand as well. Okay. Okay. Done, beta? Sure? Right? So now just imagine that if there is, this is the visceral hump, okay? So visceral hump, and let's say this is that soft spongy layer, this is that mantle. So the space in between these two, it is your mantle cavity. This one is that mantle cavity. And this mantle releases shell, which is made up of calcium carbonate and conchiolin protein. So it is the one that form what? A shell. Okay. So shell can be internal, external, or it can be absent as well. We'll talk about it. Okay. we will talk about it. Is that clear? Any doubts here? Any doubt here? So digestive system, if you will talk about digestive system, it is going to be complete, and they have radula, right? Not all will have radula. So radula is just like a grater beta. It is mentioned as file-like rasping organ, right? It is having rows of chitinous teeth. So this is the file-like rasping organ. Rasping organ, matlab you can consider it like a grater. So it is in the buccal cavity. Where do you find it? Obviously in the buccal cavity. Understood? Obviously in the buccal cavity you'll find it. Understood? Sure? Any doubts? Beta, any doubts here? Okay, so we have in our syllabus the example loligo, in which you will see radula is absent. You can underline this part, right? In the loligo, we will see that the radula is absent. Got it? That the radula is absent. Understood? Sure? Sure? Okay, so this then we have Cnidia as well, matlab cnidium. For excretion there is organ of Bojanus also, Cnidia. So this one, it is again important beta for respiration as well as excretion. That is what you have to remember. Okay, this Cnidia. And here anus opens into mantle cavity. That's also you can remember. Where will it open? It opens into the mantle cavity. Am I clear? It opens into what? It opens into the mantle cavity. Got it? Sure? Done beta? So gills, Cnidia is there, right? And in the case of your apple snail, right? Which one is the apple snail? Apple snail is your pila. Which one is apple snail? That is pila. So in case of pila, you will also see there is a pulmonary sac that help in respiration on land. Okay? Done beta? So this information which I'm mentioning here, it is very important, right? It's very important. Okay? Done? Done beta? Okay, so circulatory system, as I told you already, right? Once again, I'm repeating it. Open. It's the class Cephalopoda, which is even the most advanced class, where you will see closed circulatory system. So how to remember the example here? Loss. Focus on closed. Loss here. L. Loligo. No radula. Octopus, also known as devilfish. It's a false fish. Why? Because true fishes, they belong to Pisces. Then comes squid. They have, right? They have the, yes please. So, sorry, not the squid, sepia, my bad. Cuttlefish it is. So they have the closed circulatory system. Okay? They have the closed circulatory system. Okay? Done? Any doubts? Any doubts? Done? So beta loligo mein shell is internal and reduced. Shell is internal as well as reduced. Here there is no shell, right? And 8 arms. In the octopus you will see 8 arms. And in the case of sepia, the shell is internal. And here, how can you differentiate the sepia and this octopus? Here you will see 10 arms are there, right? 10 arms are there. Got it? So yes, this information is important. Do write it. Okay. So this is how you can, like we in our lab, basically we used to differentiate the sepia and octopus, right? So you can even count what? You can even count the number of what? You can even count the number of what? Number of arms in that case. Done? So 8 arms they have. Done, beta? Okay, so here 10 arms are there. So you will see 8 arms plus 2 long tentacles they have, right? Plus 2 long tentacles they have. That's how you can very easily differentiate them. So loligo, which is actually also known as squid. So that is it. So let's go. Let's discuss the another part. Okay, so this is apple snail pila, right? So this is having the operculum. All the body organs are present there. This is your octopus. So this is pink tada is the pearl oyster, right? Pink tada is actually the pearl oyster, right? So this is again a PYQ beta. So how this pearl formation is there? There is a necker, which is mother of pearl, which this layer is also referred to as mother of pearl. Actually, this pearl formation is body's response against any impurity in the body. Just let's suppose what happens is कि if in their case, right, just necker is present, right, there's any impurity is there, so the body will get irritated due to that impurity. So what will happen? A layer will start forming around it. That is the necker. That is the mother of pearl. Okay, that is the mother of pearl. So basically, whenever you see the pearl forming, so we deliberately insert the impurities in these shells, and we keep on rotating them in such a way that whenever they release this particular layer, it should be oriented properly, round. So in the nature, if you will get the true pearl, that is not going to have that the proper round such shape. Okay, so that's how it works. Got that? That's how it works. So otherwise, overall in the snails also, you will see the statocyst. You will even see osphradium, right, which is basically for the osmoregulation. So now let's mark it here. So second largest phylum, terrestrial or aquatic. Terrestrial snails are also there. Organ level of organization, symmetry is bilateral, right? Triploblastic, and you know that few are asymmetrical. Already discussed that in the case of gastropoda, torsion is there. So do remember it. So gastropoda, the largest class, right? It will show what? It will show the twisting or the torsion, right, beta? So here they will be secondarily asymmetrical, means not from the starting itself, but later. So they are secondarily asymmetrical. You have to remember this. So calcareous shell is there having conchiolin protein as well. So unsegmented they are, but yes, I do remember neopilina, which is a connecting link, as well as a living fossil, as well as a segmented mollusk. Understood? Understood. So next here is a soft and the spongy layer of skin forms the mantle over the visceral hump. Space between that is the mantle cavity. Again a PYQ. Feather-like gills are present. So they didn't use the word here. So you should know that feather-like gills, what do we call them? We call it as ctenidia. So they are not just for the respiration, but along with that, so respiration and excretion. Respiration and excretion. That's how it works. Okay, so anterior head region has sensory tentacle. Mouth is having rasping organ, radula, right? Usually dioecious, oviparous. So pila is the apple snail, pink tada is pearl oyster, sepia is the cuttlefish, loligo is the squid, octopus is the devilfish. So aplysia, see here beta. Dentalium is the tusk shell here. Dentalium is what? Tusk shell. So this dentalium, right beta? Just like the elephant, array. Just a minute. Tusk shell. It respired through mantle. And then this chitin, Chaetopleura, having 8 plates on its shell. I'll show you. Right? So yes, this is it. So diagram is not there, but if you will see that chitin is having 8 plates on it. Diagram is not added. So next, Echinodermata. So this Echinodermata, you will see the water vascular system here, very important. What will you see? Water vascular system, also known as ambulacral system here, right? What else do we call it? We call it as ambulacral system. Here, you know that mesodermal endoskeleton is there. Matlab, these are the characters which resemble with the chordates as well. Mesodermal endoskeleton, we are going to see. We will see anterocoelom in their case, right? Even they are deuterostomas. So these are the advanced features in their case. Matlab, this is how they are resemble with the chordates. That's what you have to remember, okay? That's what you have to remember here. So these are the advanced features here. Am I clear? These are the advanced features here, okay? Okay, Baki, you know that this is the starfish. In the case of Asteriasis, in the case of your starfish, right? Male and female are in different body, beta. Indeed, but dimorphism is not there. Matlab, they are unisexual, but there is no dimorphism. When you look at them, you cannot say which one is male, which one is female. Moreover, starfish also show autotomy. Like, let's suppose if any predator captures its arm or something, it will break it. It will be like, you take it, I'll regrow. That's how it is, right? So they have endoskeleton of calcareous ossicles, right? So Echinodermata mane spiny bodied beta. Here you will talk about all are marine. So initially also I told you this phylum is of exclusively marine. So there are 3 phylums which are exclusively marine: your Ctenophora, Echinodermata, and Hemichordata. These are 3 which are exclusively marine, right beta? So adult are radially symmetrical, larvae bilateral, triploblastic, coelomate organism. And yes, in mollusca the coelom is quite reduced, right? So that's it. So we'll show you the water vascular system. So these are the different shapes: feather star, sea cucumber, starfish, sea urchin is there. So Asterias is the starfish, exam watch mane echinus, sea urchin, Augie is Ophura, brittle star, Antedon, sea lily, Cucumaria, sea cucumber. So examples and another names, both are important. So this is the starfish here. Look at this is the starfish here, how beautiful it is, right? 5 arms are there, you can see that. So now just see this part, right? Just have a look. So first of all, there is a, you know, a sieve like structure. There is a sieve like structure through which water will enter, right? Water will enter, we call it as madreporite. So water will enter here, then you will see this is the central canal, right? And then these are the radial arms. So in their surrounding, you will see the structure tube feet also will help in the locomotion as well, right? So in their case, you are not going to see, right beta, you are not going to see, no heart is there, no excretory organ is there, no head is there rather, right? No head, no excretory organ, nothing they have, even the heart is also not there, right? So look at this tube feet they have present. Look at the tube feet, right? Ampulla, podium, gonads are there. So through madreporite, right, a sieve-like structure through which the water will enter. Am I clear? So it is the most advanced feature here, beta, right? So that will help in locomotion, food capturing, even transport of food, even in excretion and respiration. Done? Okay. So overall, their body is having minute pincer-like structures which will not allow the debris to grow in it. We call it as pedicelaria. Pedicelaria. Understood? That is pedicelaria. Understood? Okay, so nervous system is also simple, right? That will develop a nerve ring, simple sense organ, no head, no brain. So remember them: no head, no brain, no excretory system, That's how it works, right? Anus is also on that dorsal side there. So let's mark this. So here you people can see animals have an endoskeleton of calcareous ossicles. The name Echinodermata man is spiny bodied. All are marine. So this is also exclusively marine. Organ system. Adults are radially symmetrical. Larvae are bilateral. So adults are showing radial symmetry. We put it in that category. So they are triploblastic, coelomates. Digestion is complete. Mouth on the lower side, yes, and anus on the upper. Anus on the upper side, mouth on the dorsal side. So here they have water vascular system. So this water vascular system is also referred to as ambulacral system. What do you call it? Ambulacral system, right, beta? So how will it help in the locomotion, food capturing, its transport, even in the respiration? Excretory system is absent, sexes are separate, reproduction is usually sexual but usually external, right? So larvae are present. These are the examples. Examples are important, okay? Examples are important. So extra features I have already explained you. So that is it. So they have madreporoid, they have the ring canal, they have the radial canals, right? So tube feet are also associated with it, okay? Okay. So common names and all, they are important. Now let's move to the next part, people. Let's move to the next part. But yeah, do make sure that you remember these examples also, okay? Okay, done? Yes, beta, pearl is formed like that, Christina. So next is hemichordata, right? Which are believed to be the connecting links in between the chordates and non-chordates. So balanoglosses, we call it as connecting link between chordates and non-chordates. Okay, so beta, they were considered to be, initially they were placed in the phylum chordata only. Why? Because they are having a structure that is stomochord. So this stomochord was confused with the notochord. So they were kept in the phylum chordata, but later on we got to know that it is just a kind of, you know, I would say that gut part, that stomochord. It is actually not the notochord. So yes, these hemichordates, they were initially, they were kept where? They were kept in the phylum chordata. Got it? They were kept in the phylum chordata. So they have worm-like appearance, right, beta? So you can see that body is having anterior proboscis, a collar, and a long trunk. They have the proboscis gland, which is meant for the excretion in their case. Are you getting my point? Which is meant for the excretion in their case. Got it? Yes. So overall, we know that they are also enterocele, closed circulatory system. So imagine we are moving towards advancement and we are having these worm-like structure. Oh, sorry, circulatory system is not closed. It is open here. Here also it is open. Right? Done? So they have rudimentary structure, which is the stomochord. So this is also exclusively marine phylum, okay? Okay? Overall, this phylum is also what? Phylum of exclusively marine organism, right? So they have gill slit. Yes, they do have the characters of both. They have pharyngeal gill slits for exchange of gases. Okay, for exchange of gases. Am I clear? Am I clear? Sure. Right? So hemi mane half beta. So gill slits is there. Their nerve cord. Their nerve cord is not well developed, but yeah, they do have dorsal nerve cord. Dorsal nerve cord, which is partly dorsal. So that's why you are considering them as half chordate. Okay. That's why we are considering them as half chordate. Okay. Okay. So balanoglosses, which is also known as tongue worm or acorn worm. Write it quick. Done. So they have stomochord. They have what? They have stomochord. Guys, done? So let's mark it. So you can see the diagram. That's how it is. It's a worm-like structure. It doesn't even appear like an advanced organism, right? So they were considered under the as a subphylum under Phylum Chordata, but now they are under a separate phylum under non-Chordata. Why? Because they have rudimentary. Rudimentary mane like it is not well developed. In Hindi, we call it as avshesh, like something which is not fully developed, but some part of it is there. So in the collar region, they have that is stomochord. So stomochord is a bit of hollow, right? Hollow tubular outgrowth from the buccal cavity, or I would say from the foregut region, right? So this is a kind of small hollow outgrowth. Is that clear? Small hollow outgrowth from the buccal cavity. Okay, okay. And where do it extend? From the roof of buccal cavity and extend into proboscis. And extend into what? Extend into proboscis. Got it? Right? So this phylum is having worm-like marine animals with organ system level of organization, lateral symmetry, triploblastic coelomate, cylindrical body, anterior proboscis is there, a collar and long trunk. So sometimes they can also frame the question at the place of anterior. If they will write posterior, then it will be incorrect. So circulatory system is of open type, respiration through gills. Basically, they have pharyngeal gill slits actually through which they respire. Excretion is through the proboscis gland. Sexes are separate. So this is your tongue worm or eccrine worm, or this is the secoglossus. Is that clear? Is that clear? So next is basically Phylum Chordata, in which chorda mane a thick string. So that's what you have to discuss. Okay. Okay. So Phylum Chordata. Chorda mane thick string. What's the meaning of chorda? Chorda mane thick string. Ata means to have. Right. So beta, I have already given you a break for 15 minutes initially. So let me complete this portion, then we can have a break. Okay. Right. Then we can have a break. Okay. So see, in marathons, usually some teachers, they do not complete the topic. They just give the overview. We are discussing everything in detail. So even if you are watching this lecture for the first time, it is helpful for you. Okay. So that's why I will take class 11 syllabus either in 3 parts or in 4 parts, right? But whatever we are going to complete, we will complete it, right, properly. So this is what we have to discuss in the chordates, right? So in the case of chordates, 4 basic characters. First of all, one second. I don't know why students make the mistake here. So it's not like whenever somebody will ask you the basic characters, you will put the hard part first. When it comes to 4 basic characters, there are other characters also. 4 basic characters will include presence of nerve cord. It will include presence of, not nerve cord, presence of notochord, right? Then there is the presence of dorsal hollow nerve cord. There is the presence of pharyngeal gill slits. Then there is the presence of post-anal tail. These are the 4 basic characters that we consider when we talk about the Phylum Chordata. Organisms should have this in their lifespan. Okay, and yes, in the chordates, the heart will be on the ventral side. We are the chordates, our heart. This is our ventral side. So our heart is on the ventral side. That's what you have to remember. Okay, so in the Phylum Chordata, because here we have to read it up to the class level. So Phylum Chordata, what do you see in Phylum Chordata? You see 3 subphylums, right? We are going to see 3 subphylums. Yes. So it's you can win. So you mane Urochordata, can Cephalochordata, win Vertebrata. Vertebrata, you can win. and out of that, uro mane tail. That's what you have to remember. So here, only in the larval stage, right? Only in tail region, even only in larval stage, only in tail region, you will see notochord. So it is not in adults, right? So even it is not even considered as a true chordate, because adults will not show any chordate like character. So here, from head to tail, you will see notochord is present, and that persists throughout the life. That persists throughout the life. So it is considered as a true chordate. Vertebrata, here we know that notochord in the lifespan will be replaced by vertebral column, right? Notochord will be replaced by vertebral column. So always remember, important it is, all vertebrates are chordates, but all chordates are not vertebrates. But all chordates are not vertebrates. That's an important part. Okay? That's an important part, okay? All vertebrates are chordates, but all chordates are not vertebrates, because these your urochordates and cephalochordates. So ultimately they are what? They are the primitive chordates. They are the protochordates, right? They are the protochordates. They are acraniates. They do not have cranium, that is the brain box. So that's what we have to understand here. Okay, so that is what we have to understand here. So this information is important. So lower chordates may, yes, so lower chordates may we have your protochordates, we have your protochordates, these are acraniates, that is they have no brain box, right? Vertebrates will come under the advanced category. So overall, right, in NCERT they didn't mention much regarding these urochordates and cephalochordates, but I'll share a few important points with all of you. So cephalochordates are considered as the true chordates. Why? Because this is the first chordate, rather first chordate. Why? Because it will show, the adult will show all the chordate like character. Is that clear? Is that clear? Done? So these protochordates, basically these are subphylums, right? So if in paper they will ask about the phylums, then you know which phylums do we have. In the name, when it is the phylum, phylum is your Cnidaria, Echinodermata, Hemichordata. They are exclusively marine. What are they? They are actually beta exclusively marine, right? And here, these are the subphylums, Urochordata and Cephalochordata. So Uro and Cephalo, these are the subphylums. And these subphylums, what are they? They are exclusively marine. Am I clear? They are exclusively marine. Is that clear? So here you will see, right, that adults are primitive and larvae are advanced. So adults are fixed, larvae are movable. So here urochordates may, in general, you see retrogressive metamorphosis. So retrogressive metamorphosis means your larval stages are more advanced than the adults. Understood? Understood? Yes. So here all the adults member, all adults have a covering, that is tunic or tunic is the word that we use for covering test. So that's why you also call them as tunicates. What do you call them? You call them as tunicates. Understood? Understood? So, very important point, notochord is found only in, that's why the word is Urochordata. So, notochord is found only in tail region of larva and lost during metamorphosis. Lost during metamorphosis. Am I clear? And it will be lost during when? During metamorphosis. Understood? Understood? Done? Done? So larva is having dorsal nerve cord, but adult will have but adult, in adult, it is replaced by nerve ganglion. So imagine, adult is having ganglions. Right? Adult is having ganglions. Larva is having this. Okay? Okay? So in all chordate characters are in larva. All chordate characters are in, where do you see this? All chordate characters are in larva and adult contain only one chordate character, that is pharyngeal gill slits. Got it? That is pharyngeal gill slits. Understood? That is pharyngeal gill slits. Understood? Sure? Sure? So open type blood vascular system, but heart is on ventral surface. So these are few points. So you can see retrogressive metamorphosis they have, right? So blood vascular system, BVS mane, here I'll write it. This is the short form that I will use further. So blood vascular system, blood vascular system. This is the meaning here. So it is open type, right? But heart is ventral. But heart is what? Heart is ventral. Am I clear? So the urochordates may you have the trick that is sad. They are sad. Sad is the trick here. So this is how you have to remember the example. So jaise we have salpa, we have acidia, we have the doliolum. Okay, we have the salpa, acidia, doliolum. These are the examples. So they are the tunicates because they have test around it. Retrogressive metamorphosis. Why? Because adults are the primitive in comparison to the larval stages. So next is what? Cephalochordata. Right? Next is what? Cephalochordates. So they will be in shallow water. Both are free swimming here. Right? Adult and larvae. Both free swimming. Both are free swimming. That's what you have to remember. Adult as well as larvae. Both are what? Both are free swimming. Right? So notochord and nerve cord. Right? It is basically remain extended from anterior to posterior region and notochord persists throughout the life. Throughout the life. Done, beta? Sure? Sure? Okay. Done, bache? So here, blood vascular system is closed. Respiratory pigment is absent there. Blood vascular system is closed for excretion, right? This is again in the excretory organs and elimination part also we discussed. For excretion, they have protonephridia. That's a very important point, right? Which you are also calling as selenocyte. So this is what they have for the excretion, okay? Flame cells, या selenocyte, या protonephridia, okay? So exceptions are there. So this is the, so all the coded characters will be there in the adult as well, okay? So they are the first complete, या फिर typical chordate. As adult will also have all the chordate-like characters. Got that? Right? So branchiostoma or amphioxus is the example. Branchiostoma or amphioxus is the example. Lanceolate is the word that you have to write here. Lanceolate. Got it? Done, beta? Sure. So now we have to move to the subphylum Vertebrata, but let's understand this portion from the NCERT, then let's go. So phylum Chordata, let's have a look, then we will just discuss that. So phylum Chordata is there, right? So they are fundamentally characterized by presence of notochord, dorsal hollow nerve cord, and pharyngeal gill slits. Bilaterally symmetrical triploblastic coelomates with organ system level, right? Possess a post-anal tail and a closed circulatory system. So here I have specified which one are the four basic chordate characters. This diagram needs to be very clear. So notochord is your mesodermally derived. Do not forget that. It is made up of what? It is mesodermally derived. Like it is made up of mesoderm. Above it you have dorsal hollow nerve cord. So if in any non-chordate the nerve cord is present, it will be on the ventral side. It is going to be solid. It is going to be double. Pharyngeal gill slits, they are paired. And this is the post-anal tail. Okay. So then comes this table, notochord present, absent. So all that points are there. So heart is ventral, heart is dorsal if present, right? If present, then it is on dorsal. Which tadpoles are you talking about, beta? Done, bache? So these differences are literally very important. So next is this part here. So in the NCERT, right, in the NCERT, so subphylum Urochordate and Sphelochordate referred to as protochordate. So yes, even in the NCERT, do mention it, do mark it that they are also the protocordate. We also say acraniates. Yeah, they are acraniata. They do not have, they are without cranium, without a brain box. Got that? Done, beta? So notochord is present only in the larval tail, while in cephalochord extend from head to tail. Examples are discussed here, right? So members of subphylum Vertebrata, you know that in their case, it is the notochord which will be replaced by, right? Which will be replaced by the vertebral column. That vertebral column can be cartilaginous, it can be bony, it depends. So that's why we say all vertebrates are chordates, but all chordates are not vertebrates. So basic chordate characters may, you know that other than, dekho, besides means in addition to the basic chordate character that I have discussed. So you will see ventral muscular heart, 2, 3 or 4 chambered heart, kidneys for excretion and osmoregulation, and fins or limbs are present. So when it comes to this part, basic chordate characters. So these are that 4 basic characters: notochord, dorsal hollow nerve cord, pharyngeal gill slits, for post-anal tail. Do not make the mistake there, okay? So this is a CDR. Diagram-based questions can also come in the paper. So next is this classification. So vertebrata, 2 divisions are there. Agnatha, it lacks jaw. So it is having class Ostracodermy. Actually, there is one more class which is, or this is Cyclostomata, Ostracodermy. There used to be one more class, Ostracodermy, but it is extinct now. All the members are extinct. So there is a class Cyclostomata. Here you will see circular mouth in the organisms, and here obviously they are ectoparasites as well. So they do not have the jaw. Okay. Now, when you talk about this, this is the super Glythostomata mane, they have jaw. So superclass Pisces, they will have fins, right? So study of Pisces, yes please, study of Pisces, what do you call it? You call it as ichthyology. You call it as ichthyology, right? So class is Chondrichthyes, Osteichthyes. So there is one more class here, that is Placodermy, which is again extinct. Which is again extinct. Got that? So tetra mane 4, poda mane limbs. They have these limbs, beta. So here you have the trick. It's ARAM, ARAM, amphibia, reptiles, apes, and mammals. Now you are going to find it very easy, but yeah, ultimately you have to remember it. Is that clear? Okay. Matlab bache, if ma'am please mention the chapter name. Beta, if you don't even know the chapter name, then what are you doing here in the session? Right? Please do not waste your time watching this lecture. Do something productive. Okay? Because if you don't know the chapter name, then what are you doing here? So here I'll strictly stick to the NCERT, because here the main thing is from the examples, and then obviously you'll get the break, and then we'll start the plant kingdom. So Cyclostomata, right? What is the word that we have here? Cyclostomata, right? Ectoparasites, they are on some fishes. Elongated body bearing 6 to 15 pairs of, it is not single, paired gill slits are there for respiration. Sucking and circular mouth, with the help of that, they can attach itself to a large fish. So they are without jaws. Body is devoid of scales and paired fins. Means this is the point that I have to remember. No scales on the body, just like fishes, and no paired fins. That's what you have to remember. That's what you have to remember. Then 16 to 15 pairs. So pair is the important thing here. Pairs of gill slits here. Done, beta. Cranium is there because obviously it comes under the vertebral column. So cranium and, it comes under the vertebrate. So cartilaginous. They are circulation closed type. They are marine but can go for spawning to the fresh water, right? So this is the migration that they show. Okay, so after spawning, within a few days they die. Larva will metamorphosize, and after that it will return to the. So this mixin hagfish, hag mane old lady, it appears like that. It doesn't show any migration. So no migration will be there. No migration will be there in the hagfish. Is that clear? You can write down, right? You can write down. Okay, please write down. So just focus on the main pointers here only. Done? Done? So just say you have types of fish migration. Anadromous. Anadromous migration may, what is happening? In this case, you will see that fish lives in seawater and migrates to fresh water for spawning, for gamete release and all. So this is anadromous, and catadromous may there will be vice versa. Okay, okay. So both the type of migrations can be there, right? So That's what you have to remember. There are other types also, but main is this. So marine to migrate fresh water. They are marine but migrate for spawning to fresh water. So now you can see what type of migration it is. Understood? Understood? Yes. So lamprey is there, which is again the ectoparasite, right? And here, just here they are saying that they usually they don't have larvae, right? But here they are mentioning why this petromyzin is having the larva. It is ammocoete larva. Ammocoete larva. Is that clear? It is having this ammocoete larva, which is considered as a connecting link between the cephalochordate and these cyclostomata. So this larva basically, beta, right? So anadromous migration is there, larva is ammocoete, and here in myxin, which is also the hagfish, right? It remain attached to the host gill, to the gills of host, yeah, host's gill. Done. So in this case, you won't see migration, okay? So they don't have long distance migratory behavior, only the local movements, right? So that's a PYQ regarding the myxin. Do check that out. Now, next is this is petromyzin lamprey. Chondrichthyes, cartilaginous fishes, we are also calling them as what? And now, even the cyclostoma is also the, cyclostoma is what? This is also the false fish, right? True fishes, they only belong to superclass Pisces, जैसे devil fish. Even, I forgot to tell you, Arthropoda में you have one example, Lepisma, that is the silver fish, right? That is the silver fish. In our very old books, right, in Almiras, you find this insect. So this is again not a true fish. Why? Because it doesn't belong to the Pisces. Right, so these are the false fishes. Just your devil fish, which is a mollusca, right? Your cuttlefish, which is again a mollusca. They all are the false fishes. Even your cyclostomata, right? They are also the false fishes. True fishes will belong to what? They will belong to superclass Pisces. Okay, okay. So Devonian period during evolution, right? It is basically the age of fishes, when fishes they were dominating. So you have study of fishes as theology. So we have chondrichthyes, that are the cartilaginous fishes, and we have the osteichthyes as well, right? Which are basically what? Osteichthyes as well, right? So that are basically the bony fishes. So placodermi was one more class there, which is extinct. Got it? Done. So here you are going to talk about the true fishes. Obviously, we know that they are cold-blooded. Cold-blooded, right? Warm-blooded are birds and mammals only. So that is poikilotherms. They are poikilotherms. They are cold-blooded. Is that clear? Sure. So their body, baby fishes are called fry or hatchlings. But okay, that's not our part. So it is class Chondrichthyes, which is also referred to as elasmobranchi, right? Elasmobranchi. Understood? So what are the main points here again? So they are marine with streamlined body, have cartilaginous endoskeleton. Here, mouth is located ventrally, notochord persists throughout the life. Gill slits are separate beta but without operculum. They don't have a covering. We can clearly see them. No gill cover is there. Skin is tough, containing minute plecoid scale. Teeth are also these plecoid scales beta, backwardly directed. Jaws are quite powerful. They are predaceous, predatory word. Like your shark belongs to this. They don't have air bladder. So no air bladder. No air bladder. Yeah, swim bladder. They have to move constantly, otherwise they will just sink. So their tail, mostly you will see the tail fin is not equal. So tail here in this case will be heterocircle tail. Heterocircle tail fin, because it is not same. Is that clear? Done beta? So male fishes may heart is two chambered, one auricle, one ventricle, but two accessory chambers are also present. That is sinus venosus, truncus arteriosus, right? They have sinus venosus, truncus arteriosus. So they have single circulation, right? Overall in fishes, you will see single circulation. They have venous heart. It means that heart pumps deoxygenated blood, right? That heart pumps deoxygenated blood. What will heart do? Heart will pump deoxygenated blood, beta. So gills they have for the oxygenation of blood. Understood? Done? Done. So some of them will have the electric organs, जैसे torpedo, pedo, E, electric, ray, it is poison sting, trigon, sting, G. So cold-blooded, no capacity to regulate their temperature. So males have claspers, beta. It is important. This one help in reproduction, help during reproduction. Okay. So internal fertilization, and majorly they are viviparous. So scalidon dogfish, Pristis sawfish, its rostrum is modified into a teeth saw-like structure. Carcharodon, great white shark, and the trigon is there. Okay, so this information is important here. If you will even stick to the NCERT, that will be sufficient. So they have, overall, they have 5 to 7 pair of gills are present. Do mention it. 5 to 7 pairs of gills are present in their case. Right, 5 to 7 pairs of gills are present in their case. Am I clear? 5 to 7 pairs of gills. Done? Sure? So one more is chimera, which is basically, right, this is actually the king of herrings, yeah, ratfish, right? King of herrings. या फिर ratfish. This one is the connecting link between cartilaginous and bony fishes. So when it comes to extra points, yes, we can do a lot of extra points here. But the point is, right, we are running out of time. So let's stick to the topic. Let's cover this NCERT. Done, students? So these are the examples. So yes, your Pristis, right, your Pristis, the sawfish, it is a true fish because ultimately it belongs to your sawfish. It belongs to your Chondrichthyes. Okay, matlab to superclass Pisces. Done? Done. So next is the Osteichthyes, also known as teleostomy. This is a class, not the superclass. Keep it in your mind. So they are found in both marine and freshwater. Their endoskeleton is not just cartilaginous, it is bony. They have streamlined body. Mouth is mostly terminal. Last time what you discussed, mouth is basically ventral. Okay, ventral. Ma'am, please help me giving all direct and indirect of phylums and fertilization. Beta, what sort of trouble that you have in this? Just open the NCERT. Make on a paper, just write down phylums which are showing direct development, phylums which are showing indirect development. It is that as simple as that. So 4 pair of gills they have. How much? 4. So they have operculum, Chondrichthyes, they were not having that. Skin is having cycloid, ctenoid, and just say in the case of fishes, you see the gills, right? So here cycloid is there, ctenoid, teeth-like structure. Even you also see ganoid scales, right? So scales are present in their case. Here we only talked about the placoid scales, and that placoid scales are dermal in origin. Matlab from which layer they are arising. So they are dermal in origin. They are dermal in origin. Am I clear? So ear bladder here is present. Done, beta, which regulate buoyancy. So they'll keep on swimming, right? Right? They have no need to just keep on swimming because they have air bladder that will maintain the buoyancy. But Chondrichthyes, they keep on swimming. Again, heart is 2-chambered here, right? Same condition. Here also 2 accessory chambers are present, right? Here also 2 accessory chambers, sinus venosus, truncus arteriosus, they are present, right? So sexes are separate, fertilization usually external. There it was usually internal. Mostly oviparous, development is direct. So exocetus is the flying fish. Hippocampus is the seahorse. So hippocampus is the pregnant male. The only male in the animal kingdom which is pregnant in its brood pouch, right? The female is also going to lay the gamete. So male is going to show the parental care. Male is going to show everything there. Is that clear? Beta heterocircle hetero mane different. The tail fin is not equal heterocircle tail, right? In the Osteichthyes, you will see both the tail fins are equal mostly. Homocircle tail is there. Homocircle tail or heterocircle tail. That's it. That's it. Please pay attention here. Okay. Done. So you have Catla, Labio, Clarius, which is even the Magur. So you should know the common name. So beta fighting fish, pterophyllum is the angelfish. So there is one more fish, tetradon, puffer fish. Tetradon. Right. So it is also, Clarius is also referred to as the catfish. Okay. Done? So male, pregnant male is hippocampus, seahorse bitches. Understood? Any doubts? Done? So hippocampus, pregnant male having brood pouch, this is the catla. So these are actually the scales on it. So placoid scales are in your chondrichthyes, cycloid, ctenoid, ganoid scales are in osteichthyes. Next is the class amphibia. So amphi means dual, right? So they started taking a step towards the land habitat, but they are not true land animals. They don't have that modification. True land animals are the reptiles. So frog is the example, which is again the part of your syllabus. So here you know it very well that as the name indicates, amphi mane dual, bios mane life. So amphibians can live in aquatic as well as terrestrial habitat. They have two pair of limbs. So this is important. No neck here. No neck here, only head and trunk. This is important, right? Tail may be present in some. I'll come to this also. Skin is moist. So when skin is used for the respiration, we know that it is cutaneous respiration, right? So it is without scales. Eyes have eyelid. Tympanum, which is the middle ear. It is the tympanum which is representing the ear. It is the middle ear. Alimentary canal. This is important. Even the urinary and reproductive tract, they all open into a common chamber, that is cloaca. So in the cloaca, you will see alimentary canal, urinary canal, या फिर excretory system, reproductive system, they will all open here, and this is the cloacal opening, the cloacal aperture. Got that? Right? So respiration is by gills, then cutaneous, lungs, pulmonary, skin. Sorry, gills means branchial, pulmonary, cutaneous. Heart is three-chambered here, right? Cold-blooded, sexes are separate, fertilization external, oviparous and development is indirect. Got that? So, heart is three-chambered. So here you will see incomplete double circulation. Incomplete double circulation. Jैसे you have auricle, ventricle. This is, two auricles, one ventricle. So blood will get mixed here, right? Blood will get mixed here. So here you will see incomplete double circulation. Am I clear? Incomplete double circulation. And yes, here also that two accessory chambers, sinus venosus and truncus arteriosus, are well developed. Are well developed. They are well developed in their case. Am I clear? They are well developed in their case. Okay. So you have the example. So overall, here you will see they have the cranial nerves. So cranial nerves are again here also 10 pairs. Just we have 12 pairs. Here you will see there are 10 pairs. So Bufo, Rana, Hyla, Salamander, and Ichthyophis, limbless amphibia. Very important MCQ, right? Very important MCQ it is. Okay. Okay. So Salamandra is having axolotl larva. So this larva, beta, it shows a property of neoteny, that is reproductive organs develop in large world stages. That's what it is. Okay. Okay. Done. So in the class Amphibia, you see there are 3 orders that we see basically, right? That is, one is Anura. So Anura mane your without what? Without tail. Just say you have the example of frog. Are you getting my point? Frog, bufo, they are the example. Then comes the Urodela, which is also chordata. Urodela, which is also chordata. Chorda mane tail, have tail, have tail. Example is of Salamandra having axolotl larva. Then you have Apoda. Apoda mane no poda mane limbless. Here you have the example of Ichthyophis, which even looks like what? It looks like the Annelida. Okay, so you have the example of Ichthyophis. Is that clear? Ichthyophis. Okay. Then Phys. Ichthyophis. Next is reptiles, right? Next is reptiles. So successful land animals, lungs are well developed, their eggs, they can also survive that conditions. So that's what we have to discuss. So reptilia it is. So basically it means to creep. So creeping or crawling mode of locomotion, they have the so Latin may repair or reptum mane to creep or crawl. So their terrestrial body is covered by dry and cornified skin, because now they are showing the land adaptations. Right now they are showing what? They are showing the land adaptation, students. So dry and cornified skin is there that can conserve the water in their body. So epidermal scales or scutes they have. No external ear opening. Tympanum will represent the ear. So this external ear you will see in the mammals only. So limbs when present are 2 pairs. Heart is usually 2-chambered. Sorry, usually 3-chambered. Just say here heart is actually incompletely. Matlab ventricle is having a septum which is not completely developed. But here also you see 4-chambered heart. Just say in the case of crocodile you will see that, right? Right. So snake and they are poikilotherms. Snake, they and lizards, they shed their scales as skin cast. So you know that shedding of skin is known as ecdysis ya fir molting. So they will, the snake, in the case of snake, you will see the complete skin it is going to shed. Is that clear? Right? Is that clear? Yes or no? Done? So in their case, sinus venosus is ill developed, like not completely developed, and truncus arteriosus is absent. Absent. Got that? So sexes separate, internal fertilization. So chelon is turtle, testudo is tortoise. Turtle and testudo, how can you differentiate them? So in the case of chelon, right, turtle, you can see that digits are not there. It is just like a fin, right? In the case of tortoise, you will see that they will have digits. Done. And you have one homework, you will check the difference in alligator, crocodile, right, and all, etc., etc. Then beta, only poisonous lizard, if you will ask me, is your Heloderma. Only poisonous lizard, Heloderma, right? Only poisonous lizard it is. Got that? And here you will see, just like us, 12 pairs of cranial nerves. Understood? 12 pairs of cranial nerves. So poisonous snakes may, and do check the difference in poisonous and non-poisonous snakes. Naja, that is cobra. Bungarus, crate or vipera is viper. Okay. Done? Done? So it's Bungarus. Okay? So vipera is viper. Right? So you talk about the python and Hemodactylus is wall lizard. Right? So Calatus is garden lizard, and the python is not a poisonous snake. It's a non-poisonous snake. Non-poisonous snake. Even better, we have the sea snakes also. So there are different examples. Okay, so that's what you have to remember. That's it. So any doubts? Any doubts? So do check the difference in poisonous and non-poisonous snakes. Done? So in the poisonous snake, poisonous glands are modified labile glands actually. What are they, beta? They are modified labile glands. Are you getting my point? Modified labile glands, and they are actually homologous to parotid gland. That's it. Chaliye, next part. Class aves, birds. You know that here you talk about the feathers. You will talk about the air sacs, right? Though body is not having any gland, but you will talk about a preen gland, which is also the oil gland, which is also the uropygeal gland, right? Uropygeal gland. Done? So that's what you have to remember here. Beta, we are about to finish, then you will get a long break, okay? You will get a long break then, okay? Done? Sure? Any doubts? So there are other points also, but firstly tell me this part is clear. Done, beta? So in their case, only left ovary is functional because they have right aortic arch na, so to balance the body weight. They have pneumatic bones. Pneumatic mane hollow bones, right? They will have keel sternum, right, to which flight muscles are attached. Plus, here we also talk about one wishbone, that is furcula. Furcula, it is the wishbone, right? So basically it is actually acting like a spring in between two pectoral girdles, right? Two pectoral girdles. It is acting like a spring. It's a kind of V-shaped bone, right? It's a kind of V-shaped bone. Just a clavicle or interclavicle actually, they join and they form it. Is that clear? Sure. Is that clear? Okay. So overall, beta, Huxley has said, right? Huxley has said that birds are glorified reptiles, right? Birds are what? They are glorified reptiles. So we discuss about one of the missing link as well, Archaeopteryx, which shows the Archaeopteryx lithographica, which shows the character of both birds as well as the reptile. They have the beaks. We know that they don't have the teeth. The jaws are basically forming the beak there. So we know that the largest egg is also the ostrich egg, which is even a flightless bird. Kiwi is the flightless bird. So here in this case, the sound production is by syrinx. Larynx is also present, but sound producing organ, right? Sound producing organ is what? Your sound producing organ is syrinx. Larynx is also present. I'm not saying that it is not, but sound production is done by syrinx. So apes mein feathers are there, beta. That is a very important thing, right? So most of them can fly except flightless bird like the ostrich. So ostrich egg is the largest egg. So they have beak. Forelimbs are modified into wings. So hind limbs have scales, beta. Hind limbs, but they are modified for walking, swimming, and clasping the tree branches. They are dry without glands. So they have oil gland, which is also the preen gland, and uropygeal gland at the base of the tail. So endoskeleton is fully bony. They have hollow bones with air spaces, air cavity. That's why pneumatic they are. This is also very important. I'll underline this part, that the digestive system is having additional chamber, crop and gizzard. Gizzard is also known as proventriculus. In cockroach part also I told you this. In cockroach part also I told you this, that in the cockroach also you have crop and gizzard. So in the insects basically. So birds are also having that. They are warm-blooded, right? That is homeotherms or endotherms, right? So air sacs are attached to the lungs. Air sacs are not playing role in not exchanging gases. Matlab air sacs are not to exchange gases, but they are supplementing the respiration because they have to fly. When they are flying, these air sacs are also having the lungs. They'll provide it. They are also having the air. They'll provide it to the lungs, right? So they themselves are not helping in the gaseous exchange, but they are supplementing the respiration, students, right? So sexes are separate. Fertilization is internal. Oviparous they are. So corvus, crow, columba, pigeon. This stecula is their parrot. Struthio is ostrich. So Echinodermata, penguin. Neophron is there, which is vulture. Okay, right? So penguins, seabirds of Antarctica. So in the case of penguins, forelimbs are, right? Forelimbs are modified into fins. That's what you have to remember. So penguins may be, beta, it is the cooperation of the male and the female, right, for raising their baby. Just a female will lay the egg, then female will provide that egg to that male. So many male partners, they together, they form a kind of arrangement. They hold that egg. They don't let it to touch the ground, because if they will allow it to touch the ground, then that egg will not develop. Female in meanwhile will search the food, and to keep that baby safe, even a lot of fat from that male body will shed off. So that's how they protect. And then who will come back? Then your female will come back. So collectively they raise their child, right? Do read about that. It's very interesting. So here, this in the case of penguin, forelimbs are modified into fins. Sorry, not fins, flippers. Sorry, flippers for swimming. Into flippers for swimming. Got that? Into flippers for swimming. Am I clear? Done? So ostrich, you know that Struthio is the largest living bird. So that's it. So kiwi is also there, having hair-like feathers. Smallest flightless bird is your kiwi. So that's it. That's what you have to remember. Or there are many points which we can also discuss, but chalo. Okay, let's stick to the NCERT part here. So let me show you the diagrams. Do remember them, beta. Do remember them. And here you can see the air sacs which are attached to the lungs. So they they supplement the respiration. They are themselves not playing any role in the gaseous exchange. Okay, so air. So syrinx is there. So in birds, that sound is produced by syrinx. So syrinx is the one which will produce voice. Produce sound producing organ. Then comes the mammals. You know that we have here. So basically here the specialities, we have the mammary gland, right? Females they are functional. They are used to nourish the baby. Is that clear? Is that clear? So here hoofs, nails, all that things are also present. So we can live in the variety of habitats, even in the polar ice caps, deserts, mountains, forest, grassland, and dark caves. So adapted to fly, even to live in the water, right? So we have milk producing gland, very important feature. 2 pair of limbs which can be adapted for different things, right? We have unique in possessing here. We have pinning, right? So far we didn't discuss external ear pinnae anywhere, right? So teeth, different types of teeth are present in the jaw, heterodont dentition, heart is four-chambered, respiration by lungs, sexes are separate. So examples are given here. So you should know the examples, right? I'll tell you, just say ornithorhynchus, right? Ornithorhynchus. That is basically your duck-billed platypus, right? That is again this duck-billed platypus. What's that, students? This duck-billed platypus is a connecting link between reptiles and birds, right? Reptiles and birds. So it is again an oviparous egg-laying mammal. Are you getting my point? Are you getting my point? Yes, please. So ornithorhynchus is there. That is oviparous. You should know about it. Okay, you should know about it. Okay. Okay. And yes, how can I forget to mention it? Urinary bladder and, right, one is urinary bladder and copulatory organs are absent in birds. This part. So here I'll stick to the topic itself. So you have the ornithorhynchus, duck-billed platypus it is, which is again, as I said, the connecting link. You should know that, beta. Okay, you should know that. So these are basically the monotremes or prototherians, right? They are the prototherians. You have one more example here. And viviparous mein, right? Macropus is their kangaroo, but in the case of kangaroo, I hope you know that they are metatherians, okay? Metatherians. So, baki all the examples are written here. You should even know their scientific names also. So, whale is the aquatic mammal. Your flying, so whale is the aquatic mammal, right? The bat is actually what? Bat is the, yes, please, flying mammal. So, we have different adaptations there, okay? Done, bache? So this is all about this chapter. Okay? So overall in class Mammalia, you have, right? Overall in class Mammalia, yes, we do have the subclasses, right? We do have subclasses, and here you will see there are 3 subclasses. So these placental mammals, eutherians they are, right? What are they? They are these placental mammals. They come under the eutherians category. So prototherians mein, monotremes mein, prototherians are also there, right? Which are monotremes, where you will talk about the egg-laying mammals. One example I have already given you. This is Ornithorhynchus. The another is Tachyglossus. Okay, right? Another is what? Another is your Tachyglossus. Okay, Tachyglossus. Done. So Tachyglossus may you have, sorry, this is your spiny ant eater. You also call it as echidna. And here monotremes done. Prototherians also known as monotremes. Is that clear? Then another one is beta, we have the metatherians, where you talk about the marsupials. Metatherians, where you talk about what? You talk about the marsupials. You have the example of kangaroo here. Done. You have the example of kangaroo here, not just kangaroo, Didelphis is also there. Right? Didelphis is also there, which are opossums. Okay? So Didelphis, this is a scientific name. So that's it. This is all about this kingdom. So do check this table, do read it, right? Do follow it for quick revision, right? So next is the plant kingdom. So let's start it after a break. So let's take a break. Done. Let's take a break till 40 minutes ka break. Okay, happy? So after that, there will be no break. After 40 minutes, there will be no break. We will complete the plant kingdom, then you will have the break. So today we have Now, guys, now, am I? Now, am I? Now, am I? I think now I'm still. Now, okay. Okay, so guys, sorry for this. Even I didn't realize. So basically, we are talking about the plant kingdom from where 3 to 4 MCQs can come. So you have algae, you have bryophytes, you have pteridophytes. Then there will be the gymnosperms. Then there will be the angiosperms. Is that clear? So algae, bryo, pterido, gymno, and angio. Then after that, here, right, I made a flow chart. You can see this, but all the points are concluded here. So embryo is absent in algae. Yes, brown algae will be considered as an exception also. Right here, you will see some members will show different types of life cycle. We'll discuss about that too. Without vascular bundle, like the plants in which xylem and phloem is absent, algae and bryophyte. With the vascular bundle, the one which are having the xylem and phloem, they are the tracheophytes, so you have pteridophytes, gymnosperms and angiosperms. Seeds are absent, cryptogams, algae, bryo, pterido. Seeds present, phanerogams, spermatophyta, gymno and angio. So that's what you have to remember. Is that clear? Is that clear? So this is about the plants. This is what we have to discuss here. So in the case of this chapter, we will start with the classification part. Then I'll teach you about the life cycles beta, right? And then we will start the algae part, okay? So it is one of the most important chapters. So please pay attention here. Is that clear? Is that clear? So let's talk about the plants. We'll start with the handwritten notes here. So I hope it's clearly visible to everybody. So in the case of plants, you know that all eukaryotic chlorophyll containing organisms are present here, right? They are eukaryotic, they have the chlorophyll, right? No doubt we know that plants are producers, they are the autotrophs, but students, there are some plants which are insectivores, right? Which are parasitic as well. Just a bladderwort you have, right? Utricularia, Venus flytrap is there. Okay, Cuscuta is a complete heterotroph. Cuscuta is a total stem parasite. So it's a plant and it's a total stem parasite, Cuscuta, which is also known as dodder plant and which is also known as amar bale. Am I clear? Which is also known as dodder plant and amar bale. Then life cycle will have two phases in the plant. We'll talk about the haploid phase and the diploid phase. So yes, in the case of plants, I can say that there is alternation of generation. What will be there in the case of plants, people? Alternation of generation will be there. You will talk about the haploid phase and even the diploid phase, right beta? So length of these phases will vary, that will make the different plants. So let's start the classification part. So in the classification, the very first thing that we need to discuss is the artificial system. What is the very first thing that we need to discuss here? Yes, everybody. What is the very first thing that we need to discuss here? We will talk about the classification and we will start with the artificial system. So who were the contributors? Aristotle. Aristotle, who's also referred to as father of biology and father of zoology as well, right? Theophrastus is the father of botany, right? Carolus Linnaeus, father of taxonomy, right? John Ray, I don't know whether you remember it or not. John Ray was the one who gave term species. He was the one who gave term species. Is that clear? Is that clear? So the earliest system of classification, they have used only gross means superficial morphological characters, right? Only the morphological characters, that how that a plant is looking, what are the number of leaves, the habit, the color, the number and the shape of leaves. This was the thing that they have considered beta. So in this particular type of classification, yes, please. In this particular type of classification, they prioritize, they give the equal priority to the vegetative as well as the reproductive characters. So in this system, equal weightage was given to the vegetative as well as the sexual characters, and it is not acceptable. Why? Because vegetative characters are more easily affected by the environment, right? Vegetative characters, let's suppose, see, in the case of a plant, plant has to flourish first, like the stem, the root, the leaf should properly form. Then basically after that the flower came. Why? Because this vegetative phase will make sure that the plant is totally attached to the soil. It is having all the nutrition. It is capable of, further providing the nutrition to the developing seed as well. So vegetative part is very important, and it is it gets affected by the environment, right? Like, let's suppose if I have to give you a very simple example, in case of some plants, if you keep them in the sunlight for a longer period, their leaf size might get short. But instead of that, if you will keep them in shades, it grows. So you can see how the environment is affecting it, okay, right? So it's not advisable to give equal weightage to both plants, to sexual as well as to the vegetative character, right? So this system was made mainly on the vegetative characters or on the endometrium structure. Which one? The artificial system. So that's why it was having the drawback, because it was not talking about any of the evolutionary relationship. It doesn't consider the evolutionary relationship, right, beta? So characters, they were changing with the environment, right? So how can we take such characters which are so easily changing with the environment? That is why. And here, this system, this artificial system has separated closely related species because it was based on few characters. So that's why this classification was not reliable. Now, after this, what else do we have? Natural system, which is also known as two-dimensional system, which is also known as horizontal system. So here, the contributors, they were Bentham and Hooker. This is very important. The scientist name here is very, very, very important. So George Bentham and Joseph Dalton Hooker. So they were the one who gave the natural system. So in the natural system, students, many taxonomic characters, they were considered. It was on the basis of natural affinities, mane similarities among organisms, and considered not only the external feature, but also the internal characters. So it was not the one who considered just the external feature, but along with that, internal characters were also considered, like the ultrastructure, the anatomy, the cell structure, right? The phytochemistry, embryology, everything was considered. So Bentham and Hooker, those who were the English botanists, they wrote that thing in their book, Genera Plantarum. Genera Plantarum is different, species Plantarum is different. So if you will further study the botany, you definitely need to study the Bentham and Hooker. So here they divided the plant into these two parts, cryptogams and phanerogams, but they kept gymno, dicots, monocots, right, as inner classes and in different classes. Got that? So natural system was still better than the artificial system. Why? Because they were considering the internal structure. So if they were considering the internal structure, then indeed, related organisms will be kept together. Natural relationship amongst organisms will be considered. So, right, when you study the internal structure, obviously, you'll get to know how relatable two things are. Is that clear? So then comes the phylogenetic system, which is also known as vertical system, which is also known as three-dimensional system. So what else do we have? Yes, phylogenetic system, vertical system, and 3D system. So it is just the rearrangement of the natural system. What's that? It is a rearrangement of the natural system, right, beta? So here the evolutionary relationship between organisms, it was considered. So it believed, this is again a state PYQ beta, that same taxa will have the common ancestor. Right, the same taxa will have the common ancestor. So who supported that? Yeah, who were the main advocates? Euler and the Engler and Prandtl, they were the main supporter. And overall, the phylogenetic system is preferred. Why? Because evolutionary relationship, when you study that how organisms are related, connected to each other, right? How are they related to each other? So that is something which is very important. Am I clear? Am I clear? Am I clear? Sure? Done, bache? So next is the branches of taxonomy, where you have numerical taxonomy, also known as phonetics, also known as taximetrics. So the word is numerical taxonomy. What is the word that you have here? Numerical taxonomy. So it is carried out using computers and based upon all the characters that we can observe. Is that clear? right? So carried out using computers and based upon all the observable characters. So all selected characters, whatsoever characters were selected, equal importance and equal weightage was given to that. So in the numerical taxonomy, again, you'll solve the PYQ, you'll get the question on the basis of that. Number and codes, they were assigned, beta. Number and codes, they were assigned to the characters. Is that clear? Is that clear? So numbers and codes, they are assigned to all the characters and then their data is processed by using computers. Am I clear? So next is the cytotaxonomy, right? Next is what? Next is the cytotaxonomy, students. So when you are talking about the cytotaxonomy, then what do you see? Cyto means cell. So it depends on the cytological information, like what is the chromosome number, structure, and the behavior. Then comes the chemotaxonomy. So when you talk about the chemotaxonomy, you are talking about the chemicals. That's a very good way to classify the organisms, to classify these plants. So based on the study of chemical constituents of the plant, which will include the secondary metabolites, the chemical nature of proteins, to resolve the confusion. Right? So this is used to resolve confusion. This is the one that tells us about the chemical constituents, right? All the secondary metabolites that we are getting from the plant, just say your alkaloids, right? Any oil or something. So they are the one that help us to resolve the confusion, okay? Okay. Right, just say beta-cyanin pigment in the beetroot, presence of raphids and cystolith. So they tell us that how, you know, two plants, they are also connected because they will have somewhere nearby the chemical composition. So that is what we have to discuss here, right, students? So the next part that we have to discuss is algae, right? What are we going to talk about? Now we will talk about the algae. So let's discuss that. You know that in the case of algae, what do we need to study? So we'll start with this part first. You have chlorophyce first. What do you have? You have chlorophyce. Then we have pheophyce. Then we have rhodophyce. This is what we have. Yes, please. This is what we have. But guys, before going forward, before discussing this algae, let's talk about the life cycles. Okay, let's talk about the life cycles. So, green algae, chlorophyce, right? Pheophyce is your brown algae. And when you talk about the rhodophyce, that is the red algae. That is the red algae, right? This is what we have in the case of algae. Here, you have a trick that is self-DP. Why do I call DP instead of discipline? Because you people are literally not disciplined. So, sorry, not the self-DP here, my bad. The green algae, under this you have the trick. You have the trick that US have very clean college. That's the trick here. US has very clean college. US very clean college. You have to remember triple C here. Why? Because it will be Eulothrix, right? Spirogyra. Some people also call it as Spirogyra. Volvox, Chlamydomonas, Chlorella, Cara. These are the examples that you have to study here. Here you have self-DP. I told you na, you people are not disciplined. That's why I'm not calling it as discipline, self-discipline, self-DP. So it's Sargassum, it's Actocarpus, it's Laminaria, then it is Fucus, then it is Dictyota, then it is Pheophyce. Done. Then here you have the trick P2G2C. So P2G2C means P for Porphyra, Polysiphonium, Gracilaria, Gelidium, and Chondrus. Is that clear? These are the examples which you have to remember. So green algae, chlorophyce, US as very clean college, U for Ulothrix, S for Spirogyra, right? So Volvox, Chlamydomonas, Chlorella or Cara. Self-DP, Sargassum, Actocarpus, Laminaria, Fucus, then Dictyota, Phaeophyce, then P2G2C, Porphyra, Polysiphonium, Gelidium, Gracilaria and Chondrus. These are the examples which you have to remember. Got it? Right? These are the examples that you have to remember. Now, before going forward, right, we have to understand the life cycles here. Are you getting my point? What you need to understand here, you need to understand the life cycles here. Though I have, okay, now in this PDF, the diagrams are not there for the life cycle, though I have added it. Just let me check once. So they are not there. So, but don't worry, I'll discuss it. You'll get to know about it, okay? You'll get to know about it. Just allow me 1 minute. So let's discuss that. So we will talk about the life cycles in the case of plants. So here it is alternation of generation. First thing, right? Why? Because during the lifespan of plant, what will you see? During the lifespan of plant, you'll see the haploid and the diploid phases there. So 3 types of life cycles are there, right? We will talk about the haplontic life cycle. Please pay attention here. We will talk about the diplontic life cycle here. We will talk about the haplodiplontic life cycle. Is that clear? Yes. Is that clear? So 3 types of life cycles are there: haplontic, diplontic, haplodiplontic. Now, before going forward, what you have to understand, there are 2 words. Firstly, gametophyte. Then comes the sporophyte. Done? Gametophyte, that forms gamete. What's the meaning of gametophyte? Gametophyte is the one that forms gamete. It itself is haploid. Sporophyte, that forms spore. And spore is also haploid. This is what you have to remember. Right? I'll write few lines which you have to remember. Wherever you have any doubt, you can ask me. Number 1, gametophyte, which itself is haploid. So gametophyte is the one that forms gamete. Sporophyte 2n, that forms spore. These are the 2 lines which you have to understand here. So haploid phase, if somebody will say haploid phase in a plant, you are talking about the gametophytic phase. If somebody is asking you about the diploid phase in a plant, you will talk about the sporophyte phase. What will you discuss? You will discuss the sporophyte phase. So ultimately what is happening? Your spore, which is what? Haploid. It germinates, माने it grows. जैसे seed grows ना, वैसे spore, they germinate, they grow. They develop into gametophyte. This is the process in general. Because spore itself is haploid. If it will grow, if it will become multicellular, right? If it will grow, it will form the gametophyte. What that gametophyte will form? Gametophyte forms the gamete, right? You know that when there is the male gamete and the female gamete, it will fuse. It will result in zygote. Zygote itself is what? 2n, right? So it will grow into sporophyte. And sporophyte ultimately will do what? Sporophyte ultimately, you know that, will form the spore. So this is the complete thing which is going to happen. जैसे sporophyte forms the spore, they germinate, they form gametophyte, they form the gametes, they fuse, fertilization occurs, zygote form, zygote itself is diploid, it forms the sporophyte. Firstly, this is the story. Now, if you will think that this is from the deleted portion, actually it is not true. If you don't understand the life cycle, you won't be able to understand the topic. Is that clear? Is that clear? So what exactly is happening? We are talking about the life cycle. We are saying here we have alternation of generation. We are saying we have the haploid phase here, which is the gametophytic phase here, and we have the diploid phase here. So the duration, time span of these phases in the life cycle of a plant will tell that which life cycle is there. So jaise haplontic life cycle, diplontic, haplodiplontic, these are the three types of life cycle. Now before understanding these life cycles, there are few things which we need to keep in our mind. See, if you are talking about the haploid phase, you are talking about the gametophyte. If you are talking about the diploid phase, you are talking about the sporophyte. So gametophyte will form what? Will form gamete. Sporophyte will form what? Will form spore. And what's the story ultimately? Your spore will grow, will germinate, will form gametophyte. Gametophyte will form gamete, and gametes fuse, they will form zygote. Because zygote is diploid, obviously it will give rise to the diploid generation, that is your sporophyte, and sporophyte ultimately will form spore. So that's how everything is related. Got that? Now, next point. We will discuss about two divisions. Mitosis. You all know that mitosis is an equational division. I believe you all know this. Mitosis is an equational division. So if mitosis is an equational division, it means that the daughter cells will have same number of chromosomes just like parent cell. That's the meaning of a gametophyte. That's the meaning of the equational division. That's the meaning of the mitosis. Is that clear? Is that clear? That's the meaning there, right? What's the meaning here? That the daughter cells will have same number of chromosomes just like parent cell. Then comes the meiosis. Meiosis is the reductional division, right? It reduces the chromosome number to half. Here, if the parent cell is diploid, because meiosis occurs in two rounds. Firstly, meiosis 1, that will give you 2 haploid daughter cells, and then you have meiosis 2 in these cells, which will result in the formation of 4 haploid daughter cells. So these are the basics. Okay, these are the basics. So now understand one thing, students, that if you have gametophyte, which is N, and if a N structure need to give rise to N only, so mitosis will occur. So basically, I'm trying to tell you that if there is a haploid structure, if you have to make the haploid structure from it, you have no need to go for the meiosis, you will go for the mitosis. Now, sporophyte, which is 2N, spore, which is N. So if sporophyte is forming the spore, it will form it by meiosis. So in fungi, in plants, you won't see that it is the meiosis that is forming the gamete. No. In these cases, you will see that it is not the meiosis that forms the gamete, rather it will form the spore. That's what you have to remember. Got it? That's what you have to remember. So this is the basic actually. Now, the next part is the type of life cycles that we have to study. So haplontic life cycle, that's the first part. Haplontic life cycle, that's the first part. Haplontic means haploid, means haploid phase is dominant. What is the meaning of this particular line? Right? Haploid phase is dominant. It means gametophyte. It means it is the gametophyte which is the dominant phase. Are you getting my point? Are you getting my point? So you see this phase mainly in algae. Just say, let's suppose this is an algae. Okay. You just say in the case of humans. In the case of humans, we are having all our cells are diploid. है ना? In our case, all our cells are diploid. Our gametes are not. Our gametes are haploid. So you know that in our case, we have germ cells, that is gamete forming cells. They will undergo meiosis, that will form gametes. Gametes, male and the female gametes fuse. They form zygote, and again a baby chintu will grow, and that chintu is also diploid. That's what happening in our case. But this is not true in the case of plants. You cannot say that in the plants also the same thing is there, that all the plants are diploid. If you will look at a mango tree, in the case of a mango tree, you will see all its cell. You'll see they are diploid. You'll see they are diploid. The main plant body is diploid. Means it is the sporophyte plant. Why? Because it is diploid. Now, on the other hand, if you people will see the algae, We cannot deny this fact now that algae is also a plant. But when you will see the algae, exceptions are there. So here you will see that it is, all the cells are haploid here. So can I not call it as a gametophyte because it is haploid? Can I not call it as the gametophyte because it is haploid? Of course I can. So in our case, this is the story, but in the case of plants, anything can happen. Now, because we are talking about the haplontic life cycle, right? Because we are talking about the haplontic life cycle in which haploid phase is dominant, and gametophyte, so it means gametophyte is dominant, means when I see the algae, so in the case of algae, students, its structure is a thalloid, it's a thallus. What's the meaning of thallus? Thallus means it is undifferentiated. It is undifferentiated into true root, stem and leaves. Undifferentiated into true root, true stem and true leaves. It doesn't have true root, true stem or true leaves. It is undifferentiated into that. So this is the thallus. And here you will see the cells. What are they? They are haploid. So this is the haploid phase which is dominating. It is the gametophytic phase. Gametophytic phase. So this haploid phase is prolonged in the lifespan here. Now what will happen? If it is the haploid phase, mane it is the gametophyte, it will form the gamete. How will it form the gamete? Mitosis only. Mitosis will occur here. So because of the mitosis, why? Because already it is haploid and I need the haploid cells only. So mitosis will occur. So gametes will form. So you know that male and female gamete, they will form the zygote. So zygote, we know it's diploid. Zygote, we know it is diploid. So zygote, don't you think it is diploid? Diploid mane sporophytic generation. So zygote is diploid. So we will say here that it is first and the only cell of sporophytic generation. Why only cell of sporophytic generation? First cell means it's the first diploid cell. Why only cell? Why only cell in haplontic life cycle? Because this zygote, it undergoes meiosis. It undergoes meiosis. This zygote, it undergoes meiosis, right? So if it will undergo meiosis, definitely it is going to form the haploid spores because it is the first and the only cell of sporophytic generation. So definitely it will form the spores, right students, because it is undergoing meiosis, right? So we also call them as meiospores. We also call them as meiospores. As why? As spores develop due to meiosis. As spores develop due to meiosis. Now what will happen? This spore again germinate, means the spore is what? Single celled. It germinate. Mitotically it will grow. Mitotically it will grow into a multicellular gametophyte. That's the story here. Got that? It will grow into multicellular gametophyte. That's the story here. Got it? Right? This is what the haplontic life cycle is. So haploid phase is dominant. When I'm saying in the plant life cycle a phase is dominant, means it is the gametophyte which is photosynthetic, which is independent. Obviously, if you will start earning your money, you will be independent now. You don't need approval, right? You are independent. You can buy something if you want to. So gametophyte is the dominant phase here, means this is the photosynthetic. This is the independent phase. This is the one which is free living, right? This is the one which is free living. And obviously, this gametophyte is multicellular here. Now, if I talk about the sporophyte, that is the diploid generation. So sporophyte, the diploid generation. So sporophyte, which is the diploid generation, So this sporophyte is zygote is showing the sporophytic generation. So zygote is the first and the only cell of sporophytic generation. Why only cell here? Because it is the one that is undergoing the reductional division. So zygote, which is the first and the only cell, so zygote is dependent on gametophyte for its nutrition and encourage, for its attachment and nutrition. Got that? So zygote is undergoing meiosis. So is also called zygospore, because the zygote is undergoing meiosis. So now you remember the story. Spores, they germinate to form gametophyte, gametophyte form gamete, gamete fuse, they will form zygote, zygote, right? Zygote is the sporophyte, yeah, first cell of sporophytic generation. It will either it grows into the diploid phase, or here in algae or in haplontic life cycle, it is undergoing meiosis. So this is the life cycle here. So you see it in most of the algae. Most of the algae. I am still saying that not all the algae, because the brown algae will not show it, but otherwise in most of the algae, this is what you are going to see. So please quickly revise it. Please quickly revise it and do let me know if there is any doubt. Quick. 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 Guys, quick. Done? So this is about the haplontic life cycle. So haploid phase is the dominant one here. The diploid phase is for the sometime shown by the zygote, which is further dependent on the gametophyte itself. And you know it will also undergo this meiosis. Then comes the diplontic life cycle. Obviously now it's a different life cycle. Where do you see this? In the case of gymnosperms and angiosperms. Where do we see this? In the case of gymnosperms and angiosperms. And we have some algae also that shows. There's a fucus. Fucus is it's a brown algae. Yes, students, it's a brown algae that is also having the diplontic life cycle. Now let's discuss it. So as the name is suggesting, it is the diplontic life cycle. So diploid phase, that is sporophyte 2N, it is dominant, it is independent. In gametophyte was dominant and independent. So it is dominant, independent, free living, multicellular structure. है ना? Multicellular structure. It is differentiated into, like not in the case of fucus of course, but otherwise this is the sporophyte here. Just say, let's suppose, right, this is in the case of a angiosperm, so sporophyte 2N. Right, so it is the one which is differentiated into true root, stem and leaves, except in the case of fucus, which is itself an algae. So now gametophyte, which is one or few celled, is dependent on sporophyte for its nutrition and encourage. So in haplontic life cycle, right, the gametophyte is dominant, sporophyte, which is represented by one and only cell zygote, right? So this one here, right? So this is what you have to remember. So here on in the case of diplontic life cycle, it is the diploid phase, that is the sporophyte, which is dominant, which is independent, which is free living, which is multicellular, right? Obviously, why is it photos independent? Because it is photosynthetic. So gametophyte is dependent on it. So gametophyte is one or few celled. In some cases it can be few celled, otherwise one or few celled it is. So it is dependent on sporophyte for its nutrition and encourage. So now look at this. Just say, let's suppose this tree is representing a sporophyte. Obviously you know that not complete tree is going to turn into the spore. It will be having the structure in which meiosis will occur and they will form what? They will form spores. So spores, we know it very well, they are haploid. Spores, we know it very well, they are what? These spores are haploid. Now, you know that, already told you this, that spore will germinate. Spore will germinate and it will form the gametophyte. Spore will germinate and it will form the gametophyte. Is that clear? Gametophyte. So this gametophyte is dependent on sporophyte. It is not independent, and we know that this gametophyte, by mitosis, it will form male and female gametes, which will further fuse, which will further show fertilization, and you will see the zygote, students. But here what is the different thing? See, zygote, because ultimately main plant body is what? Diploid or haploid? Which one is the main plant body? Is it diploid or haploid? Diploid. So this zygote will not undergo meiosis, rather mitotically it will grow into sporophyte. Mitotically it will grow into sporophyte. That's how it is. So in the haplontic life cycle, what happened is main plant body, just say in the case of majorly algae, they will show that, right? So this haploid body will form mitotically gametes, which will fuse, which will form zygote. But here zygote will undergo meiosis. Why? Because main plant body itself is haploid. So there is no point of the zygote to develop into a multicellular structure, because ultimately it has to undergo the meiosis. So it will go directly, it will go the meiosis here. So because here it is undergoing the meiosis and the spores are formed due to meiosis, so that's why we are calling them as meiospores, right? Because they develop due to meiosis. Okay, so here what is happening? So this zygote, as I said, undergoing meiosis, they germinate mitotically to multicellular gametophyte. That's what is happening here. Now, in this case, what is happening? Gymnosperms and angiosperms, they show the diplontic life cycle. We also have an exception, fucus brown algae, diplontic life cycle. So, diploid phase, that is the sporophyte, is the dominant, independent, free-living multicellular phase. It will be, right? It will be there in the majorly, it is the one which is there in the life cycle. So, gametophyte, one or few celled, it is dependent on sporophyte for its nourishment. So, gametophyte, we know that will depend on sporophyte, mitotically will form gamete, will fuse, zygote. Here, zygote is not undergoing meiosis, rather it is growing mitotically into the sporophyte, and then you will have the new sporophyte. Is that clear? Is that clear? So spores will develop into, so zygote will develop into a sporophyte, right? That's how it is. Any doubt? Any other doubt? Any other doubt? Sure? So next is what? Next is the haplodiplontic life cycle. Next is what? Next is haplodiplontic life cycle, students. Haplodiplontic life cycle is the next part that we have. So here, it is an intermediate condition. What sort of condition it is? It is an intermediate condition. Okay. So here you will have the diploid, both the phases are there for a decent amount of time. So haplodiplontic life cycle is present in bryophytes and pteridophytes. It is present in bryophytes and pteridophytes. Are you getting my point? Right? Even some brown algae also show this. So, in bryophytes, so there are some differences as well. No doubt it is present in these two, but there are some differences as well. Now, when I'm talking about the bryophytes, I told you already that in bryophytes, there will be no vascular bundle. There will be no vascular bundle. No seed is there. I hope you remember this. है ना? So here, which one is the dominant phase? Here, dominant, comma, photosynthetic, comma, independent, comma, free living, multicellular phase is your gametophyte. Yes, just like algae, here also main thallus is gametophyte. Now, sporophyte, which is, so gametophyte ultimately here is what? It's long-lived, right? Because it is an intermediate condition. Both the phases are there for a decent amount of time, but it is the gametophyte which is long-lived. So what about sporophyte? So here you will see sporophyte is again multicellular. Just in algae, sporophyte was single celled. Only zygote was representing the sporophyte in the case of algae. But here sporophyte is multicellular, but totally or partially. Like in some cases totally, but in some cases partly, dependent on gametophyte for its food and nutrition, right? For its food and nutrition. This is what we will see here. Now, let's see. If I'm making a life cycle, I'll show you the life cycle here. So just let's say this is a bryophyte. So as I told you, haploid phase is the dominating phase. Hai na? So haploid phase is what? It is your gametophyte. Why? Because all the cells are haploid here. All the cells are haploid here. Are you getting my point? So bryophyte mein it is the gametophyte and it is also thalloid. Like it is also not differentiated into true root, stem and leaves. It's better than algae, but not undifferentiated into, not differentiated into true root, true stem and leaves. Now, you know that it is the gametophyte. What will it form? Mitotically again, mitosis. It will form gametes. Where do that gametes form? Obviously in the sex organs. Mitosis will occur, beta. Gametes will form. Now, you again know that when there is the male or the female gamete, they'll fuse. They will form the zygote 2N. So here, just in algae, only in haplontic life cycle. Only in haplontic life cycle, you people will see that a zygote is undergoing meiosis. Here also, zygote grow mitotically into a multicellular sporophyte. So if life cycle is clear, rest everything will be easy for you then. So it will grow into a multicellular sporophyte. Say here you will see, right, the sporophyte will be there. So in this sporophyte, further meiosis will occur, haploid spores will form, and that haploid spores, they germinate, right? Again they grow, they germinate, again they grow into gametophyte. That's how it is. So in algae, zygote was immediately undergoing meiosis, but here it is showing it is growing, it is making the multicellular structure. So it was growing mitotically into a multicellular sporophyte, right? This is what you people can see. It is forming the meiosis, in sporophyte, right? We will see with time that it is showing foot seta capsule. So there will be meiosis, haploid spores will form. That's how it is. So you can even compare. Even in algae, gametophyte is the dominant phase, right? Here also gametophyte is the dominant phase, right? But in algae, sporophyte was represented by single-celled zygote. But here, sporophyte is short-lived and it is multicellular. It is totally or partially dependent on the gametophyte for its food and nutrition. So there you people can also see. So now any doubt? Yes, please. Any doubt? Any doubt? Guys, quick. Done, students? So this is in the case of bryophytes. Is that clear? Is that clear? Yes, please. So no vascular bundles, right? No seeds there. So this is the dominant photosynthetic independent free living multicellular phase is gametophyte. It is again represented by a thalloid-like structure. Sporophyte, which is short-lived, it is multicellular, totally or partly dependent on gametophyte for its food and nutrition. So that's how it works. Is that clear? That's how it works. Is that clear? So next part is what? In pteridophytes, because in pteridophytes also, right? In pteridophytes also we have, which phase do we have? Haplodiplontic life cycle is there, right? In pteridophytes, haplodiplontic life cycle is there. What is there? Haplodiplontic life cycle is there. But here in pteridophyte, students, dominant, photosynthetic, independent, dominant, photosynthetic, independent, free living, and even long-lived phase. It is represented by gametophyte. It is represented by gametophyte. Is that clear? Is that clear? So it is represented by what? Gametophyte. So beta, the gametophyte here will be, because this is a pteridophyte. So oh sorry, my bad. It is not gametophyte, it is sporophyte. It is sporophyte. So here you will see, yes, not like algae or bryophytes. Here it is sporophyte. Okay, what's that? It is sporophyte. Are you getting my point? So here this sporophyte is basically having, in pteridophytes, this is what you are going to see. Yes, students, in pteridophytes you will see that sporophyte is having true root, true stem, true leaves. This is what we see in the case of pteridophytes. They have true root, true stem, true leaves, right? They have vascular tissue, right? They are the first plants having the vascular bundles. So yes, the plants having the vascular bundle, they are considered as tracheophytes. So yes, pteridophytes are the first tracheophytes. So what do you see here? Right, that dominant, photosynthetic, independent, free living, and above all, long-lived, long-lived phase is the sporophyte. So it is dominant, it is photosynthetic, it is independent, it is free living. So it is the Sporophyte. So sporophyte is having true root, true stem, or true leaves. So vascular tissue is also present here. So this is the tracheophyte. Is that clear? Done. Now, gametophyte, again, here also gametophyte. This is the speciality here. Kindly pay attention. Gametophyte here is, no doubt, this phase is short-lived. So, but gametophyte again is independent. It is free-living. It is photosynthetic. So you cannot say that here gametophyte will be dependent. Not at all. Not at all. So gametophyte here is short-lived. It is independent. It is free-living. It is photosynthetic, but it is just that it is short-living. And of course, it is multicellular. It is multicellular. That's the different part here. That is the irony here. Is that clear? Is that clear? So here you will see the gametophyte, which is the free living, which is heart-shaped. So that's what we have to remember in the photo, in the pteridophytes. So if we have to see the life cycle here, but do underline this part, beta. It is very, very, very important. So multicellular, photosynthetic, free living, independent, and short-lived gametophyte is there. So that's how you can see. Just let's suppose. So just let's suppose here you have in the case of pteridophytes, so here you can see the dominant phase is sporophyte. So as I told you already that sporophyte is having what? Sporophyte is having true root, true stem, true leaves. This is what they have. True root, true stem, or true leaves. Now, sporophyte, it will undergo, like there are structures which will undergo meiosis. You know that. They will form what? They will form spore then. You know that spore has formed due to meiosis. It is haploid. It germinate and will form gametophyte. It will germinate and will form gametophyte. So as I told you again, gametophyte is multicellular. This one is also independent. This one is also free living. But yeah, it is short-lived. It is not dependent on the sporophyte. So gametophyte will form gametes, male and female gametes. We know it very well. They'll fuse. Zygote will form. Again here only in the haplontic life cycle zygote is undergoing the meiosis. Here also mitotically it will grow. It will become multicellular structure, right? It will become multicellular structure, and then finally it will grow into a sporophyte. This is how it works. This is how it works. Is that clear? Right? Sure. So this one is dominant, this one is photosynthetic, this one is long-lived. Understood? Sure. Quick. Done, beta? Sure? Done? So this is about the pteridophyte. So this is all about the life cycle. So let's draw So here let's write about algae, bryophytes, pteridophytes, gymnosperms, angiosperms. Now, in the case of algae, which life cycle do we have? Haplontic life cycle, right? Which life cycle do we have mainly? Haplontic life cycle. And as I said, exceptions are there, right? Exceptions are there. So which one is the dominant phase? Which one is the dominant phase? So dominant phase is gametophyte, right students? Here, sporophyte, who's representing it? Zygote, right? So it is dependent on gametophyte. Understood? Now, bryophytes, the life cycle that we have is haplodiplontic. Here, what will you see? You will see that dominant, comma, independent, comma, free living. So dominant, comma, independent, comma, free living, photosynthetic, long-lived, gametophyte. So sporophyte, partly or completely dependent on gametophyte. It is multicellular but short-lived. So this is a comparison table. Then comes the pteridophytes. Again, here also life cycle is haplodiplontic. And here, in both the cases, it is going to be diplontic life cycle. So here you people will see that dominant, independent, photosynthetic, long-lived is sporophyte, which is 2N, and gametophyte is also there, but gametophyte will be short-lived. So it's short-lived. It is also photosynthetic. It is also free-living. It is also independent. Now here, dominant, independent, multicellular, sporophyte. One or few celled, dependent is your gametophyte. So this is the comparison table, everybody. So let's revise the life cycles of plant here. Okay, so let's revise the life cycles of plant. That too very quickly. You people can see algae. Majorly algae shows the haplontic life cycle. Exceptions are there, like your fucus, like your ectocarpus. They do not show the haplontic life cycle. Then here the dominant phase is the gametophyte, which is a thalloid, which is multicellular, which is the one which will be free living, photosynthetic. Then sporophyte, which is represented by a zygote, right? Only zygote is the first and the only cell of the sporophytic generation here. It is dependent on gametophyte for its food and nourishment. It will undergo meiosis. Now, the next life cycle that we have is of bryophytes. So bryophytes are haplodiplontic here. The dominant, independent, free-living, photosynthetic, and even long-lived phase is the gametophyte. So gametophyte is the long-lived phase here. So sporophyte is partly or completely dependent on gametophyte. It is multicellular. It is short-lived. Next to it is the pteridophytes. So here in pteridophytes, it is the haplodiplontic phase, so life cycle which is present. Here, dominant, independent, photosynthetic, long-lived phase is sporophyte, which is having two roots, stem and leaves. So gametophyte here is short-lived, but yeah, that's the point where you need to put the emphasis. It is, gametophyte is not dependent on the sporophyte here, right? It is photosynthetic, it is free-living, even it is also independent. Now, when we move to the gymnosperms and the angiosperms, so what do we see there? We see the diplontic life cycle. So it is dominant, independent, multicellular phase is sporophyte, right? Having true root, stem and leaves, and one or few celled is your gametophyte, which is dependent on the sporophyte. So that's how you can revise it. Now, the next part that we have to start is the algae in detail. Already told you about the tricks. I hope it's done, right? I hope it's done. So let's get started. So we are talking about the chlorophyceae, phaeophyceae, and the rhodophyceae. It's green algae, brown algae, and the red algae. That's what we have. Green algae, brown algae, and the red algae. US has very clean college. So Eulothrix, Spirogyra, Volvox, Chlamydomonas, Chlorella, Cara. Right, self-DP for the brown algae. Sargassum, Actocarpus, Laminaria, Fucus, Dictyota, and Phaeophyceae there. Here, P2G2C, Porphyra, Polysiphonia, Gracilaria, Gelidium, and Chondrus is the case. So Fucus, these are the exceptions. It shows a diplontic life cycle. It's an algae. It shows the diplontic life cycle. Am I clear? It shows what? It shows the diplontic life cycle. Understood? Understood? Right? So your Actocarpus, haplodiplontic life cycle. So that's what you have to remember. So now the next part is, let's start the algae, which is having different shapes, which are, very important, of course. So when we talk about the plants, we start with them, okay? Okay, but here one more thing that you all need to remember. Just like Chlamydomonas and Chlorella, they are unicellular. They are unicellular eukaryotes. They both are. So as per that 5 kingdom, they are kept in the kingdom Protista. But why do we study it here in the plant kingdom? Because their life cycle resembles here with the plant. That is why we study it here. So now we will start the algae. Just look at this. Study of algae, phycology. Important. It is important. Study of algae is phycology. Phycology. So father of phycology, Felix Eugene Frisch. Father of Indian phycology is M.O.P. Iyengar. Now when you move to this part, you can see this diagram. It is this is Volvox, right? You know that we have read it under the green algae. So this is Volvox, a colonial green algae, students. And here you will see which cells are making it. Chlamydomonas. They are together making this Volvox. So now let's understand it one by one. You can see the Chlamydomonas, which is unicellular. So I told you it is kept in the kingdom Protista, but its life cycle resembles here. So that's why we study it here. This is the Volvox, multicellular. So it is having the daughter colony with fixed number of cells. With what? With fixed number of cells. Yes, please. With what, beta? With fixed number of cells. So that is why we also use the word synobium here. What is the term that we use here? Synobium. Then Spirogyra, multicellular filamentous. You can see kelp. Kelp represent what? They represent the brown algae. What do they represent? They represent the brown algae. Am I clear? So let's discuss it. So these are the different examples here, right? So let's get started. So algae is what? Algae is predominantly aquatic. Mainly it is what? It is aquatic actually. So both freshwater and marine water. So some are terrestrial and grow in moist places, soil pe, wood pe. Even they can grow on the ice and the snow. And the one which are growing on that, you can call them as the cryophytes. This is the term for them, right? So it occurs in association with fungi as well. We know that this symbiotic relationship is what? This is lichen. So on the sloth bear as well, right, this algae can grow. Is that clear? Is that clear? Yes, everyone. Yes, everyone. Any doubt? So here I would like to specify one thing. Just even in the NCERT, right? In NCERT, sloth bear is given. Sloth bear is different, right? Sloth is different. Sloth and sloth bear, you check it, they both are different. So one of my students, she told me this, okay? She checked it out. We all remember it like an example. She checked it out. So actually it is the sloth on which algae grows, right? It helps that sloth to show the camouflage. Yes, please. It showed that it helped that sloth to show the camouflage, and because it is having the moisture in its fur, so algae is capable of growing it. So that's actually no doubt in NCERT they have mentioned sloth bear, but truly it is what? But actually it is what? It is sloth. Actually it is what? It is sloth. Is that clear? So some algae are also parasitic, like Cephalurus, which is the example of green algae. So its common name is red rust. It affects the tea plant. Okay, it affects the tea plant. Is that clear? Now the next point is algae. You know that here also it is thalloid, right? The plant is undifferentiated into true root, stem and leaves. And here you will see that algae, its cell wall is not just having cellulose and pectin, but it is also having the galactins, mannins and minerals like calcium carbonate. Understood? So it is having what? It is having the galactins, mannins and the minerals like calcium carbonate. This is an important point. Now move to the next part. 3 types of reproduction is there. Vegetative reproduction will include fragmentation, like a part of it will get separated, will regrow into a new plant. Then you have asexual reproduction and the sexual reproduction. So next to it is what? In asexual reproduction, mainly what is happening, it is the spore formation. Spore, always remember, it is going to be haploid. But this is not developing due to the sexual reproduction. The spore here is developing, forming due to the sexual reproduction. So the main spores, the one which are given in your NCERT as well, I have read the I mentioned the word main here as well. This is zoospore and aplanospore. Zoo word, you know that it is associated with animal, and animals, you know that they are motile, they are movable. So when I'm saying zoospore, which is a very important thing, here you will see flagella is present. What do you see? Flagella is present. What do you see, guys? That the flagella is present, so they are the motile spores. So yes, they are flagellated, they are the most common spores, they are the thin spores, so they are the motile spores. Yes, you have to remember this. Even in fungi also you are going to see the zoospore. Then aplanospore, they are non-motile, they are thin-walled. Now, whenever you talk about any of the spore which is thick-walled, in the case of plant, if you are having these thick-walled spores, so they act like they help in perennation. What is perennation? Right? Yeah, organ of perennation, yeah, structure of perennation that help to survive under unfavorable conditions. That help to survive under unfavorable conditions. That help to survive under what? Under unfavorable conditions. So let's talk about this, right? Let's talk about this. So you have zoospore, you have aplanospore, and you have hypnospores as well. So hypnospores, they are beta non-motile. So when it comes to the motile spore, you only talk about this one, that is the zoospore. So this information is literally very important. So they are non-motile, they are thick-walled, they help in perination. So perination means it will help to survive under unfavorable conditions, right, students? So you have zoospore, different type of flagellation is there, aplanospore, hypnospores that are having the thick walls. Now let's go ahead. So even in during the asexual stages, palmilla stage and echinids, they also form. So what is this palmilla stage? Yeah, what are these echinids? Palmilla stage, in this case, the parent cell, right, from that algae, that parent cell, if it is having flagellite, is going to lose that. It will become circular, it will acquire a round shape, and it will start secreting the thick mucilaginous covering, right? So obviously that will also help in perination. That will also help it to survive under the unfavorable conditions. Am I clear? Am I clear? So next is echinid, which are the modified vegetative cell, students, with lots of food reserve and thick walls that help in perination. So they are the modified vegetative cells. They are having the food reserve and they are having the thick walls as well. So flagellated spore is only one, the most common one that we have discussed, that is your zoospore. Next is the sexual reproduction that we have to discuss here. So we know that where that gametes will form. Gametes are required for the sexual reproduction. Where that gametes will form? In the sex organs. That is obvious, right? That is obvious. But here what you have to remember, that the sex organs are unicellular, students. They are non-jacketed. Non-jacketed means that these sex organs are not covered with any protective layer, right? No doubt an exception is there, and exceptions are always important. So exception is Cara, which is also known as stonewort. Right, so here you will see sex organs are multicellular and jacketed, otherwise they are not. Otherwise they are not. Understood? So Cara, this diagram used to be now it is not there in class 12th reproduction in organism chapter. So there is a PYQ on the basis of this Cara as well. Now so that chapter is deleted. So this diagram was once there in the paper. So they even asked which one, upper one is the female part, yeah, lower one is the female sex organ. So female sex organ is oogonium, male sex organ is antheridium. So yes, in the paper they have asked the questions like this also, right? So sex organs are basically unicellular, right? Sex organs are basically unicellular. So beta, antheridium is for the male sex organ, oogonium is the name for the female sex organ. You say anther is the male part, right? Anther is the male part, same like this. Okay, okay, done. So next is what? So here you can see this is Cara, right? And this is Cara, okay? This is what you can see. Now, the next to it is what? That algae. You know that it reproduces by zygotic meiosis. What's the meaning of zygotic meiosis? That zygote is undergoing the reductional division. So, of course, here I can say no embryo is there. But yeah, we do have exception. And when it comes to the exception, you will talk about the brown algae. So, in brown algae, embryo is present, right? In brown algae, embryo is present. That's important. Done. So types of sexual reproduction, again a very important topic here, right? Again a very important topic here. What? The type of sexual reproduction. So iso mane beta same type of gametes are there. If one is flagellated, another one will be. Their size, shape, everything, right? Their size, their shape, everything is what? Everything is what? Their size and shape, everything is same. So flagellated gametes, which are similar in size, right? Just like chlamydomonas, it shows the isogamy. Though chlamydomonas are present in all three types of sexual reproduction. So in case of non-flagellated, matlab gametes can be flagellated and similar in size, isogamy. They can be non-flagellated as well. So if they are non-flagellated but again similar in size, example is Spirogyra, which is again the green algae. So do remember the example, just say Actocarpus. It's a brown algae. It is having motile gametes, movable gametes, right? Flagellated gametes, again showing isogamy. Your Spirogyra is having non-flagellated gametes showing isogamy. There is one more example, that is of green algae, Cladophora. Here also gametes are motile, but here they are identical in shape and size, but physiologically they are distinct. So yes, they are going to ask you the questions right from this part. This is a very important part. Got it? Got it? This is a very important part. So types of sexual reproduction, isogamy, the gametes are similar in size. So chlamydomonas, even the ectocarpus, even the spirogyra, cladophora, they are the examples. But what you have to remember, it is also given in NCERT that spirogyra is having the non-flagellated gametes. Chlamydomonas is having the flagellated, and for ectocarpus and all also, I have mentioned the points there. Do remember it. Okay, next, so see, isogamy, female and male gametes both are like same. And isogamy, here also you will see there is a mainly there will be the difference of the size. If one is flagellated, another one in an isogamy, but the size is different. In oogamy, this condition is fixed, that the female gamete is going to be large, it is going to be non-motile. Male gamete is going to be small, it is going to be motile. Okay, it is going to be motile. Got it? So let's see this. So anisogamy, fusion between two gametes. So here what will you see? These gametes are dissimilar in size. What are they? They are dissimilar in size. So both gametes are usually motile, but one is larger than another. So as I said, what is the main difference in isogamy? In anisogamy, the main difference is of the size, but both can still move. So Eudorina, again, it is the example which is given in NCERT. Again, which is the example given in NCERT, Eudorina. It's a green algae, even Chlamydomonas species. So oogamy, which is again the subtype of anisogamy, one gamete is larger and non-motile, that is going to be the female gamete. Other is going to be small and motile sperm. So Chlamydomonas, Sargassum, Volvox, Fucus, Cara, they are the examples. So examples here are very important. Am I clear? Example here are very important. So now let's talk about the green algae. Okay, so ultimately there is a table. Yeah, most advanced is the oogamous. So ultimately, beta, there is a table here, right? There is a table here which you need to remember from where they can ask you the question. Rest, you keep listening to me. The notes will be shared in the Telegram group, right? So let's have a look. So basically, what is the first part that we have? We have chlorophyce, right? Which is also known as green algae. Which is also known as green algae. So this green algae, this is the most advanced one, and they are believed to be the ancestors of the higher plants. So it is the most advanced one and believed to be the ancestor of the higher plants. So any type of plant body can be there. So unicellular green algae mein, you have the examples like Chlamydomonas, which is overall, which is a motile. I'm not talking about the gamete, about the structure, motile. Chlorella, non-motile type of algae. And there is Acetabularia example also, which is the largest unicellular plant. It is colonial. Again, a PYQ. Volvox is the case. You have seen that. So in the Volvox colony, the cells here are of Chlamydomonas. And you people will see right here in between, daughter colonies will also form. We use the word synobium for it. What is the word that we use for it? Synobium. Why do we use the word synobium here? Because the daughter colonies is having fixed number of cells. Okay, next is filamentous algae. You have the example of Ulothrix and Spirogyra. So now you remember the examples, right? So this is going to be, right? So this is going to be easy for you. So they have rigid cell wall, but in green algae, what do you see? In green algae, you will see that the inner layer is having cellulose, outer layer is having pectose. Inner layer is made up of cellulose, outer layer is made up of pectose. So in the case of green algae, you have chlorophyll A, chlorophyll B, even other than that, right? You have other pigments also, but always remember it is the chlorophyll A which is referred as universal or the major pigment. It is present in all the photosynthetic, right? It is present in all the photosynthetic plants and animals. So do remember it. Is that clear? Do remember it. So in the case of green algae, chloroplast will show the different Shape, students, if it is cup-shaped, C for cup-shaped, C for chlamydomonas. If it is spiral, S for spirogyra, or ribbon-shaped, if it is spiral or ribbon-shaped, spirogyra, very important. Reticulate means just like a network, it is going to be Oedogonium. Girdle-shaped is in Eulothrix. So other photosynthetic pigments are also present, told you already, right? Jaise ki here you will see carotenoids are there, right? But जैसे carotenes, xanthophylls, but fucoxanthin and phycobilins are absent in green algae. A very important line, a very important line. So fucoxanthin, that gives brown color to brown algae, and these phycobilins are absent. Understood? And these phycobilins are absent. Understood? Understood? Sure? Any doubts? Any doubts? Sure? Chaliye. So next part that we have here is, yes please. So here you see that in most of the members, here the food is stored in the form of pyrenoids. So these are the quick points that you have to extract. So pyrenoids means they have the proteins in addition to starch. So in green algae, what we have concluded so far, they are the most advanced one, believed to be the ancestors of green algae, have different shapes, being the ancestors of the higher plants, have different shapes, have chlorophyll A, B, carotenoids are also there, but they do not have fucoxanthin and phycobilins, right? Chloroplast is of different shape, right beta? This is what we have discussed. So food, cell wall mein pectose and the cellulose is there, and here the food is stored in the form of pyrenoids in the chloroplast. So some algae, they can also have the oil droplets, but here you will see that the food is stored in the form of pyrenoids. So it is contain the proteins in addition to starch. So vegetative reproduction may usually take place by fragmentation. Asexual reproduction, it is occurred by flagellated zoospores, right? It occurred by flagellated zoospores produced inside the zoosporangium. Now, if there is the formation of any spore within a structure, what is the name of that structure? The name of that structure is sporangium. Sporangium. If there is the formation of a spore within a structure, you will refer that spore as endogenous in nature. Right? Endogenous in nature means it will develop inside a structure. So here, because zoospore will form, so you call it as zoosporangium. Is that clear? So asexual reproduction occurs by flagellated zoospore, which are formed inside zoosporangia. So in the case of green algae, you will have 2 to 8 flagella, but they will be apical. Mostly 2, but it can be 2 to 8. They can be at the, they are present at the tip. Okay. Now, different type of sexual reproduction is there. All the 3 types are there, right? All the 3 types are there. So now these are the examples written in front of you, and just as Spirogyra, you know that it will show isogamy. Chlamydomonas show all 3. So that's your homework. That's your homework, right? Just say Volvox was showing what? Oogamy. So that's your homework. You will tell me that which one is showing which reproduction. Okay, so that's going to be an important part. Next part is the brown algae. Vegetatively very advanced, because it is not just the, you will see the very large plant in it, like kelps, right, which grow in the seas, like kelps. This is what you will see. What is the speciality here? That here you will see that the embryo is present. In the brown algae, we will see embryo is present. I'll give you an example. Fucus is the example here, diplontic life cycle. So if in fucus, diplontic life cycle is there, means the thallus of the fucus is what? Diploid. So it means its zygote is not undergoing meiosis. Even in ectocarpus, you will see, right? You will see haplodiplontic life cycle. So if in ectocarpus, haplodiplontic life cycle is there, again, the zygote is not undergoing the meiosis here. Are you getting my point? So that is what you have to remember in the case of, that is what you have to remember in this case. Am I clear? Sure? Sure? So give it a reading. Done? So not just the ectocarpus, you will also see the calps. Calps are also having what? Haplodiplontic life cycle. So exceptions are very important here. So yes, it found primarily in the marine habitats. You won't see any unicellular form, yeah, colonial form. So they are branched also. They have great variation in their size and form. So here also thallus is present. So thallus can be simple branched, filamentous, right? Just a filament like, it can be filament like ectocarpus. It can be highly branched calps. So calps are also showing haplodiplontic life cycle. Ectocarpus is also showing haplodiplontic life cycle. Fucus is showing diplontic life cycle, right? So overall, you will see that the plant body of highly advanced form will have more or less root-like structure. Matlab here, you know that they don't have true root, stem and leaves, right? They do not have true root, stem and leaves. Okay. Guys, do check it in your book as well, right? I missed to add this. No, I have added the diagram. I thought I missed it. So here you people can see the diagrams, right? You people can see the diagrams here, right? So look at this part. This is your fucus. In NCERT do market, it is having diplontic life cycle, right, beta? So that is your dictyota, having the which is dichotomously branched, right? So here you will see there is a hold fast in the case of brown algae that will help it to get attached. That will help in the attachment here. There is a stipe there, the stalk-like structure here. And here you will see frond. These are the leaf-like structure here. So they are not true root, they are not true stems, they are not true leaves, they are leaf-like, they are root-like, they are stem-like structures. Okay? Okay? Sure? Done? So this one is what? This one is laminaria. So this is what you guys can see. Okay. So fucus. Fucus may one more thing that you have to notice is that they have the air bladders. So that air bladders will help it in buoyancy. Right. That will help it in buoyancy. Is that clear? So let's discuss it. So diagrams, I hope they are clear because diagram based questions, they also ask in the final paper. So this is what we have. Where is it? Brown algae. This is what we are discussing. So you can see that they are highly advanced vegetatively, right? They are highly advanced. So they have root-like, not true root, hold fast for attachment. Tubular structure, which is basically the stipe or the stalk beta, and leaf-like. Fronds are there for the photosynthesis. So fucus or laminaria, right? Just now you guys have checked the images as well. So largest brown algae kelps, their genus are Nereocystis or Macrocystis. So it is also showing haplodiplontic life cycle. So mostly brown algae, they are attached to the stones, rocks, right? So we use the word lithophile for that. Some are free floating also like sargassum. So in beta North Atlantic Oceans, the sargassum, it grows too much. So that is also referred to as sargassosea. And that is also a hurdle to the ship that are moving there. Is that clear? Is that clear? So next is again cell wall, important to remember. Inner is made up of cellulose, but here outside you will see the mucilaginous layer, and that is having the phycocolloids, the one that can hold the water. So algin is present there. So if you will study their economic importance, right, if you will solve the PYQ, so algin is obtained from what? Algin is obtained from the brown algae. So it is a phycocolloid, yeah, hydrocolloid, right, the one which can hold the water. So it will protect that thallus from getting dried when there are low tides and the freezing under low temperature, right, and from the mechanical injury. That's how it protects. So algin is also used, but it is having the economic importance as well. So when it comes to the pigment, so this is brown algae. In the table also I will show you, right? You'll get to know that comparison. A is present in all. I told you all the photosynthetic organism will have the chlorophyll A. It is a major pigment. C is there. So in brown algae, brown color is due to the xanthophyll fucoxanthin. It is the fucoxanthin which is giving it the brown color. That's what you have to remember. Now, stored food, another important question, another important PYQ, that is mannitol, all. So sugar alcohol it is, or laminarian. Vegetative reproduction में fragmentation can occur. And now when you talk about the zoospores here, so zoospores में you will see, right, flagella are present laterally. They are of two different types. So this is heterocont, right? They are not of equal size, right? So they are, no doubt, they are biflagellated zoospore. They are pyriform, माने pear shaped, right? They are pyriform structure, माने pear shaped structure. So they are two unequal flagella. They are laterally attached to this pear shaped structure. So sexual reproduction में you will see isogamy is also there, your ectocarpus, oogamy, fucus, laminaria, dictyota, and isogamy is also there, but that examples are not mentioned in our syllabus, so that's why I'm not talking about it. Okay, done. So no zygotic meiosis in brown algae. So this is what you have to remember. No zygotic meiosis is there. So diploid zygote, obviously that will develop the diploid thallus then, right? Because zygote is not undergoing the meiosis. So in diplontic life cycle, that will be the case. Okay, so it's many brown algae, dominant phase is the dominant one. So these are again the examples given in front of you. So fucus is the one with air bladders, right? So do check it, which one is showing which reproduction, very important. So algin, which is extracted from the kelps, right beta, I told you it is having the economic importance. So it is used as a thickener for making soaps, creams, even for the emulsifiers in ice cream or polish, right? So it is even used in the preparation of pills or ointments and in the dressing material. Next is the rhodophyceae, very interesting here. It can even grow in the deep oceans. Why? We'll tell you. So predominance of the red pigment. Why do they have red color? They have chlorophyll A, they have D and D. A is present in all. It is also having the D. So red, rhodo, is the thing. So it is the Floridian starch. The stored food here is what? Floridian starch. So this is the algae in which there will be no motile stage in life cycle. No motile stage in its life cycle. That's what you have to remember. No motile stage in the life cycle is there. So predominance of red pigment is there. So R phycoerythrin is responsible for that. It is not just the phycoerythrin, phycocyanins are also there. Okay, phycocyanins are also there. Understood? Sure? Yes, please. Okay. Done. So majority of red algae, it is in marine water, but I'm not saying that all will be in marine water. We have the example of Batrachospermum, right? So Batrachospermum, right, also known as frog spawn algae, it is found in fresh water. It's a very old PYQ, not of recent times, right? Not of recent times, but it is a very old PYQ. Okay, so they occur in both well-lighted regions, which are very close to the surface, and also at the great depth in the oceans where the little light can penetrate. Why is it so? Due to the pigment of phycoerythrin. But this phycoerythrin, it's the speciality of this, right? You know that different pigments, they absorb different wavelengths of light. So this phycoerythrin can absorb blue-green part of the sunlight as well, and this blue-green part, due to its shorter wavelength, it can penetrate deep into the oceans. It can reach into the deep layers as well, and because phycoerythrin is present in the red algae, right, so that can also absorb that wavelength of light, can show the photosynthesis. So if you will see the red algae, which is growing very deeply, it will have more phycoerythrin. Why? Because it has to absorb the sunlight, these shorter wavelength. So in that case, the red algae, which is growing very deeply in the oceans, you will see it will be having very dark red coloration due to this more and more phycoerythrin, that sometimes it start appearing like black Okay. Okay, so that is why red algae can grow, can reach maximum depth in the sea where no other algae can grow due to this phycoerythrin which can absorb the shorter wavelengths. Is that clear? Now when it comes to the red algae, usually multicellular, one example of unicellular is Porphyridium. Do not confuse it with Porphyra. Porifera is different. It is Porphyra, but here unicellular is Porphyridium. Ribbon-like is Chondrus. From this we will talk about the carrageenan, important compound here. So cell wall will have cellulose, will have pectin, and polysulfate esters are there. What do we have in the cell wall here? Polysulfate esters. So phycocolloids or hydrocolloids, which agar-agar or carrageenan is present here. So carrageenan is not obtained from the green algae. It is obtained from the red algae. Got that? Right? So red algae may also secrete deposits like calcium carbonate that appear like corals. So corals we studied in the Cnidaria. So some red algae can also secrete such things. So overall, motile flagellated forms are absent in the life cycle. Right, as I said, chlorophyll A and D is there. Carotenoids are there. These water-soluble phycobilins are there. So phycoerythrin will give red color. Phycocyanin will give blue color. Jैसे mollusca are having blue blood, they have hemocyanin. So same way. So food is stored as Floridian starch, which is quite similar to amylopectin and glycogen in structure. Why is it so? Why is it so? Because this one is also branched. It is also polymer of glucose, yeah, polysaccharide of glucose. That is why. Are you getting my point? That is why. Right? So yes, its structure is similar to the amylopectin and glycogen because both are highly branched, both are the polymers of glucose. So that is why. Okay? Right? That is why. So it is more similar to this glycogen because it is also Floridian starches, highly branched. Okay? It is also highly branched. So vegetative reproduction में fragmentation is there, asexual reproduction में non-motile spores, because in the starting itself I have clarified they do not have any motile structure. When it comes to the sexual reproduction, non-motile gametes are there. So oogamous accompanied by complex post-fertilization changes. So here you have Porphyra, Polysiphonia, Geladium, Gracilaria. So this Polysiphonia also show haplodiplontic life cycle. Haplodiplontic life cycle. Am I clear? Yes, please. And from this, so from this chondrus, this carrageenan is obtained. From this Geladium and Gracilaria, agar-agar is obtained. Am I clear? Agar-agar is obtained. Right? So P2G2C is the trick here. So C here stands for chondrus, from which the carrageenan is obtained. Right? So Porphyra is edible. Right? So chondrus is also referred to as Irish moss. It's a false moss beta because mosses, they belong to bryophytes and it is an algae. So carrageenan is obtained from it, which is a gelating agent in food industry. As an emulsifier also it is used. So overall algae is having the economic importance, that is half of total carbon dioxide fixation is carried out by them. They increases the level of dissolved oxygen. Edible one is Porphyra, Laminaria, Sargassum, and among 70 species of marine fishes. Again a PYQ. So water holding substances, these hydrocolloids, जैसे algin is obtained from brown algae, carrageenan is obtained from red algae, particularly I told you chondrus. So alginic acid is acting like a thickener, emulsifier it is, right? It stops the bleeding, used in the preparation of pills and ointments. So carrageenan is extracted from chondrus. So told you about this as well. So agar, we know it very well, beta. So it's a commercial product obtained from Geladium and Gracilaria. We use it in lab to grow microbes and the preparation of ice creams. So one more green algae example, this is chlorella. So chlorella is a unicellular green algae. Which green algae it is? It's a unicellular green algae. Which green algae it is, students? It's a unicellular green algae and it is carried by the space travelers as a food. Why is it so? Because it's a good source of protein. And not just this, right? So chlorella. So chlorella is a unicellular algae rich in protein used as a food supplement by the space traveler. So there is one more question, like there is a PYQ on the basis of that. One more is spirulina, but that is a BGA, blue-green algae, means that belongs to kingdom Monera. That is also a good source of protein, right? So they both are carried by the space travelers as a source of protein. Is that clear? Beta, what is emulsification crystalina? You'll get to know the answer of emulsifier then. Chale. Next is bryophytes. Study of bryophyte is bryology. Bryophytes are also referred to as the amphibians of plant kingdom. Why do we call them as the amphibians of plant kingdom? Because they started taking a step towards the land habitat. But they were not truly the land plants. Why? Because they are dependent on dependent on water for the sexual reproduction. So who's the father of Indian bryology? Professor Shivram Kashyap. Right? Professor Shivram Kashyap. Right? So first non-vascular terrestrial plant it is. Because they are non-vascular, they do not have xylem and phloem. They do need the moist, shaded, damp habitats. So they are also multicellular. They are having diploid sporophytes. They live as a parasite on an independent multicellular haploid gametophyte. So guys, if you know the life cycle, then only these points will be easy for you. So we have discussed the life cycle. Right? So here you know that it is the gametophyte which is the main plant body. What is the function of gametophyte? To make the gamete. Where will it make the gamete? Obviously in the sex organs. So sex organs are Antheridia and Archegonia, and unlike algae, here sex organs will be multicellular. Here they will be jacketed, means they will be covered, right? So Archegonia is there, Antheridia is there. So within the Antheridia, which is the male sex organ, antherozoids will form, which are the motile sperms, movable sperms, which are the biflagellated sperms. Done. And Archegonia mein egg. So they will form zygote that grow mitotically into embryo. Then the sporophyte will form. So here we will see, beta, sporophyte will have this thing. There will be foot, there will be setae, there will be capsule. Of course, not in all the cases. And here, beta, you will have, right, certain spore mother cells. So it is the mother cells, right? When mother cell is written, always remember it is going to be diploid, and it is the structure in which meiosis will occur. So meiosis will be there, spores will form. So in liverwort, spores will directly form gametophyte, but in mosses, indirectly they will form gametophyte. That's what we need to discuss. So let me complete this part, okay? And then I'll come back to the life cycle again, and by the end of this lecture, by the end of this chapter, you will realize that the life cycles that I have mentioned here, they are basically simplifying your topic, right? If you understand the life cycle, then it is the only part which you need to focus, okay? So you know it already that here the main body is gametophyte. Again, it is thalloid, but it is better than algae. Just it's like you got 30 marks in your exam, your friend got 35 marks. You both are not able to qualify, but obviously the one who got 35 marks is the, in comparison to 30 wala, is better performer. So it is like that. So now when you look at the structure, it is prostrate or the erect. Here also you have root-like structures. So here you will talk about the rhizoids, which can be unicellular, which can be multicellular. So here also no true root and stems are there. Here you will see rhizoids, which can be unicellular, which can be multicellular. Just say under bryophytes, we discussed two things: liverworts, hepaticopsida, and mosses. So liverworts will have unicellular rhizoid, mosses, right? Your bryopsida will have multicellular rhizoid. So rhizoids are root-like structures which will help it to get attached to the substratum. So yes, they have leaf-like or stem-like structures as well, right? So this rhizoid point is very important, right? This rhizoid point is very important. So roots are completely absent. They are replaced by unicellular liverworts, or multicellular rhizoids in mosses for attachment to the substratum. Done. For attachment to the substratum. So vegetative reproduction mein beta fragmentation, gemma, tubers, adventitious branches are there. So for us, this one is the most important one. We will discuss it here. Okay. Okay, so sexual reproduction mein already told you that is the comparison in between this and algae, multicellular and jacketed. So this jacket is sterile, it is not forming gamete. Just if I have to give you an example of the archegonium, just say let's suppose this is the structure. Here the gamete will form. So it is covered with the cells that are providing it the protection. They are itself not making the gametes, they are just providing the protection here. Got that? So archegonium, in which the female sex organ, which is flask shaped, in which egg will form. Male sex organ is antheridium, in which biflagellated motile antherozoid will form. It's biflagellated, it is motile. Is that clear? Is that clear? Chaliye next. So plants, they can live in soil, but antherozoid, they are motile. There is a purpose now, that's why they are motile. They need water. They are dependent on water for the sexual reproduction. That water will act like a medium by which they'll move and they'll find their way to the archegonium. So that's why, because they need water for the sexual reproduction, they are the amphibians of plant kingdom. Okay, so if life cycles are clear, everything is going to be crystal clear to you. So yes, archegonia, they release the chemicals to attract the antherozoid. They'll fuse, zygote will form. So zygote is the first cell here. This is important. First cell of the diploid generation, first cell of the gametophytic generation. So it will not undergo reduction, division immediately. It will not show meiosis immediately, just like algae, right? It develops, it grows mitotically. It forms a sporophyte, multicellular sporophyte, which is also the sporogonium, but it is not free living. It is partly or completely dependent on the gametophyte. So you will see here in the case of sporophyte, there will be a foot with which it will be attached to the gametophyte. There will be seta, there will be a capsule. So actually in this capsule beta, you will have spore mother cell, and that spore mother cell will undergo meiosis, and you will see that's how the spores will form. Understood? Spores will form. Understood? So sporogonium, which is basically foot, seta, capsule, right? So some cells of the sporogonium, spore mother cell, they are formed inside capsule, they undergo reduction, they form the spores, right? So meiosis is basically making the spores. Okay, so these are the bullet points here, the very important points here, will tell you the reason. So beta, in your liverworts and hornwort, Antherocerocopsida, spore will directly form the gametophyte, but in mosses, there is indirect development, protonema stage is formed. Now, let's suppose this is your spore. If this spore is directly growing into a thalloid structure, thalloid gametophyte, right? Thalloid gametophyte. So obviously it is going to be the direct development. Yes or no? But if, let's suppose, right, if that spore is not, rather it is developing into a creeping green filamentous structure, which we are calling as protonema, and then it is forming the gametophyte, which is a very unique feature here, right? So then it is the indirect development. Then it is the indirect development. So indirect development is in mosses, and the direct development is there in your liverworts. So overall, when you talk about the bryophytes, you people will see that there will be liverworts, there will be hornworts also. Hornworts are not given in NCERT, but yeah, in bryophytes we also have that. And we have the mosses, the pioneer community as well, beginner as well on the barren land with lichen. So Liverworts, mosses. This is given in NCERT. So this line is very important. Here spore directly form the gametophyte. There indirect development is there. Even here also direct development is there. Understood? So this is a very important point, guys. This is literally a very important point. Is that clear? Is that clear? So yes, there are two things that you study under the bryophytes. One is Hepaticopsida. Liverwort is associated with word hepat na. So Bryopsida, mosses. Okay, so let's talk about the liverworts first, where you have the example of Marchantia. So I'll start with the examples, then let's discuss it. So first of all, if here I have to give you a trick, so the trick is my first place secure. So my Marchantia, repeated, Funaria, Polytrichum, S. sphagnum, secure se sphagnum, R. rixia. So this is the trick that we have. Now, Marchantia is in the category of liverworts. Even your rixia is also in the category of liverworts. Rest all will be mosses. Right? Rest all are what? They are mosses. Little better than bryophytes. Number one. Now, when I'm talking about the Marchantia, beta, it's a very common sort of plant. Matlab, jaise wherever you have the water tank where it is leaking, right near to that walls, you find them growing. But we confuse them with what? We confuse them with algae. Are you getting my point? But we confuse them with what? We confuse them with algae. So Marchantia, they are, I'll show you the structure, they are dioecious. That's again a PYQ. Do underline it in your NCERT. That is, they are unisexual. So these are the rhizoid because they are the liverworts. So their rhizoids are going to be unicellular. So they have gemma cup, which is meant for what? Asexual reproduction. So these are that cup-like structure in which branched, stalked gemmae are present that help to grow. So Anthridiophore is the branch that is having the male sex organ. Archegoniophore, just like a flower it is. So it is having female sex organ. This is the example of the moss. Here, can you see this leafy part? This is a very unique feature of the mosses. Leafy gametophyte, students. And here you have the sporophyte attached to it, having foot, seta and capsule. So it is actually the capsule in which you have spore mother cells. Mother cells are always diploid. They undergo meiosis. They form the spore. So this is the another moss that you have. This is sphagnum. This is sphagnum. It is also known as peat moss. We'll talk about it, that why it is so. So diagrams need to be very clear. So it is the mosses. In their case, rhizoid is going to be multicellular. That's how it is. So mosses along with lichens, right? Mosses along with lichens, they are the pioneer communities. Pioneer mane beginner during ecological succession. So they basically convert that barren land into the soil, right? So that's how it is, okay? That is how it is. Done. So let me show you these liverworts here. So they are having these cup-like structures. These are that gemmae. These are that cup-like structures, right? Dichotomously branched they are. And mosses, that's how you see them, okay? That's how you see them. These are the mosses basically. They have these capsules. Later on they will develop, okay? Right? So diagrams from the NCERT, they are very important. So Marchantia, you have to know which one is male, which one is female. And when I'm talking about that gemmae, right, that gemma cup is having these branched, can you see that? Branched, stalked these branched, stalked structures, right? Okay. So let's discuss that. Diagrams are important. These topics itself are very important, okay? So bryophytes in this class have shape like liver, Markensia, or they are flat, Rixia. So we are calling them as liverworts. First point clear? First point clear? Beta, those who come to the class and mention, here it's 5:30, only 2 chapters are done. Matlab, what do you expect? How do you want to be a doctor? Like, what do you think that in 5 or 7 hours teachers should complete all the chapters? Yes, we can do that. After these detailed marathon, we will just pick up the most important topics. Then you tell me, how do you remember that much? Because throughout the year you are not able to remember or retain 32 chapters. Then you want that a teacher should, in 10 hours the teacher should finish 4 to 5 chapters, yeah, 7 to 8 chapters in that 10 hours, that too in depth, that too with all the important points. Matlab, you tell me, does it make sense? Does it make sense? The last marathons where we just say na complete class 12th biology or 11th biology, beta, we pick up the most important topics. Otherwise, do you think it's possible to finish, right, 20 plus chapters in just 10 hours, in just 20 hours or something? Matlab, why such type of comments are there? Like, why? You don't want to study, you leave it. Simple as that. It's as simple as that. But if you think that we will take the marathons and the quality content will not be there, I'm sorry, can't help it. At least I cannot help it. You want to study, most welcome. You don't want to leave it. Doesn't matter to me. Okay. So next part. So liverworts, they are the moist, shady habitat. This is what they need. So around the, the marshy ground, streams, they'll grow. Why? Because they don't have xylem and phloem. So because they do not have xylem and phloem, so they need to grow near the places which are, moist, so that they can just have that moisture. So structure here is again beta thalloid. So you know that dorsiventral flat green structure, they are having the midrib, dichotomously branched, closely oppressed to the substratum, that is attached to the substratum. So just say, let's suppose this is the structure, midrib is there. So trihizoid here are going to be unicellular and unbranched, a very important PYQ. Very important PYQ it is. Okay? Okay? So next is leafy members are also there. You know that they are leaf-like, not true leaves, right? And stem-like structures are there in the thallus. So next, you know that fragmentation is there. So very important is gemma. So gemma are nothing. You have seen that diagram. They are the green. They are the stalk. Just this is gemma cup. In it, you will see gemmae. In it, you will see gemmae, which are green, which are stalked structure. They will have the small stalk. They are multicellular, right? They are having the asexual buds, which are produced inside a cup like receptacles called gemma cups, right? Receptacles called gemma cups. So they will get detached from this parent plant and will grow into a new thallus. Is that clear? Right, next comes the sexual reproduction. Your gametophyte, they develops beta 2 types of multicellular jacketed organs they have. We know it very well. Again, the same story is getting repeated here. Just say you have antheridio4, the branch on which antheridium will be there. Antheridia rather will be there, right, in which antherozoids will form. Right, so sex organs are again antherozoids. So sex organs are again, right, the multicellular and jacketed. So this is the flash shaped. It is having the egg. So fusion of male and female gametes which occur inside the venter, they will develop a diploid zygote. Understood? So zygote will grow mitotically, will form sporophyte. As I told you already, having foot seta capsule in Markensia, and inside capsule, spore mother cells undergo meiosis, spores will form. That is it. So Markensia is dioecious, important. It is unisexual. But Rixia is what? It is monoecious. Now come to the mosses, right, where indirect development is there. So if life cycles are clear, then the plant kingdom is very easy. Okay, then literally Plant kingdom is very easy. Okay. Okay. Okay, guys, just 1 minute. So it's okay. The PPTs, it just merged, but it's okay. I'll directly start the mosses from NCERT then, right? There is some issue with the PPT, but it's okay. I'll continue it from this part only. I'll further provide you the handwritten part. So mosses may you know that here also gametophyte is the main stage, but here the spore will form what? The spore is going to form protonema here. So this is again having the evolutionary significance also. How come evolutionary significance? Because this protonema is just like algae. Like algae is having greenish structure, है ना? The greenish structure and filamentous structure. So it shows that evolutionary relationship that somewhere bryophytes, they arise from the algae-like ancestors. Okay, so predominant stage, the main stage here is what? It is basically having two stages. The first stage is the protonema, like your spore. Firstly will form protonema, that will develop directly from what? Spore. From spore, what will form protonema. So what exactly is this protonema, beta? It is creeping, it is green, it is branched and frequently filamentous stage. Okay, the second stage is what? It is the leafy stage, which will develop from secondary protonema, right, as a lateral bud. So basically, if I have to talk you about the life cycle, so spore firstly will form protonema. So that is a greenish structure, that is a filamentous structure. So here you will see some buds there that will form into the gametophyte, and this is the speciality of the mosses. Their gametophyte is leafy, right? Their gametophyte is leafy stage is there, which is important, okay? So leafy gametophyte can develop from secondary protonema as a lateral bud. So they are having upright cylinder axis, spirally arranged leaves are there, right? So here you know that in the mosses, I have repeated it multiple times, multicellular and branched rhizoids are there. And these leafy gametophytes will have the 6 organs, right? So fragmentation, even the budding, in the secondary protonema will be there. So again, Antheridia archegonia is the sex organ, right? After fertilization, zygote will again develop here. It will have foot seta and capsule, but it is better elaborated than the liverworts, obviously, right? We also see some cells in these sporophytes. We call them as elaters, right? Which are hygroscopic in nature, which will absorb moisture, which will help in the rupturing of the capsule so that spores can release. So example, Funaria polytrichum and sphagnum, right? Funaria polytrichum and sphagnum. Is that clear? Examples may, what do you have? Polytrichum, sphagnum, polytrichum and polytrichum, Funaria and sphagnum. So look at this part. In the case of mosses, right? So when underwater male sperm join to form the female sperm, right? So this is what they'll form the egg that will further grow. So this is a capsule-like structure which will develop here, right? And later on it will rupture and the spores will be spread there. So just 1 minute, guys. Just 1 minute. So this is it. So indirect development is present here, okay? Indirect development is present here. Just allow me 1 minute. So let's start. So this is all about the mosses, okay? So indirect development, branched and multicellular, right? Indirect development, branched and multicellular rhizoids are there. Understood? So next part. So here you can see in the bryophytes, these are the gemma cups and gemmae. And when it comes to the life cycle of the mosses, so look at the diagram here. Look at the diagram here. So you can start. This is the spore. Okay, so spore is forming this creeping green structure. Okay, so these are the protonema. They are having the buds. So you can see that then it is forming this leafy gametophyte. Okay, then it is forming. So if it is the male gametophyte, it will have antheridia. Female gametophyte will have archegonia. So sperm or egg, they will be there. You will have the zygote, fertilization. It will grow. Embryo will be there. Further, it will grow. So you can see foot, seta and capsule. And look at this part from the capsule, right? When spores will form, then it will rupture. They'll release. They'll regrow, right? They will regrow. So that's how it works, okay? Okay, so this is protonema. Secondary protonema is also there. And these are funaria, polytrichum, sphagnum. Okay. Chaliye, next is so next is ye dekho, mosses or bryopsida. But let me specify one more thing here, that is hornworts. Hornwort is not the part of our syllabus, but I would like to tell you a few points here that even in the hornworts, rhizoids are unbranched and unicellular. Sporophyte is not having the seta, it is just the foot and capsule. And here, right, because in the starting itself, I told you that sporophyte is partly or completely dependent on the gametophyte, है ना? So partly dependent is in the case of this hornworts and mosses. Even in the mosses also, beta, the sporophyte is partly dependent. Why? Because some cells will start performing the photosynthesis. They are having the chlorophyll. So dekho. So again, here also the sporophyte is not completely dependent on gametophyte. It is partially dependent. Why? Because some cells will become photosynthetic due to the development of chloronema in a capsule. Okay. Right? So same is the case for the mosses. So baki, see the diagrams here, buds on protonema, they are forming the leafy gametophyte. So leafy gametophyte is a peculiar feature here in the case of mosses. Right? Right, students? So ye dekho, predominant stage is having two stages. Number one is the protonema, number two is the leafy gametophyte. Okay, this is the first stage. Yes, this is the first stage, protonema. Filamentous, creeping, greeny, branched, juvenile, directly forms from spore. Have two types of branches, chloronemal branches which are photosynthetic, vertically growing, rhizoidal branches, multicellular, helping in attachment. Next is the leafy stage, second stage. It is the adult gametophytic stage, develops from protonema, right? So here this gametophyte, as it is directly from the NCERT, upright cylinder axis bearing spirally arranged leaves and attached to the soil by rhizoid. Okay, so presence of leaf-like structure in gametophyte is the unique character of mosses. So because in plant kingdom, any gametophyte do not have leaf-like structure. Got that? Do not have leaf-like structure. Got that? Chaliye, next is fragmentation and budding. Right? So secondary protonema, you know that it formed by any cell other than spore. So that's how it grows. So rest everything is just like this is basically from the NCERT only. So funaria, cord moss, which is monoecious, sphagnum is your peat moss or bog moss, polytrichum is your hairy cap moss. So sphagnum is very important. It is used for transshipment of the things as well. It can hold the water as well. And when they are not completely when their decomposition occur, which takes time, slowly when they decompose, they also form the peat. That is a good carbon source. That is a good fuel, right? So in the mosses, your sporophyte is partially dependent on the gametophyte. Right, so this is what you have to remember. So in economic importance, they prevent the soil erosion, right? They act like a mat on the soil, prevent the impact of falling rain. Plus, They make the substratum suitable for the growth of new plants. And sphagnum, which is the peat or the bog moss, and any other mosses can absorb water many times of their weight and used as a packing material for transshipment of living material. Right, so sphagnum also form the peat and manure, very important point. So peat is the source of carbon, it's a fuel, and manure is the peat moss. So mosses along with lichens, they are the one which belongs to the pioneer community for ecological succession. So next is the false mosses, matlab they are not true mosses. One example we have discussed is of Chondrichthyes, Irish moss, hai na? This is a red algae, but still you are calling it as moss. Selaginella, which is a pteridophyte. Again, this example is given in NCERT students. So it is the spike moss. It's again not a true moss. So let's suppose if in the paper they are giving you a question. Spike moss belongs to, right? Belongs to A, B, C, D. So A is algae, B is bryophytes, C is pteridophytes, D is gymnosperms. So sometimes students, they think it is a moss. Moss means bryophytes. But no, it's a false moss. Spike moss means I'm talking about what? I am talking about Selaginella, which is a pteridophyte. So answer will be C here, right? And the lichen cladonia is referred to as reindeer moss. So this type of question can also come in paper. Okay, so pteridophytes, the reptiles of plant kingdom having true root, stem and leaves. So I'll explain it with the help of a diagram and then we will discuss it further. So pteridophytes are the first successful land plants, but actually their gametophyte is also water dependent. That's why their distribution is restricted. Just let's suppose this is your pteridophyte, okay? You know that it is having true root, comma, stem and leaves. Matlab it is having true root, stem and leaves. Now, what will happen here? In this case, because main body is dominating body is sporophyte, yes or no? Main or dominating body is sporophyte here. So here you will see that some structure, just some leaf-like structure on it, right? Sporangium will be present. Matlab on some structure you will see sporangium. Sporangium is singular, sporangia is plural. So sporangium is what? Structure in which spores form. What is sporangium? Sporangium is a structure in which spores form. Yes or no? Now, they are present on a leaf-like structure. So the leaf-like structure which is having a sporangium, you call it as sporophyll. There's a fill word is with the leaf. It is sporophyll, means it is the one that can form the spores. So it can also perform the photosynthesis. So it is having the sporangium. So rest story is same, that sporangium will have the spore mother cell which will undergo meiosis, right? So sometimes on these sporophylls, you will see that sporangia, they form the groups, right? They form the groups. Sometimes this is also you see, right? So each group is what? Sorus. It's a group of sporangia. It's a group of sporangia. So on leaves, so many sori, plural is sori, sori are present. And sometimes beta, these sporophylls, they get reduced, they form cone-like structure, and then you are calling it as strobilus, right? So now what will happen? The story, rest of the story is same. Inside this spore mother cells are there beta. They will undergo meiosis. They will form spores. That spores will germinate into gametophyte. That spores will germinate into gametophyte. That story is same. Okay. So here gametophyte is heart-shaped, right? So this gametophyte is heart-shaped, which we also call as prothallus. Do not confuse it with protonema. It is the prothallus. And this is having the rhizoid, right, for the attachment to the substratum. So if you remember, in the pteridophytes, gametophyte is what? It is independent. It is free living, right? It is photosynthetic. Is that clear? Done. So this prothallus will have sex organs, है ना? So within that sex organs, what will happen? Gametes will form. That gametes, you know that male and female gamete fuse, zygote will be there. So this zygote basically developing on this inside that female part only, right? That zygote is developing. So we know that zygote will grow into what? It will grow into the sporophyte. So it is still on that, this gametophyte only. So it is like a precursor for the seed habit. So rest story is same here. Okay, so here you will see the concept of what? The concept of sporophylls. Okay, okay. Chali. So let's discuss this part. Let's discuss this part. So we are about to finish this chapter in next 1 hour. Matlab within this 1 hour, we have to finish it because angiosperms in detail are not given in your syllabus, right? So it is just the gymnosperms. Matlab angiosperms are not given in plant kingdom, although they are present in morphology, anatomy, sexual reproduction in flowering plants. But in this, under this chapter, you don't have it. Yes? Okay? Done? So let's discuss that. So pteridophytes, they are the vascular cryptogams, means they don't make the seeds, but they have xylem and phloem. So they are most adapted terrestrial plants, right? Right, they have vascular tissue. Now, they are not completely successful, beta. Why? Because they also need water for the fertilization. So pteridophytes, they need moist, cool, shady places and rocks for their growth. Okay, for their growth. So this life cycle can help you to remember this. So you will see some will have small leaves, जैसे microphylls. Micro माने small, leaf, fill, selaginella. Macrophylls are there in the fern. So this is again a PYQ. This is again from your NCERT. It is not at all extra, students. It is not at all extra, okay? And I told you about na, जैसे pteridophytes में ferns are very common in our households, right? And do Google it if you don't know what ferns are. When you will check these ferns, na, you'll be able to find, right, that they are having some brownish kind of dark colored spots. This thing, right? We just don't notice it, but actually we have seen it. So this is what? This is the sorus, as I told you. So it is nothing but it is the group of sporangia, right? The structures in which the spores will form. Am I clear? Am I clear? Right, so sporophyll, right, so you can see that is reduced, that is forming the cone like structure as well. So just keep it in your mind. And this diagram, this is of what? This is of the young sporophyte on the prothallus. So if you will observe it, prothallus is the female gametophyte, prothallus is the gametophyte. Prothallus is the gametophyte. So on it, because in female gametophyte, the egg is there, fertilization will occur, zygote will be there. From that zygote, sporophyte is growing. So these kind of things, they are showing that this is prothallus, that is the gametophyte, this is young sporophyte. So this is basically a precursor for the seed habit. So let's discuss this part. Okay. So plants, they have conspicuous, mane visible, sporophytic plant body, but inconspicuous, not visible, but independent gametophyte. So for its gametophyte, yes, you do use the word prothallus. So it is well differentiated, beta. It is having xylem as well as phloem. Xylem, you know that unidirectional mode, having tracheids, vessels, xylem parenchyma, xylem fiber. But here, because just this xylem and phloem, first time we will see it here. So vessels are absent in these pteridophytes. Even in gymnosperms, vessels are absent, except in Selaginella and Dryoteres. Important. So vessels are absent. Vessels is the feature of angiosperms, but yeah, we do have the exceptions in pteridophytes as well as in gymnosperms where you see the vessels. Baki companion cells and sieve tubes are again absent here. So leaves may I told you, if small leaves are there, microphylls, example is Selaginella. Large leaves may ferns are there. So here there is a trick. Please eat ptero. This is the first trick here. And under ptero, we will see padded stake. So these are the two tricks that we have to remember here, okay? So one is please eat ptero, and under this ptero, we will see padded stake. So we'll discuss it, right, about the tricks. So up to this part, all clear? Yes, everybody. Up to this part, all clear? So again, the story is same, that sporophyte is the dominant phase. Sporophyte will have sporophyll. On that sporophyll, you will have sporangia, in which spore mother cells are there, that will undergo meiosis, you will form spore, and that spore germinate into gametophyte or prothallus, which is itself either photosynthetic or saprophytic. Right now, because it is the gametophyte, it will make the gamete in these sex organs. Antheridia will make antherozoid, Archegonia will make egg, then zygote, then embryo. Back to the story. So these life cycles are actually very easy, right? It's a very easy way to revise this complete topic. So you can see sporophyte, it is having sporangia in which spore mother cells are there. So sporangia, where will you find it? On a leaf-like structure. So we are calling them as sporophylls or reproductive leaves. So the leaves which are not having sporangia, right, they are known as trophophylls or the vegetative leaves. So sporophylls having sporangia in which spores will form. Other leaves are the normal leaves, photosynthetic leaves, vegetative leaves, yeah, trophophylls. Now, sporangia, their group is called sorus. In pteridophytes, sporophylls itself are photosynthetic, which is a unique character. Already discussed that with you. So in some pteridophytes, these sporophylls, they can also form a cone-like structure, that is strobili or cones. That is strobili or cones. So Selaginella and equisetum is there. Selaginella and equisetum is there. So now beta, if let's suppose there is one kind of spore, matlab there are some pteridophytes which are having one kind of spore only. Homo, mane same kind of spore. That is homosporous. There are pteridophytes which are heterosporous as well. So hetero mane what? Hetero means, students, it means different. Yes or no? So what does it mean? Dekho, you know that what is the function of the spore? Spore ultimately germinate into gametophyte. Spore ultimately germinate into gametophyte. If all the spores are same, so this gametophyte is going to be monoecious. That is, it is going to be bisexual. That is, let's suppose if this is the gametophyte, it will have both male as well as female sex organ if it is homosporous. Now, when different kinds of spores are there, small size spore will be your microspore. That will form male, because in nature, male gametes are small. That will form male gametophyte. And there will be large spore as well. That will form female gametophyte. There will be male gametophyte and female gametophyte. This one will have male sex organ. This one will have female sex organ. Is that clear? And then you know that the gametes will form further. So heterospory, as per NCERT, this is again a repeated PYQ. The trick here is he, see, sa. What's the trick that we have here? It's he, see, sa. What's that? He, see, sa. It means see, Selaginella, salviniae. Salvia is different, it's salvinia. He means heterospory. So that's the trick you have to remember, right? These are the examples from the NCERT, very important they are. So keep revising it. So one kind of spore, homosporous, like your Lycopodium dryoteris. Heterospory you will see in he, see, sa. So these are two extra examples, but these two examples are given in NCERT. So Selaginella and Salvinia is there, and then comes the Zola and Marsilia. Okay, important part, right? So they will have separate male and female gametophyte, microspore, megaspore. So dioecious gametophyte will be there. Okay, so soil में this gametophyte, which is inconspicuous, which is but multicellular, right? It formed by the germination of these spore. You are calling it as prothallis, which is basically heart-shaped. It is having the rhizoid as well for the attachment to the substratum. So it can be, the gametophyte can be photosynthetic or saprophytic, right? So here you can see gametophyte in the soil. So it is free living, mostly photosynthetic, sometimes saprophytic. So it needs water. That's why the distribution of pteridophyte is restricted. So it requires cool, damp shady places to grow. So this is the specific requirement and the need for fertilization. So that's why these gametophyte is limited and restricted to narrow geographical regions, because here again the water is required. So do read it quick. Do read this line. Okay. Sure. Okay, so next part here is there is no relation between the sporophytic plant and its prothallus. So prothallus is basically thalloid structure having rhizoid non-vascular. So beta, it is very important to know that in the plant kingdom, we do not have any gametophyte, right? You can talk about the gametophyte, but you are not going to see that they are having the vascular tissue. So they are always dependent for the water requirement. Is that clear? This is a very important line, okay? This is a very important line, okay? So next, as I told you, sex organs are here also. They are beta multicellular. They are jacketed, okay, with sterile jacket. So archegonia will form egg and theridium will form antherozoids, but they are not always biflagellated. They can be biflagellated, multiflagellated, sometimes ciliated as well. Jaise ferns mein, they are multiciliated antherozoids, okay, but ultimately they are motile. This is what I'm trying to say. So they need the water for the transfer of this, right? So female gametophyte in heterosporous plants, they are retained on parent sporophyte for variable periods. So development of zygote into young embryos take place within female gametophyte. This is actually an event for the precursor of seed habit. Just see, let's suppose this is a female thallus, okay? This is having the female sex organ. The zygote will form in it. Of course, the zygote will form in it. It will grow. It will develop into a sporophyte. So sometimes what happens is this sporophyte is still attached to the gametophyte only. It is growing on it only. So this is basically a precursor, the beginner for the seed habit, right? So this is what they are saying, that development of zygote into young embryo take place within this female gametophyte, because ultimately it is the female gametophyte that is providing the nourishment, right? So that's how it grows, that's how it works, okay? Okay, so this is a habit for the precursor for the seed habit. And guys, here you can see the ferns, you can see these brownish structures are the group of sporangia. They are the sorus in which spore formation is there. So again, let's revise the life cycle once again. So this is the fern, rhizome, roots, young leaves are there. So look at this, they are having the sorus, group of sporangia. This is the immature sporangium. It is having that sporogenous tissue. So that sporogenous tissue having spore mother cell, right, which are diploid. So they are undergoing meiosis, right? They are forming the spore which are germinating. See this? So spore will form gametophyte. So you can see that this is the gametophyte having the prothallus. You are having a mature gametophyte. You are having the prothallus. So here you see the egg. You see the you see the archegonium and theridium. So you will see they will form the gamete, the zygote will form. Now this zygote is developing in this archegonium itself. It is forming young embryo, then it is growing into a young sporophyte. So still this gametophyte is here, and this is how the young sporophyte will grow, and finally it will become mature. So please have a look. Quick. Please have a look. Done? Chale, so let's move to the next part here. So these are the different examples. Here you will see, just say ferns, ornamental. This is the azola, that one fixes the nitrogen with anabina. This is equisetum, right? This is having the cone. So here in this case, you have different classes as well. You have different classes as well. So as I said, please eat taro. That's going to be the trick. Please eat taro. So P mane Psilopsida, Lycopsida. Please eat taro, right? So it is basically Psilopsida, Lycopsida, Sphenopsida, Pteropsida. Psilopsida, Lycopsida, Sphenopsida and Pteropsida, right? PLS, you can see that. So here, Psilopsida, example, Psilotum, it is the whisk fern. It is also a living fossil, important. Lycopsida, as the name is saying, Lycopodium, which is also the club moss. Again, it is a false moss. So please, sir, this sir, S, you have to remember here only, Selaginella also. It is against the spike moss. Then Sphenopsida, please eat, equisetum. This is what you have to remember. Horsetail it is. So Pteropsida mein, we have the trick, that is pad at stake. So first thing is Pteropsida. So P, silent, Pteris. D, Dryoteris. Here you also see Adiantum, which is also the walking fern, right? You also see Azola and Salvinia. So Salvinia individually is not mentioned here in the examples, but here it is, right? Overall it is there in the topic, just he sisa. So that is how you can remember that. So Azola is the aquatic fern, you know, that smallest radiophyte and biofertilizer. So it is he sisa, which will show heterospory, right? Other examples are also there. So baki Pteropsida. So examples, you really need to remember them, okay? Done? Sure? Sure? Okay. So the next part that we have is gymnosperms, right? Gymnosperms. So let me make few things clear now onwards. Jaise algae and bryophytes, they are homosporous. So algae, bryophytes, they are homosporous. Pteridophytes, some are homosporous, majorly they are homosporous, some are, right, heterosporous, right, some are heterosporous. And when it comes to gymno and angio, they are heterosporous only, right? That's what you have to remember. They will have two different types of spore and gametophyte. Now in the case of gymnosperms, we do not have, right, we do not have the flower, the fruit, but here the examples that you are going to read is Cycus, which is a living fossil. You will talk about Pinus, let's say Ephedra, Welwitschia, these are the examples. So Pinus, we will see that it is basically branched, right? Here actually you're going to talk about the cones. What are we going to discuss? We'll be talking about the cones here, right? Just Pinus may, you will see there will be male cone, there will be female cone, right? There will be male cone, female cone on same plant. So we will see that it is what? Monoecious, that is bisexual, right? This is monoecious, bisexual. Cycus is basically dioecious, unisexual. You will see it is also a living fossil, right? It is also a living fossil. So in the case of cycle, you will see even the female cone will not form. Female megasporophylls are together. So because they show the heterospory, so obviously in this case, you will see the sporophyll is having sporangium. So if this sporophyll, let's suppose if it is microsporangium, you know that in nature, right, in nature, these are the male gametes are small. So microsporophylls will have microsporangium within which microspores will form. That's what you have to remember. So microsporophyll on which microsporangium, inside which microspores will form. same for the female. Megasporophyll, megasporangium, megaspores will be there. Okay, so when these sporophylls are together, right, they form a cone-like structure. So male cone and female cone will be decided accordingly. So let's discuss the gymnosperms. I'll come to the life cycle, this flow chart of life cycle later, but let's discuss it first. Okay, so gymnos means naked beta and sperma means seed. Gymnos means naked, sperma means seed. So they are the most primitive seed plants in which ovules are not covered. They are directly present on, because ovule is what? Megasporangium beta. Ovule is the structure in which female spores will form. Ovule is megasporangium. So ovule is directly there on the megasporophyll. It is not covered. It is not covered within a flower, right? So ovules are not enclosed by ovary wall. They remain exposed both before as well as after fertilization. So in this case, the seed formation occurred for the first time in the Cyclostomata. They are now extinct. It is a fossil group. So overall, you know that gymnosperms, they are perennial, they are persistent, they are the woody plants. They can be in the form of shrubs or trees. So just say in the temperate regions, in the hilly areas, basically you see the gymnosperms, they are quite dominating. Like Pinus is the example. Why? Because here they were not eliminated by the angiosperms, because they have the adaptation, they can survive the xerophytic condition, they have the thick needles, they have the sunken stomata as well. So overall, that's what we have to discuss. So once, no doubt, these gymnosperms, they were dominating, but now they are restricted to the cold region, because here in these cold regions, due to their modifications, they were not replaced by the angiosperm. So that's something that you have to remember. So tallest gymnosperm, even it is not just the tallest gymnosperm, it is even the tallest tree. So it is sequoia. It is the giant redwood tree. It is the giant redwood tree. So smallest is zamia. So obviously the life cycle is what? Diplontic life cycle. So the main plant body is sporophytic. So it is also having two root, stem and leaves. Got that? Now plants made taproot system is there. Now you know what taproot is. This in the embryo, radicle is there. So from that radicle, the roots will form. There is a one primary root. There are secondary roots as well. Then they have the terrestrial roots and their branches also. So it's like that. So in the case of Pinus, right, even in our previous marathon, we have covered it. Mycorrhiza is there, right? That Pinus, the roots of this Pinus, they are in symbiotic association with the fungus hyphae. So this is the symbiotic association. In the case of Pinus, you see that. So fungal hyphae. So fungus is of genus Glomus. So they are in that close symbiotic association. Done. So Cycus, from C, you have to remember coralloid, right? So they are also having coralloid roots in which nitrogen fixing cyanobacteria are there, beta. So this is blue-green algae. These are the photosynthetic bacteria as well. So they are present in these root cells. Am I clear? Am I clear? So both the roots are important. Cycus is having coralloid. Pinus is having mycorrhiza. So stems, beta, when it is unbranched. So whenever you talk about the unbranched stem, right, where you have the leaves at the top, you call this arrangement as caudex, right? You call this arrangement as caudex. So if in paper they ask caudex gymnosperm, the gymnosperm, right, not angiosperm, gymnosperm in which stem is unbranched, the example is Cycus. Branched stems are there in Pinus and Cedrus. Both examples are from NCERT. There is nothing extra that I have added here, number one. Then leaves, you know that, right? Because we know that they are mostly restricted to the cold regions. Why? Leaves are simple as well, where leaf lamina is not cut, and compound as well, where you know leaflets are present. And when it is compound, you will see pinnately compound leaves. But we know that what type of arrangement, what type of leaves we will see. We will see thick needle-like leaves with thick cuticle sunken stomata. Is that clear? Thick cuticle sunken stomata. So Cycus, pinnate leaves, conifers, corn-bearing plants, they have needle-like leaves, because these are basically their adaptations in the xerophytic condition. Xerophytic condition mane dry conditions. Is that clear? So xerophytic conditions mane dry conditions, xeric condition where the water scarcity is there. So yes, needle-like, surface area is reduced, cuticle is thick, sunken stomata is there, so it can reduce the water loss. Got that? Right? So yes, xylem and phloem are present. So xylem mein vessels are not there except the gymnosperms of order Neetails. So order Neetails, they are having the example jaise Neetum, Ephedra, Welwitschia. These are the gymnosperms where true vessels are present. These are the gymnosperms where true vessels are present. So phloem, they lack sieve tubes and companion cells. Rather, you will see sieve cells are there and albuminous cells are there. Right? And albuminous cells are there. Okay, so reproductive structures are there in the form of cones here. No flowers are present, but seeds are there. They are also not covered. So obviously they will show heterospory. You will have microspore that will form male gametophyte, megaspore that will form female gametophyte. So this part, microspore is produced within microsporangia. So what is microsporangia? Even in angiosperms, you use the same term. You are talking about the pollen sacs there. So in these microsporangia, your microspores are formed, beta, and that microspores develop into male gametophyte. So which one is the male gametophyte here? Even in angiosperms also, it is pollen grains. What are they? They are pollen grains. Is that clear? What are they? They are pollen grains. Okay. Right. So overall, microsporophyll having microsporangia in which microspores form. So overall, this is the male cone. So this is NCERT language that microspores they produced within microsporangia that were on microsporophyll, and these microsporophylls are spirally arranged to form a male cone or strobili, right? So these microsporophylls, they are spirally arranged, right? How are they spirally arranged? They are spirally arranged to form the male cones or strobili. So microsporophylls are having microsporangia, spongium, in which the microspore mother cell will form the microspores by undergoing meiosis. Okay. Okay. Same for the female gametophyte, megasporophylls having megasporangium. So it is the ovule which is the megasporangium. It is the ovule which is the megasporangium. Okay. Done. Okay, beta. So here you can see these are the coralloid roots. But before that, let me show you this is the cycus. In many hotels, restaurants, places also, like in our homes also, sometimes we grow them. So these are the cordex. Then there is the pinus. Pinus is monoecious. That's a PYQ again. So you can see in the cycus, male will still form the cone-like structure. But females may. This is important to remember, beta. Females may cone will not be there. In the female, megasporophylls are together. First of all, male and females are different, right? Or like different plant, right? And it is also the living fossil. So megasporophylls, they do not form cone here. You are not going to see the female cone here. Am I clear? You're not going to see the female cone here. Understood? Understood? So see the pinus, the smaller cones are male cone, the larger one are female cone, and you will see that the pinus may they are having the pollen grains. These are the winged pollen grains that will help in wind pollination. Wind pollination is there in their case. You'll see, and this is known as anemophily. Yes, it is known as anemophily. Am I clear? So winged pollen grains are present, right? They are not present in all the gymnosperms, of course. So let's discuss it one by one. Okay, so that story is same, beta, ki megasporophyll is having ovule. So this story is same, jaise microsporophyll, right? Microsporophyll having, just making this diagram to in a simplified way so that you can understand, having microspore mother cell, ya pollen mother cell that will undergo meiosis, and you will have microspores that will grow into pollen grain. And pollen grain here is what? Actually, it is the pollen grain which is the male gametophyte. It is the pollen grain which is the male gametophyte. Is that clear? So megasporophyll having ovule. But here, let's look at the structure of the ovule. Even in morphology, we have discussed it. Just say, let's suppose this is the ovule. Beta, it is having a covering. But, right, mostly your gymnosperms, they are unitegmic. Mostly, they are unitegmic. Now in this ovule, this part here, this is your nucellus. It is nutritive. Now this portion is little different from your angiosperms, because here you will see that archegonia are present in this ovule. That's what we are going to see. So they have nucellus. Mostly they are unitegmic. I'm not saying all of them. So nucellus is indeed the nutritive tissue. So this is the doubt here. So you can see ovules are having nucellus, which is protected by envelopes. Inside ovule, no doubt there will be single megaspore mother cell. Indeed, there will be megaspore mother cell. I am talking about the developed embryo sac. I'll come to this. Right. So there will be the megaspore mother cell, and we know that in nature female gametes are less. So when it comes to the megaspore mother cell, right, meiosis 1 and 2 will result in the formation of 4 megaspores, but out of these 4 megaspores, 3 will degenerate. Only one is functional. That will form female gametophyte, which is present within this microsporangia, and that female gametophyte is having 1 or more, 2 or more archegonia. So this is different from your angiosperms. In angiosperms, this is not that we discuss कि embryo sac is having archegonia, but here it is having. So just say here I was showing you this nucellus. So ultimately in this nucellus, there will be, right, a cell which will grow, just a megaspore mother cell, right? It will undergo, I would say, meiosis, right? After, just say, let's suppose after 4, because 4 megaspores will form which are haploid, only 1 will grow, 3 will degenerate. So this one is having, it will develop into the embryo sac, right? It will develop into the embryo sac, and this one will have the archegonia. Am I clear? This one will have the archegonia. Is that clear? So 2 or more archegonia are there. So a part of female gametophyte stores food material, later form endosperm. So in gymnosperms, very important point it is, beta. In gymnosperms, first of all, you don't talk about, there is no double fertilization, right? We do not discuss about the double fertilization in their case, number 1. Right, number 1. So number 2, firstly number 1. And in gymnosperms, this endosperm is haploid. Endosperm is there, but it is haploid, and it develops from female gametophyte. So if it is female gametophyte itself is haploid. So obviously the endosperm will also be haploid. So male and female gametophyte, they are retained on sporophyte within the sporangium. So in gymnosperm, endosperm is haploid. Keep it in your mind. Wind pollination occur here, right, beta? So pollen grains with the help of wind directly reach to the opening of that ovule where, just let's suppose this is ovule, na? So this is the micropylar end. So even in morphology, even in sexual reproduction, I have explained it already. So these are the pollen grains that will form pollen tube, that will carry 2 male gamete. 1 is functional, 1 will be destroyed. So here there is no double fertilization. Double fertilization, right? Double fertilization is there in. Beta, Komal, I have explained it. Then why are you just confused here? I have already explained it, that what exactly is what exactly will be there in this female gametophyte case. So after fertilization, zygote, then embryo, and you know that this ovule will change into seed. So it is the ovule that forms, that grows into seed. Is that clear? So seed is having endosperm, but endosperm is N. Why? Because it develops from the female gametophyte. Right? So in the gymnosperms, beta, polyembryony is very common. It's a usual feature of gymnosperms. Or seed may, which one will show the parent sporophytic or parent diploid generation. The seed coat, the outer covering. The integuments form the seed coat. Even if nucellus is present, that perisperm, that shows the parent sporophytic generation. And which one in a seed shows gametophytic generation, that is haploid generation? It is endosperm. And which one will show future diploid generation, future sporophytic generation? That is embryo. That is embryo. Got that? Chaliye, next is, you know that cycas, living fossil, dioecious, winged to as a relic of past. They have primitive features like ferns, jaise they have coiled leaves. The sperms are motile here. In cycas, the sperms are multiciliated. Female cone absent. And in cycas, this is what you see. You see the two cotyledons. Two cotyledons. Got that? Done? Then, ginkgo biloba is also the living fossil having motile male gametes. So, winged pollen grains are not there in the cycas. So, Cedrus, which is also known as deodar. Texas is also the example of gymnosperm, from where the Taxol, which is anti-cancer drug, it is also obtained from the bark. So, Sequoia, the largest gymnosperm, the largest tree, also known as father of forest, here red giant wood. Pinus, you know that monoecious, winged pollen grains. So, when there is a dispersal of this pollen grains, it is called the shower of sulfur, yellowish color. It is yellow dust, it is. Okay. So, then is beta angiosperms. So, angiosperms in detail is explained in individual chapters. But yeah, you have to remember here that the smallest is Wolffia and the tallest is Eucalyptus, which can grow over 100 meters. Two classes are there, dicots and monocots. So, monocots are advanced than the dicots. Okay. Baki here in angiosperms, you will see double fertilization is present. So, because there is double fertilization in their case, right, the endosperm here is going to be triploid. Right? That's what you have to remember. Baki beta, this stamen, okay, in angiosperm, stamen is equivalent to microsporophyll, you have pollen sacs as microsporangia, you have pollen grain as male gametophyte. Then stigma, style and ovary, this is equivalent to megasporophyll, ovule is equivalent to megasporangium, and beta, you will see embryo sac, which is the female gametophyte. Okay, which is the female gametophyte. So, this is all about the plant kingdom. So, let's have a break and then let's start the biological classification. Ready? Do you need break or not? Right? Do you need it? Sure. So then let's start the so many questions are there. Acha, beta, even cell, then living word, and this is the biological classification. So then after this, let's start the biological classification. Ready? Chalo, let's have a break first. So let's restart at 7 p.m. Okay, see you then. Hi people, let's start the next chapter, that is biological classification, from where 2 to 3 questions, or I would say 2 questions are expected, okay? Rest, in the case of NEET examination, we do not have any uniform pattern, okay? We do not have any uniform pattern, so it can change, right? But yes, you guys can expect 2 questions from this particular chapter, okay? So let's get started. Sorry. So biological classification. Basically, it's important to classify the organisms. Why is it so? Because that will help us to study them easily. Just I'll give you an example. This arrangement is possible. Like, let's suppose you go to a library, right? If in that library, if there is a clutter, if books are not properly racked, if books are not properly placed, imagine you will go to the library. What will you do there? You will find, first of all, you have to invest your time and energy to find a particular book. But now, on the other hand, when we go to a library, we know there are different sections. Yes or no? Yes? Yes or no? Right? There are different sections there. Just say there will be the physics section, chemistry, biology section. You can go to the physics section. There also, they will mark that this section is having this book, this section is having that book. So we all are well aware of it. You know it very well, right? You know it very well, right? So basically, this class, like when things are systematically arranged, it makes our it save our time, it makes our learning easy as well. So same is the case for the animals. You look around you, we have the variety, we have the diversity, we have so many things around us. So if they will be properly classified, if they will be systematically arranged, then we will learn, then it will simplify our study. Like for an example, we read about Phylum Arthropoda. We know some of the general characters. Now, because you know some of the general characters of the Phylum Arthropoda, right? Because you know about the general characters of the Phylum Arthropoda, then obviously, for sure, right now, if somebody will say this is from Phylum Arthropoda, at least you will have some knowledge in your mind. You don't have to study single organism. You will have some general idea of that. So that is why this classification is very important, which we will discuss in the living word. So overall, in this chapter, we will start with the Aristotle, who first time tried to classify the organisms. He didn't use any kingdom term or something, right? But he just classified them as plants and animals. Then we had the 2 kingdom classification. We will also talk about the 3 kingdom classification, 4 kingdom classification, 5 kingdom classification, and 6 kingdom classification. And in the starting of the lecture, I told you that it is the 5 kingdom classification that we study in detail, right? So I hope you guys are well aware of it. So let's start this part. So here again, you will get the handwritten notes so that we can save some time and we can get best out of our lectures. So when you talk about the biological classification, so you know that it is the scientific procedure, right? It's a scientific way to classify the organisms into different groups. How? On the basis of their similarities and dissimilarities. We see their similarities, we see their dissimilarity, we even study their taxonomic relationship. So there are different ways by which we can classify them. We can put them in a hierarchy of categories. Is that clear? Already started with the basics. So first, a scientific attempt to classify the organism. Who performed it? Aristotle, father of biology, father of zoology. So he used the simple morphological characters, and initially the classification was on the basis of needs. Keep it in your mind. As per our needs, whatever you need, as per that, you put the things, okay? Okay, so what he did, he classified the plants on the basis of simple morphological characters into herbs, into shrubs, into trees, right? He placed the plants on the simple morphological characters, right, into the herbs, shrubs, and trees. In the case of animals, based on the presence or the absence of red blood, he categorized that. Just say we have Anaima, we have Enaima, we have Anaima, Enaima. So A mane without, this is without red blood cells. Enaima with the red blood cell. So he classified the plants on the basis of morphological characters, herb, shrubs, and tree. In the case of animals, it was Anaima and Enaima. So that's what you have to remember. Next to it is 2 kingdom classification given by Linnaeus, who's also the father of taxonomy, right? He also gave us the binomial nomenclature. I hope you remember. So he classified organisms into 2 kingdoms. He used this term kingdom here. Is that clear? Is that clear? Is that clear? So two kingdoms are having Plantae, they are having Animalia. Plantae is there, Animalia is there. So the major criteria of classification was what? Presence or absence of cell wall. So yes, this is a very important point. On the basis of presence or absence of the cell wall, he divided the organisms into two particular kingdoms. So if cell wall is present, then it is under Plantae. If cell wall is absent, it is under Animalia, right? It is under Animalia. That is what he did. Is that clear? So other than the cell wall, some other criterias were also there. Just here you can see, right? Just cell wall present, then plant; cell wall absent, then animal. Locomotion in plants, they do not they cannot change their location, right? So locomotion is absent in plants, but this locomotion is present in Animalia. Mode of nutrition, plants, they do not, right? Plants, they do not eat, they make their own food, but animals, they eat, they are dependent on others. Then response to external stimuli. When it comes to response to external stimuli, both plants and animals, all the living organisms do, they react to their surrounding. This is a very important property. This is the defining property of a living being, that yes, they respond to their surrounding. But in the case of plant, this response is going to be slow. In the case of animals, it is fast. Then comes the contractile system. Do not confuse it with the contractile vacuole. Contractile system, जैसे cells are having the proteins which help in contraction and all. So overall, that contractile system that will help in this process is absent in the plants, but it is present in the animals. But here they have clearly specified that diatoms, chlamydomonas, euglenoids, they were in the Plantae category, but still they have what? The contractile system. But main criteria was the cell wall. So he did what? He kept the, right, he kept the, even the bacteria, because bacteria are having cell wall, he kept it in Plantae. Even blue-green, right, blue-green algae, he kept that also in Plantae, right? Fungi, he kept that in Plantae, mosses, ferns, gymnosperms, and angiosperms. So even the bacteria, even the photosynthetic bacteria, blue-green algae, even the fungi, mosses, they all were kept. They all were kept together under the kingdom Plantae. And in Animalia, he kept protozoa, vertebrates, and invertebrates. Am I clear? Am I clear? So two system classification, two kingdom. So it was, there were drawbacks as well, right? It was not accepted. Many problems are there. Just first thing, if I have to say that there are certain plants, certain organisms which show the characters of both. And fungi being, right? Fungi is the problem. Fungi is heterotrophic. How can we keep that in the plants? So let's have a look. So this kingdom, this two kingdom classification, it doesn't classify or differentiate unicellular and multicellular organism. Number one, eukaryotes and prokaryotes. Number two, photosynthetic green algae and non-photosynthetic fungi, right? They were not able to differentiate them. Unicellular, multicellular, they were together. Prokaryotes, eukaryotes, they were together. Even the photosynthetic, non-photosynthetic, they were together. These are the loopholes. But moreover, they are to, chalo theek hai, plants and animals, so we can still classify. What about those which are showing the characters of both, right? Which do not fall into either category. How to differentiate that? So in that case, just for an example, euglena. You know that euglena is a connecting link between plants and animal. It is having the characters of both. And even these chlamydomonas, they slime mold, they have the characters of both. So how to classify them? These were the question marks. Fungi, which are what? Which are usually included in plants, guys. They have characters not common to plants, like their cell wall, right? In the case of fungi, we know it very well. Their cell wall is having chitin, right? They have chitin, which is also known as the fungal cellulose, plus they are heterotrophic. They are not at all autotrophic. So how can we place them together? Is that clear? Is that clear? So instead of two modes, that is ingestion in animals and primarily photosynthesis in plants, synthesis in plant, they also came up with the other mode, that is the absorption. Is that clear? So drawbacks are very important. So he was failed to classify the organism. He was failed to distinguish unicellular, multicellular, prokaryote, eukaryote, photosynthetic, non-photosynthetic, fungi with different cell wall, heterotrophic, kept in the plants, right? Euglena, chlamydomonas, slime mold, which are having other characters. How can we put them together? That is also the point. That is also the case. Is that clear? Is that clear? So not just the ingestion and photosynthesis, right? Other than that, the absorption is also the mode of the this nutrition mode. Then comes the three kingdom classification. I know this is not given in the NCERT, but how can we directly jump from 2 to 6, right? We should know what were the things which are in between. So it was given by Ernst Haeckel. Who gave it? Ernst Haeckel. So he was like, "Okay, I'll do one thing. I'll separate the unicellular organism." So he introduced one more kingdom, that is kingdom Protista. Right, that kingdom Protista was created, in which unicellular organism, along with the one where the tissue differentiation was not there, they were placed in the Protista. But again, the problem is same. What about prokaryotes? What about eukaryotes? Then four kingdom classification given by Copland. So he introduced Monera. Actually, initially it was not Monera, it was Mycota, right? Then later on it was named as Monera. So what he did in the case of, like, firstly, Monera mein they kept prokaryotes, and in Protista they kept what? They kept eukaryotes. This is what they did. Are you getting my point? This is what they did. Are you getting my point? So it include all the prokaryotic organisms, just say Eubacteria, Archaebacteria, this Monera, right? And Protista will have what? Unicellular eukaryotes. Okay, okay. But again, the problem was fungi. What to do with fungi? Now, the problem was solved by R.H. Whitaker, 1969. Yes, we have to remember the year as well. So R.H. Whitaker, 1969. Who was he? He was an American taxonomist, right? So he studied the fungi. He kept them in the different kingdom, right? Because fungi was the problem, right? Done? Done? So American taxonomist, 1969. So he gave the 5 kingdoms. So what are the main criteria? Yes, we need to remember this as well. This is also important, right? This is also important. Is that clear? Is that clear? So what is the point here? So he selected the cell structure. Beta dekho, if listen to me like a story, ki acha, first this happened, then this happened, then this happened, then this happened. It is like that. This chapter is like that. There is nothing conceptual here. It is like the story. Okay, firstly we figured out this, then we figured out that. Okay, so let's maintain the pace, because I really want to finish it with the proper content, because today I don't think we will be able to start the cell. But yeah, because 12 hours is the limit, right? And I would say 1 hour. Yes, so 12 hours is the limit, and we have break time also, right? Otherwise, so we would have finished it. We had the break also. So let's get the best content first of all. Second thing is that you can even reduce the YouTube speed, right? I will explain it. Wherever I'll have to go slow, I will go slow. But I literally want to finish it today so that we can finish diversity. Then we can target structural organization, cell, and the physiology separately, because that is also very important. So you just have a look here. The main criteria was classification was used by Whitaker. Cell character, cell structure he studied. Very important point, students. So either prokaryote or eukaryote. Then thallus organization, whether it is unicellular or multicellular, that was also considered. Are you getting my point? Then mode of nutrition, it was the most important criteria. So most important is the mode of nutrition, again a PYQ. So autotroph or heterotroph. Then reproduction, sexual, asexual, and the very important is he also considered the phylogenetic relationship, that is evolutionary relationship. So these are the main things. On the basis of that he separated these kingdoms. But yeah, mode of nutrition, it was given the main this importance. Now the next part is what? Next part is this, right? This table. This table is equally important. One by one we will discuss it. Just you know that it is only kingdom Monera which is having the prokaryotes. Rest all are eukaryotic kingdoms, right? Prokaryotes mein cell wall, bacteria cell wall, it is basically non-cellulosic plus amino acid. You will talk about the peptidoglycans here. One by one we will discuss it, okay? We will talk about the peptidoglycans here. Protista mein cell wall is not present in all. We talk about a flexible proteinaceal layer, pellicle here. Fungi is having the chitin and polysaccharide. Plants are having cellulose, hemicellulose, pectin. And in animals, we do not have cell wall. And obviously prokaryotes, the primitive cell, which doesn't have the, where the genetic material is directly there in the cytoplasm, right? Genetic material is directly there in the cytoplasm, right beta? So it is the nucleoid there, rest all are having this. Body organization may, you know that this is unicellular. Here also it is unicellular, unicellular prokaryotes, unicellular eukaryotes. Fungi is basically multicellular or loose tissue. Exceptions are there like yeast. Plantae is also having tissue or organ, but sometimes yes, we do study chlorella here, so there also we can consider. And then Animalia, it is having tissue, organ and organ system, right? Mode of nutrition mein, Monra mein, autotrophic, right? They are preparing the food by chemicals also, by light also. They are saprophytic also, that is they show the absorption. Parasitic also, same for the protista, autotrophic as well as heterotrophic. Fungi only heterotrophic, they are either parasitic or saprotrophic, that is absorption. So plants only autotrophic, not, you know that there are some plants which are even the heterotrophs. Okay, so this is in general that they have given students, and we really need to remember this. So see what they did in kingdom Monera, as per this 5 kingdom classification, all the microorganism with prokaryotic cells, they were placed there. Just your Eubacteria, Archaebacteria, Mycoplasma, Actinomycetes, right, etc, etc. Okay, Protista may generally single-celled eukaryotes were placed like Euglena, amoeba, etc, right? Fungi, Plantae is having eukaryotes, multicellular, but they are autotrophs with cellulosic cell wall. Fungi is having eukaryotes, multicellular mostly, right? Heterotrophs with absorptive mode. And then Animalia, multicellular heterotrophs with wall-less eukaryotes. Is that clear? Right. So here you can see this table, this chart as well. So you can see on the basis of complexity of cell, we have prokaryotes, we have eukaryotes, kingdom Monera. Then complexity of organism, simple unicellular or complex. You know that. In eukaryotes, you have kingdom Protista there. Then when it comes to fungi, Plantae and Animalia, no doubt we write it like this: fungi, then Plantae, then Animalia. But beta, it is not sure that it is also the case that they might all of them, they evolved at the same time, right? We cannot say that fungi came first, then Plantae, then Animalia. That is not the thing here. So mode of nutrition you can see, and ecological role may autotrophs, that is plants are producers, fungi are decomposer, and animals are what? They are the consumers. Is that clear? They are the consumers. Now next part is features. What are the features of this 5 kingdom classification system? Very important, it separates prokaryotes and eukaryotes. Okay, unicellular and multicellular organism. Kingdom Protista, a very important point, because of this kingdom Protista, because in kingdom Protista unicellular eukaryotes are there, so it brought together Chlamydomonas and Chlorella with Paramecium and amoeba, because they were placed separately. Just you know that Chlamydomonas, right? Chlamydomonas and Chlorella, they were having the cell wall, hai na? So due to which in 2 kingdom classification, they were placed under Plantae, right? And Paramecium and amoeba, they do not have cell wall. So as per 2 kingdom classification, they were placed in Animalia. But now in kingdom Protista, where the criteria is unicellular eukaryote, they were brought together. Any doubts? They all were brought together. Any doubts? Any doubts? Sure? Any doubts? Okay, so the next part is, so fungi was placed in a separate kingdom because it is different from plants in cell wall as well as in nutrition. So again here we have the drawbacks, right? We have the drawbacks. No mention of acellular organisms like virus, viroids and lichens. So virus, viroids and lichens, they were not at all mentioned. Is that clear? They were not kept in any of the kingdom. Then comes the 6th kingdom classification, which introduced the domain. So Carl was, this is important, proposed this. He gave the 3 domains. So domain is even a bigger category than the kingdom. Just say we have the kingdom, then phylum, then class. Now, so domain is first domain, then kingdom. So he introduced what? He gave us 3 domains. right? Mane very large group of the similar object. So 3 domains, they basically in this kingdom they divided the kingdom, in this classification they divided the kingdom Monera, because here they observed that there are 2 types of bacteria. One is the ancient one is the primitive one, that is the Archaebacteria. That is the Archaebacteria, right? And another one is what? Another one is the Eubacteria, right? U means true. So this Archaebacteria, the ancient bacteria which can survive in the extreme condition, and let me tell you beta, it shows more resemblance with eukaryotes. Keep it in your mind, okay? Okay, so these 3 domains, they divided actually the kingdom Monera into 2 domains and remaining into 3rd domain. So basically they were having 3 domains. One is Archaea, second is bacteria, third is eukarya. So on the basis of even the 16S ribosomal RNA sequence, they divided it. So Archaea mein they placed a new kingdom under this domain, that is your kingdom Archaebacteria. That is what? That is your kingdom Archaebacteria. And in bacteria, they place the kingdom Eubacteria. So rest all, जैसे your Protista, right? Fungi, Plantae, Animalia, they all were kept in eukarya. Is that clear? They all were kept in eukarya. Is that clear? Is that clear? So as I said, so they divided it. So domain is a super kingdom. They divided it on the basis of 16S ribosomal RNA sequence. So Archaea is there, bacteria is there, eukarya is there. So no doubt Archaea and bacteria also show the similarity, but Archaea shows more similarity with eukarya. It's a previous year question. Got that? It's a previous year question, okay? So what do we see in kingdom Monera? So do remember it, do read about it. So Archaea and eukaryotes, eukarya, they show more similarities. Now beta, we will enter in the kingdom Monera where we have the prokaryotic cell, right? So let me give you the overview here. You know that in the case of a prokaryotic cell, right? Just allow me 1 minute. So overall, what will you see in a prokaryotic cell? So I will elaborate it, right beta, because yeah, I will elaborate it a bit. Why is it so? In the NCERT, if I will just translate it from NCERT, it will hardly take me 2 hours to finish this. But I want you to understand the terms. So accordingly, we will discuss it, okay? So accordingly, we will discuss it. So overall in a bacteria, like in kingdom Monera, because we are talking about 5 kingdom classification. Archaebacteria is also there, Eubacteria is also there. But here we will discuss them as separate groups. But overall in 6 kingdom, they were kept in the 2 different domains. So in general, if I talk about a bacteria, I would say a true bacteria. What do you see? Outermost is glycocalyx, students, which is also known as cell coat. Here you will see, right, that either the cell coat, this outermost covering, can be capsule, either it can be slime layer. So it is capsule, mane, it is what? It is thick, it is tough, polysaccharide coat, right? And you know, in many bacteria, this polysaccharide coat is responsible for causing the disease. It also protects the bacteria from antibiotic, antiviral effect. It also protects it from the desiccation. It doesn't let it to dry. So slime layer is the loose sheath, right? So it makes sure that water should retain within the cell and it should not lose its nutrients. Next to it is the cell wall here. So when you talk about the eubacteria, your true bacteria, so cell wall is, students, actually made up of peptidoglycans here, right? It is made up of what? It is made up of peptidoglycans, which we also call as murines, which we also call as mucopeptides. Are you getting my point? So we have the peptidoglycans here, which are also the murines and mucopeptides. Plus here in this cell wall, you will also see some amino sugars are also cross-linked, right? The amino sugars are also cross-linked in these compounds. This is what we see. So when you look at the cell wall here, जैसे fungi is having chitin, plants is having cellulose. Here you will see that there is NAM, NAG, an important question. So NAM stands for N-acetyl muramic acid. This is N-acetyl glucosamine. That's what you have here. Are you getting my point? This is what the cell wall is made up of. In Archaebacteria, it is different. That's why I'm mentioning it here. Plasma membrane or cell membrane is same, just like your, it is also selectively permeable. It also interacts with the outside world. Baki, here we know that because you are talking about the prokaryotic cell, you won't see well-defined nucleus. There is no well-defined nucleus. The genetic material, which is basically what? It is single double-stranded DNA. Obviously, this DNA is circular. It is devoid of histone proteins. That is, it doesn't have histone proteins, right? Which we study in the case of eukaryotic cell. So it doesn't have the histone proteins. This DNA is actually, students, it is a naked DNA. Why? But it doesn't have any envelope around it. It doesn't have histone proteins around it, right? So this is nucleoid devoid of histone proteins, right, beta? So other than that, extra chromosomal DNA plasmid is there. Ribosome, which is the universal organelle, right? Which is the universal organelle, which is present in both, that's help in the protein synthesis. Baki polysomes and all, they are present. So it is in general the structure, right? The structure. And here, you know, mesosomes are also present here, right, beta? Mesosomes are also present here. So these are the finger-like extensions of cell membrane in cytoplasm. And these extensions, beta, they can be in the form of vesicles, they can be in the form of tube-like structures, they can be in the form of lamella. Okay, so these mesosomes, we consider their work equal to the, you know, mitochondria. Why? Because they have the respiratory enzymes. They help in the secretion process. So they are finger-like in folding, so they'll increase the surface area. So enzyme content and the area will also increase. Cell wall formation, it is helping. So that's how it helps. So if bacteria is motile, it is going to have flagella, which is very simple in structure, which is basically made up of flagellin protein. So these are some general points regarding the prokaryotic cell, right? These are some general points regarding the prokaryotic cell. So now when you move ahead, so in Monera, you are considering such type of cells. So we know that they are unicellular. So definitely they are microscopic, they are single-celled, right? And bacteria are the sole members here. Bacteria are the only members of this particular kingdom. So we know that bacteria can occur almost everywhere. So if I'm saying it is occurring almost everywhere, we can use the term ubiquitous. So ubiquitous means which is present everywhere. Just we also use the word cosmopolitan. Na? Cosmopolitan means which is distributed everywhere. So same way you have the ubiquitous as well. So they live in the extreme habitat, just a hot spring, desert, snow deep oceans. They can also survive there. Some can live within the host body. Some are basically the parasite. So some bacteria, beta, they are autotrophs as well, but vast majority is of heterotrophs. A very important point. Is that clear? So some of the bacteria, they are autotrophs, but vast majority is of heterotrophs. So if you relate it with the evolution, you will see that basically what came first, your autotrophs. No, initially the bacteria, the cells that came first, they were heterotrophs. Okay. Done. So this is what they have. So reproduction is asexual type. There is no true sexual reproduction. So they have rigid cell wall, beta. So it consists of peptidoglycans. Told you this. N-acetyl muramic acid and N-acetyl glucosamine, lipids, proteins, amino acids are also present in the cell wall. So this NAM-NAG thing is very important. It is very important. Is that clear? This is very important. So these are the peptidoglycans. Why am I saying it is very important? Because we will compare it with the Archaebacteria as well. So genetic material is naked, circular, not enclosed by nuclear membrane. So you use the term nucleoid, genophore, incipient nucleus, and we can also call it as prochromosome. Got this? So ribosomes are present. They are 70S type, right? So simple chromatophore. Chroma, you know beta. What is the meaning of chroma? Chroma means color. So the bacteria which shows the photosynthesis, like your blue-green algae, which we are also calling as cyanobacteria, right? So in their case, they have these structures, chromatophores. So these chromatophores, what do they have? They have the pigments in it. That pigments will help in photosynthesis. Am I clear? That pigments actually help in photosynthesis. Yeah, do you see the covering here? But true membrane bound structures are not present in the prokaryotic cell. That's why beta prokaryotic cells are just making the one envelope system. Means bacteria are having one envelope system. Means in case of bacteria, only outer coverings are present, but they do not have the membrane bound structures in it, right? No membrane bound organelle is present in a prokaryotic cell. The organelle which is present is ribosome. It is again a non-membrane bound organelle. Is that clear? Right? So gas vacuoles may be present. If present, they provide the buoyancy. Right? So they are non-motile as well, motile as well. So if they are motile, they are having a very simple flagella, right, which is made up of flagellin protein. So this is important. Why it is important? Because in eukaryotes, right, in eukaryotic cell, eukaryotic flagella when you discuss, then beta you talk about the microtubules if you remember, right? You talk about the microtubules if you remember, 9+2 arrangement of microtubules, which is made up of tubulin protein, है ना? Do you remember this? I always emphasize this point. But when you are talking about a prokaryote, you will see that the protein is flagellin there. Protein is flagellin there. Is that clear? The protein is flagellin there. But in eukaryotes, it is the microtubules which are itself made up of tubulin protein. So 9+2 arrangement we see. Okay? 9+2 arrangement that we see here. So they are not enclosed by any member and they grow at the tip. Right, so any membrane and they grow at the tip. So reproduction is asexual, mainly by binary fission. So these are some general points regarding this. Now, next, from where, yes, they do have asked a question about the shapes of bacteria. Very simple topic it is. So students, if it is spherical, you know that it is going to be coccus, right? It is going to be coccus. So spherical shaped bacteria is your coccus, plural is cocci. If it is rod-like, that is, right, it is bacilli. Singular is bacillus. It is rod-like, right? Very common shape. Yeah, cylindrical it is, right? So in such bacteria, you also see an endospore. So bacillus, it's the most common shape. Let's say Bacillus anthracis is the example. Then spirilla, right? The singular is spirillum. Treponema pallidum. You remember Treponema pallidum? During STIs, we discuss it, sexually transmitted infections, right? It causes the syphilis, if you remember. That is also an example of spirilla. Vibrio mane comma shaped. So vivrium is the singular, vibrio is plural. So here the shape is comma shaped. So this one is comma shaped, this one is spiral shaped. So that's what we have seen. I hope it is clear, right? I hope it is clear. Is that clear? So shapes of bacteria, very easy question it is. So next to is the nutrition. So again, I'm repeating this point, beta. Most of the bacteria are heterotrophic, but yeah, some are autotrophic. Those who are autotrophic, they are chemosynthetic. Those who are autotrophic, they are chemosynthetic, right, beta? And they are photosynthetic as well. Is that clear? Is that clear? Now, move to the next part. So autotrophic, which can make their own food. Heterotrophic, which are dependent on others for their food. So heterotrophic, under this category, you have saprotrophs, which can absorb the organic matter. Some bacteria in the symbiotic relationship, that's how they are taking their nutrition. Some are parasitic. But when it comes to autotrophic, beta, two categories are there. Photosynthetic and chemosynthetic autotrophs. Do you know about them? Do you know about them? Yes. Do you know about them? Yes. So autotrophic bacteria may you have the photosynthetic autotrophs, you have the chemosynthetic as well. So chemosynthetic, synthetic means synthesis, chemosynthetic, right? They use the chemical energy for making the food. They are not using in photosynthesis, photon mane light, synthesis means formation. So they are using light energy for the food synthesis, and in chemosynthetic autotrophs they are using the chemical energy. So in the case of chemosynthetic, there are many examples. One here I'm mentioning is of nitrifying bacteria, which can oxidize nitrogenous compound and can get energy. So when they take their energy from the chemicals, when they make their food from the chemicals, chemosynthetic autotrophs, when they make their food from photosynthetic light energy, they are the photosynthetic autotrophs. And it is not mandatory, students. It is not mandatory that if a bacteria is photosynthetic, then it will release oxygen. No, that is not mandatory. So they can show anoxygenic photosynthesis, that is non-oxygenic photosynthesis, and oxygenic photosynthesis. Okay, so anoxygenic means here photosynthesis is going on, but they will not release the oxygen. Why is it so? When will they release the oxygen? See, oxygenic photosynthesis we will see in the case of blue-green algae, which is also called as cyanobacteria. In the case of blue-green algae, which is also known as cyanobacteria, oxygenic photosynthesis is there. They are the first one who carried out this photosynthesis and released the oxygen. Why? Because they are using water. So if water is used as the, right, oxidizable compound, oxygen will release. Let's suppose if water is not used, if H2S is used, yeah, any other compound is used, so that will release. It is the water from where the oxygen is coming out. Okay, right? So photosynthetic pigments are there in cytoplasm. So they are non-oxygenic also. Just purple sulfur bacteria, they are non-oxygenic photosynthetic bacteria. But we are also having the oxygenic photosynthetic bacteria like your blue-green algae. Now beta, here you guys can see most bacteria are heterotrophs. They do not manufacture their own food. They do not manufacture their own food. So majority of heterotrophic bacteria, they are the decomposers, right? They decompose. And decomposers, you know that in the nature, they are very important, right? So curd formation may, these heterotrophic bacteria, they help even in the antibiotic formation. So the one which are saprotrophic, they obtain their food from dead and decaying organic matter. The parasitic bacteria, they are obtaining their food from living host. They are living within a host body, right? And symbiotic bacteria, right, which are in mutualism. So they convert the atmospheric nitrogen into ammonia and nitrogenous compound, right? There are different ways. So here we are discussing this thing that just say I'm giving you one example here. Just say let's suppose your atmospheric nitrogen, it is converted to ammonia and then into nitrogenous compound. That is how they are getting their energy. So best example that we all have studied is rhizobium, right? Even in the junior classes we study that they are in the root nodules of, right, leguminous plants. They help in fixing the nitrogen, right? This is also getting the food. So this is the example of rhizobium, which only in its symbiotic state fix nitrogen. Okay, that's what you have to remember. Okay, chaliye. So respiration beta, aerobic and anaerobic both are there. If it is aerobic, means they are using oxygen. They are not using oxygen. But here there are two words. What are their two words? One is obligate aerobes, another is facultative anaerobes, right? There is no mistake here. It is written correctly. It is written correctly here. When you are talking about the aerobic bacteria, there are two categories: obligate aerobe and facultative anaerobe. Obligate aerobe and facultative anaerobe, right? Obligate aerobe and facultative, anaerobe. This is what we have here. Now, obligate. Obligate in general means strict. Strict. You know, how do I remember obligate? From this word obligate, from this O word, I remember obsession. What do I remember? I remember obsession. Obsession for something. When we are obsessed for something, we need that thing only. We don't want anything else. We just need that only. Right? We just need that only. So here, what am I trying to tell you? Right? That jaise obligate mane obsession means you need that only. So obligate aerobes, they need oxygen. If no oxygen, they cannot respire, they will die. Facultative anaerobe. Normally, why do we say facultative? Facultative means they are not strict, right? But they are not, why am I saying anaerobe? Why not aerobe? Facultative anaerobe is a type of aerobic bacteria that normally respire by oxygen, but if it is not available, then can live without oxygen. Right? Ki normally, theek hai yaar, I'm getting oxygen. Wow, I'm happy. I need that. But for some time, if I have to live without oxygen, they can survive. Right? For some time, if they have to live without oxygen, they can survive. So obviously, they are not true to the aerobic condition. They can survive without them also. So that's why the word is facultative anaerobe. Now, obligate anaerobes, they just need the conditions without oxygen. If oxygen will be present, they will not be able to survive. They are also the facultative aerobes. Normally, they live in the anaerobic condition, but if oxygen is there, they can survive. So you can see obligate means strict, can perform only only aerobic respiration. So facultative anaerobes, right? They are the aerobic forms, but can live anaerobically also. So obligate anaerobes, they can perform only anaerobic respiration. Facultative aerobes, they have anaerobic forms but can live aerobically. So tell me if there is any doubt in these terms. Quick. Done? Sure, these terms are clear? Are you sure? These terms are clear? Beta, you don't need the examples here, and some examples you already know. Komal, start using some common sense as well. Right? You have already read a lot. But if you still want single point, na ki nahi, this example, that example, it means you are not revising. Don't you know methanogens? Methanogens are the obligate anaerobes. They cannot survive in oxygen. So these are the examples that you have already studied. I don't know why do you just like to spam here only? So please pay attention, guys. Okay, chaliye, let's move to the next part that we have here. So next is the reproduction. Obviously, here the main is asexual reproduction. Sexual reproduction is not there. True sexual reproduction is not there. Yes, we talk about the genetic recombination here, but this is not the true sexual reproduction. You do not talk about the gametes here. For the very first time, we have seen the sexual reproduction in Protista, that too not in all the members, right? Few examples are there. So this is, when it comes to the asexual reproduction, binary fission is there. Right, you know that this is a cell, right? It will divide into two. They will just show the growth. Randomly this genetic material will also divide. So, right. So when the conditions are favorable, then the binary fission is there. But let's suppose if unfavorable conditions are there, if that bacteria is not living in the favorable conditions, if there are unfavorable conditions, then in that case, what will happen? The spore formation occur. sporulation, spore formation, because spores are thick walled. They'll protect in the unfavorable condition. They are acting like a structure for perennation. So when there are unfavorable conditions, just say let's suppose bacteria is not having the sufficient food, yeah, bacteria might die. So in that case, they form the spores because they are highly resistant structures. They are resistant to temperature, radiation, antibiotics, chemicals. So they are mostly seen in rod-shaped bacteria, bacillus. You remember in that diagram also, they have made that structure. So that is endospore. That is endospore. Am I clear? That is endospore. Now let's talk about the genetic recombination, which is of three types: transformation, transduction, conjugation. This is the genetic recombination. So this conjugation is somewhere considered as a sort of sexual reproduction. Again, sort of sexual reproduction, not the true sexual reproduction. And this transformation, transduction, if you remember, in Griffith's experiment, class 12th, molecular basis of inheritance, we discussed that. This one in Hershey and Chase experiment, we discussed that. So they are basically the ways by which genetic material can pass from one bacteria to another. But how is it possible? Transformation, to get transformed. Like for an example, you have a cell here, right? If the cell accepts a genetic material from the surrounding, genetic material is there in the surrounding, if that genetic material will get incorporated into its own DNA, then obviously it will have that extra genes. Now, if you will ask me, ma'am, how can it occur? We can do it deliberately. We can create pores in this particular bacteria cell, right? Because we want it to accept it. We can create conditions like that. So in the transformation, right? In the transformation, what will you see? In the case of transformation, you see that there will be a donor cell that will release a piece of DNA which is taken by recipient cell. So we can do it deliberately. In culture media, might be this can happen, right? Now, transduction. In the case of transduction, what is happening? In the case of transduction, just remember this. Just in Hershey and Chase experiment, this has happened. You have used a bacteriophage to insert the genetic material into the bacteria. So virus is involved here. So a small double-stranded piece of DNA is transferred from donor to recipient, but how? With the help of bacteriophage. Just say for an example, this is your bacteria, this is your bacteriophage, right? When they say this bacteria is having its own DNA, this is also having the DNA. It will pass its DNA into the cell. This DNA will get incorporated into the host cell. It will start using host machinery. It will make its own multiple copies. So when it will make its own multiple copies, it will have the DNA of its own plus of this bacteria. So when it will infect another bacterial cell, it will carry that DNA. So transduction is when a virus is used to pass the genetic material from one bacterial to another. Is that clear? Then comes what? Conjugation. Beta, this is a sort of sexual reproduction because here the genetic material is exchanged. Just say, let's suppose there is a bacteria. In the bacteria, if there is a plasmid, plasmid is actually what? It is the extra chromosomal DNA. Just say, let's suppose here you have fertility plasmid. What do you have here? F plasmid. F plasmid. You are calling it as fertility plasmid. It is also referred to as F plus and it is considered as male. Now, plasmid is the extra chromosomal DNA. Means other than, right? Means other than the nucleoid in the bacteria, you also find the extra DNA that is circular, that is having its origin of replication, that can replicate independently. Now in some plasmids, they are responsible for fertility. How come this is? Here, these plasmids, they help in the growth of a tube-like structure, which is referred to as sex pilus, or you can also call it as conjugation tube. We can also call it as conjugation tube. With the help of this, it will attach itself to another bacteria, which is F minus, mane female, mane recipient. It doesn't have F plasmid. Now, This plasmid through this conjugation tube will also pass to this bacteria. So by the end of this process, this plasmid which is without this bacteria which is F minus, so it will also get this plasmid. So by the end of process, both will be F plus. This is conjugation. So from this bacteria, this piece of DNA will pass to this. So that's why we are saying it's the sort of sexual reproduction. So one cell is having F plasmid, so that will be F plus, that will be donor, that will be considered as the male. And the other cell is lacking F plasmid, as a recipient, F minus or female. Donor cell will replicate its plasmid, pass to the recipient cell through that conjugation tube or the sex pilus. That's how it works. Understood? Understood? So this is about the ways of reproduction here. So now let's understand the Archaebacteria, and always remember that Archaebacteria, it shows more similarities, right? Archaebacteria, it shows more similarities with what? Yes. It shows more similarity with the eukaryotes as well. You can check that out. It is important, right? This is your homework. You will read it, that why Archaebacteria shows more similarities with the eukaryotes. Now, Archaebacteria, ancient bacteria, primitive bacteria, oldest of all living fossil, right? It can survive under extreme conditions due to, obviously, it can survive under extreme conditions due to the some special things which are present in its structure. Is that clear? Is that clear? Yes, please. So, they are most ancient, that can live in the most harsh habitat. There are some things which we find which are different from eubacteria. That's why, you know, in 6th kingdom, they were kept in a different kingdom as well. So here you can see in the cell wall, right? Matlab in Archaebacteria, the cell wall is not having the peptidoglycans. That's why I told you that eubacteria is having peptidoglycan. Here they have pseudomurines. They don't have peptidoglycans. They do not have peptidoglycans. They lack that muramic acid. They lack that peptidoglycan. So they are made up of non-cellulosic. They don't even have the cellulose. So they are made up of, yes students, they are made up of, this is their cell wall, it lacks that muramic acid, the peptidoglycan. Rather it is having non-cellulosic polysaccharides. They have the pseudomurines, beta, right? They have different cell wall structure and they are responsible for their survival in extreme conditions. Is that clear? Is that clear? Is that clear? So no peptidoglycan, rather they have pseudopeptidoglycan or protein layers. Okay, so which are more resistant to extreme in salinity, which are more resistant to many chemicals present there. Is that clear? Yes, please. So there is, we call it as S layer as well. So it is more resistant to, you know, that extreme conditions. Even in their cell membrane is also different. It is not made up of bi-layer, of phospholipid bilayer. It is monolayer of lipids, right? And they have branched fatty acids. Very important it is, beta. So this is having the monolayer. It is having the monolayer of lipids which are having branched fatty acids. Monolayer of lipids, it is having branched fatty acids which decrease fluidity, right? So fluidity is the property, you know, that it is not letting the membrane to become a solid or even a liquid. There are the compositions so that they maintain it in a fluid-like condition so that even a cell can grow, even it can exchange the material with the surrounding. And due to fluidity, right, the phospholipids can exchange their position with the neighboring phospholipid and even the proteins. But here, because they have branched fatty acids, so these branched fatty acids, beta, these branched fatty acids, they will decrease their fluidity and tolerance to high temperature will increase. Is that clear? Is that clear? So moreover, they have ether-linked fatty acids. They do not have ester linked, right? So they have what? They have ether linked lipids, beta. They have ether linked lipids, but not ester linked lipids. Lipids like eubacteria. So that's why I was emphasizing that particular diagram. So this is more stable. This one is more stable. So this one will help it to survive under that extreme conditions. So is that clear? So they are ether linked. So they are what? They are more stable. They are more resistant to high temperature and to extreme conditions. So they even have lipid monolayer, beta. So very heat resistant they are. So these are the conditions which are helping them to survive in such extreme conditions. So I hope you are aware, beta. Nobody will teach you such important points. I'm telling you this thing. So Baki, we know that it is the 16S ribosomal nucleotide sequence that is making this Archaebacteria different from Eubacteria. So there are 3 types, beta. Methanogens are there, halophiles are there, thermoacidophiles are there. So when you talk about the methanogens, obligate anaerobes they are. They just need anaerobic conditions. We read about them in biogas, if you people remember, right? Even they are present in the rumen of the ruminants in that chamber. So that's why dung is also having this. So yes, these methanogens, they are responsible for methane gas formation, right? They are present in swamps, lakes, paddy fields, even in the rumen of many herbivores, help in the cellulose digestion, right, beta? Right? So they help in the breakdown of cellulose, told you already. Okay, so halophiles mean salt lovers, right? They can live in the extreme saline conditions. They are facultative anaerobes, right? They are facultative anaerobes. Like they are facultative anaerobes means normally they live aerobically, but if oxygen is not available, they can survive for some condition, some time. So they can live in high salinity, high salt concentration, right, beta? How come it is possible for them? Because they will maintain high intracellular salt concentration. So imagine it in this way. If their intracellular salt concentration is more, then they will not lose the water that they have, right? Rather, they need more water. So that is how, right, they have the enzymes which can operate at high temperature. Example is Halobacterium. Is that clear? Example is Halobacterium. Is that clear? Sure. Sure. Okay. So now if you talk about the thermoacidophiles, so thermal mane temperature, acidity. So they can found in hot sulfur spring, high acidity, right? So they are temperature and acid living. But let me tell you that these thermoacidophiles, they can be aerobic, they can be anaerobic as well, right? They can be aerobic, they can be anaerobic as well, okay? So now eubacteria, U means true. So here you have different names, just cyanobacteria, blue-green algae, right? Remember, they are the first on this planet Earth that release the oxygen due to photosynthesis, right? Then you have mycoplasma, which are also the only bacteria which is without cell wall. And even if you remember, guys, they are the smallest living cells. They are the smallest living cell, which is 0.3 to 0.5 micrometer in length. If I go as per the diagram given in the chapter, 8th chapter, cell chapter, if I'll go as per the diagram, then you will see it's not 0.3 to 0.5. It will be overall bacteria is 0.1 to 0.5. But mycoplasma, yes, in case of mycoplasma, you will say that it is 0.3 micrometer in diameter, right? But we have the example where it is showing 0.15 micrometer. So chlamydia are also the example of bacteria. Actinomycetes is also a kind of eubacteria. Rickettsia is also a kind of eubacteria. So there are different examples. So they were first observed in rainy water, right? Rainy water. So guys, Anton von Leeuwenhoek, right? the scientist, I don't know whether you remember this name or not. So Anton von Leeuwenhoek. Even in cell chapter, we read about it, right? He was the one who discovered the first living cell. He was the one who discovered the first living cell. So yes, he was the one who first observed it in the rainy water and later in the teeth scum and called them, because they were moving creatures, so he called them animalcules. Am I clear? He called them as animalcules. Got it? Done? Done. So next is cyanobacteria, blue-green algae, Myxophyceae. They are mostly found in fresh water, even in marine, in the terrestrial habitat. So they were the first one to produce oxygen on Earth. So they have done what? Oxygenic photosynthesis. You remember? Oxygenic photosynthesis. They were the first one to do so. They were the first one to do so. Now, in their case, they have different structure. This is the diagram which is given in NCERT, which we have to understand, right? So, beta, overall, they can be unicellular, जैसे spirulina. So, spirulina, you remember? Food travelers carry it to the space. You remember this? Food travelers carry it to the space because it is having high amount of protein. We also discuss it in single cell protein, right? Just SCP, which are available in market. What are they? Basically, the spirulina masses are formed, right? So spirulina is a good source of protein. We have colonial, the cyanobacteria as well, just Annabina. We have filamentous also, that is oscillatoria. So oscillatoria, Annabina, if you remember, these were the questions in the NEET 2025. So they are surrounded with a mucilaginous sheath. Can you see the structure there? Structure there? Yes. Nostoc is covered with mucilaginous sheath. Can you see that? Sure. Right, so they have mucilaginous sheath, obviously for their survival and protection. That's why they are covered with this, right? The sheath is basically for their survival as well as for their protection, right? It is for their survival as well as for their production because it is sticky, right, beta? So it protects from desiccation, that is from dryness, right? From desiccation. So desiccation means it will not allow them to dry, right? It also help in colony formation. That's why they are covered. Colony formation. It also help them from harmful substances, right? Even from the UV radiations and all, etc., etc. So yes, it helps them to form the colonies. It helps them from this mechanical damage, and even it also help it to get attached to substratum. To get attached to substratum. So that's the role of mucilaginous covering here, right? So filamentous, colonial, single-celled, this is what we have. Important example. So it's a blue-green algae, not just a chlorella is the green algae, unicellular green algae, but this one is the blue-green algae. That's what you have to remember, okay? Next, filamentous may be homocystis or heterocystis. Homo means same, right? This is what we have learned today. Homo means same. Like all the cells are of same kind, homocystis, like your oscillatoria, one type of cell, they all are performing photosynthesis. Now heterocystic. No doubt in diagram, in NCERT diagram, they didn't clarify it properly, but actually that's the case. Like when you talk about the heterocystis blue-green algae, when you talk about what? Heterocystis blue-green algae, like you have the example of nostoc, you will see there are large size cells also, which are non-photosynthetic. And there are small sized also, which are what? Which are photosynthetic. Are you getting my point? Are you getting my point? So these large size cells, they help in the nitrogen fixation, my dear students. You are calling them as heterocysts. So here heterocyst is the cell which help in the nitrogen fixation. They are large cell. Actually, in the heterocyst, anaerobic conditions are there. Anaerobic conditions are present. Anaerobic conditions are present, right? So, which are required for enzyme nitrogenase to work. So nitrogenase is the enzyme that fixes the nitrogen. It needs anaerobic condition. So in the heterocyst, because they fix the nitrogen, how are they able to fix the nitrogen? Because they lack photosystem 2, right? So no photosystem, no oxygen evolution. So anaerobic conditions will be provided to the enzyme nitrogenase. It helps in nitrogen fixation. Nostoc Annabina is the example. Heterocystis they are, right beta? So small size thin walled cells are photosynthetic. Large size thick walled cells are non-photosynthetic. They are the heterocyst. So it lacks PS2 that helps in the evolution of oxygen during photosynthesis. Is that clear? Is that clear? Are you getting my point? So they do not evolve oxygen. So they have the nitrogenase that help in nitrogen fixation under anaerobic condition. So that is how it works. So here you guys can clearly see, right, the cytoplasm of blue-green algae, because it has to do the photosynthesis. It is having chromatophore. We also use the word thylakoids for it. Is that clear? So they are membrane-bound structure. They have photosynthetic pigments. So inclusion body, gas vacuoles, they all are present. Okay. So gas vacuole is usually they have the membrane around them, but that's not the true lipid membrane. Okay. So symbiotic form may you have Annabina, Nostoc. So you remember Azola, water fern. So that Azola with Annabina, it shows the symbiotic association and fix the nitrogen. And beta, excessive growth of blue-green algae can pollute water bodies. So they will cause the algal blooms. Done. So next thing that we have to discuss here is mycoplasma. So this is what you have to discuss in detail. Jaise actinomycetes, they are ray fungi. They are also a type of bacteria that grows in the culture like a fungus. It is not actually the fungus, but it is like that. Okay. So let's finish this. Right. Let's finish this mycoplasma and then let's have a 5 minutes ka break. Any doubts? Any doubts? Sure. Done. Okay, so mycoplasma, highly pleomorphic. Pleomorphic means it is having different shapes. Why is it so? Because it is the only bacteria which is without cell wall, right? Why? Because it is the only bacteria which is without cell wall, right? It is the only bacteria which is without a cell wall. It doesn't have any cell wall. Is that clear? It doesn't have any cell wall. So, no cell wall means different shapes. Hai na? No cell wall means different shapes. So, that's why I'm using the word pleomorphic. Right? That's why I'm using the word pleomorphic. And let me tell you, they are also referred to as jokers of microbiological park, or even jokers of plant kingdom. So why do we call them as jokers of plant kingdom? Because at that time they were in plant kingdom, right? So smallest living cell, so around 0.3. Wese in general, this is what you have to say, that it is 0.3 micrometer in diameter, right? It is 0.3 micrometer in diameter, 0.3 micrometer in length, I would say. So they are basically facultative anaerobe. So this term is not given in NCERT, but if you will solve the PYQs, you will get it. So what is the meaning of facultative anaerobe? That's why initially I have explained it. So what is the meaning of facultative anaerobe? That normally they live in the oxygen but can survive without oxygen as well, right? So they lacks the cell wall, right? So they form some fried egg colonies like structure in the culture. So they are simple, they are smallest unicellular aerobic prokaryotes, they are even non-motile. So because they have no cell wall, so they can change their shape a lot. So they are the jokers of microbiological park or the plant kingdom. Mostly they are parasitic, but some can be saprotrophs as well. And in their case, again, genetic material is single, linear, double-stranded, no nuclear membrane or envelope is there. So they show budding or binary fission. So because they do not have cell wall, so the antibiotics like lysozyme, so the antibiotics like lysozyme, penicillin, right, cannot destroy it. Why? Why? Because they act on cell wall. So they need, if anybody is having mycoplasma infection, right? If anybody is having mycoplasma infection, that they need the antibiotics which are not targeting the cell wall, but rather than something else. So just say it is sensitive to tetracycline. Why? Because this antibiotic acts on protein synthesis, and it is resistant to penicillin because penicillin is what? It acts on cell wall. So that is why this will not be able to affect this. So overall, these mycoplasma, they were first discovered by Nocard and Rox. These were the French scientists. So they discovered it from the pleural fluid of the cattle. So that's why initially they were referred to as pleuropneumonia causing organism. So pleuropneumonia like organism, which you discuss in cell. In cell chapter, we read about the PPL. They are nothing but the mycoplasma having dimensions 0.1 micrometer. Okay, so actinomycetes ray fungi is also a type of bacteria. So they are branched filamentous bacteria that looks like the fungal mycelium. So they grow in the form of radiating colonies. So they are ray fungi. So ray fungi is not actually the fungi, it is the bacteria. Okay, done. Chaliye. Next is kingdom Protista, right? So you know that unicellular eukaryotes are there. So this kingdom is very important. Do mention it. This is a PYQ. So this forms a link between plants, animal and fungi. Here you will see all type of members are there. Okay, so its boundary of this kingdom, they are not well defined, they are not clarified. So just keep it in your mind. Then here you guys can see, here we have to remember a trick. What's that? CDESP. So how can you remember kingdom Protista? Just remember one trick. It is CDESP. Chrysophyte, dinoflagellate, euglenoid, slime mold, protozoa. Once again, CDESP. Right? Repeat it 10 to 15 times. You will be able to remember this. CDESP. Chrysophyte, dinoflagellate, euglenoid, slime mold, protozoa. Here, these chrysophytes, which will include your diatoms and desmids. That will include what? Diatoms and desmids, right? So these are what? They are photosynthetic. Just see, autotrophic means photosynthetic. Which one? Your diatoms, even the desmids, they both are under chrysophyte, dinoflagellates, and your euglenoids. They are autotrophs. Now, this slime mold, it is the consumer or decomposer. It is just like fungus. They are just like plants. Now, these protozoa, they resemble with animals. Why? Because they are ingestive protista. Right? Obviously heterotrophs and right ingest the solid food as well. So this is what we have to remember. So general characters mein again beta, some are colonial, mostly aquatic. They have, if they have cell wall, we will also see the cellulosic cell wall. Flagella and cilia is made up of microtubules, the characters of eukaryotic cell. So pseudopodia is also there for locomotion. Flagella is also there, but cilia is the fastest one. Right? So they are photosynthetic. Holozoic. Holozoic means they will ingest the solid food. Just in our case, ingestion, digestion, absorption, assimilation, egestion is there like this. So some are saprobic as well or parasitic. So they go for the absorption. And some are mixotropic. The example is Euglena, which is a connecting link between plant and animal. So these are some general characters here. Right, so here you have to see the asexual reproduction is very common. Binary fission, multiple fission, bidding, all of them are there. But also remember that the sexual reproduction first evolved in Protista. Right, in Protista. Right, so yes, the gamete formation is also there. And here the zygotes also undergo the meiosis. So this is what we notice there. So let's discuss it. Let's talk about the holophytic protistans. So holophytic are the one that are making their own food. Okay, okay. So these protistans, just if they are photosynthetic, they are free floating, right? They collectively called for planktons. Overall, देखो, planktons माने passively floating with the water current. Passively floating with water current. Passively floating with what? With water current. So if it is phytoplankton, phyto means plant. So if it is passively floating with the water current and it is photosynthetic, then you will use the word phytoplankton. If it is freely floating mane passively, right? Just go with the flow kind of thing. But they are holozoic, they eat the solid food, then it is the zooplankton. Okay, so let's start the chrysophytes, which you have diatoms and the desmids. So let me mention it here that diatoms are the chief producers of oceans, plus they are also known as jewels of plant kingdom, your jewels of oceans. And your desmids, they only found in fresh water, also referred to as golden algae. And diatoms, they are also the pollution indicator. They are also what, beta? They are also the pollution indicator. Is that clear? Their hypergrowth shows the pollution. So diatoms, very important photosynthetic protistans they are, right? Right. Out of total organic matter synthesized on Earth, half is produced by them. So that's why they are the chief producers of oceans. Moreover, their hypergrowth will indicate pollution. So they are the pollution indicators as well. So beta, they don't have flagella, right? Only in reproductive stage you will see the flagella. So they just passively float with the water current. Now, this point is the most important one. So they have silicified cell wall. Dekho. In the case of diatoms, that's how you are going to see their structure. Jaise, first of all, their cell wall is made up of silica. It is made up of what? Silica. Plus, for cell wall, you call it as shell या फिर frustule, right? It looks like a soap box. Why is it so? Because you will see this upper part is what? It is epitheca. And the lower part is what? It is hypotheca. So this is how, just say in the soap box it fits in, na? This is how its structure is. Okay. Now, when you talk about this cell wall, because it is made up of silica, so soft body, like the organism inside it will die, but this cell wall, silica make it indestructible. But silica will make it what? Indestructible. So with time, what will happen? All that cell walls, they'll be collected at the base of the ocean. It will take millions of years and then it will form diatomaceous earth, which is also known as diatomite, and it is also known as kieselguhr. Kieselguhr. Is that clear? Is that clear? So which is used for gritty, it is having gritty nature, stony sa nature. So accordingly it is used, right? Now, here you see the binary fission. You see that they will divide. Just say epitheca, below it a new hypotheca will form, or this hypotheca, it behaves like epitheca, and below it one more hypotheca form. So that's how when these cells they divide, na, when these diatoms they divide, na, beta their cell size it decreases. Right, their cell size decreases. So in the case of diatoms, when their cell size decreases, then they switch to sexual reproduction. Then they realize ki yaar, we have to go for the sexual reproduction to regain that size. So gamete formation will be there, gametes will fuse, zygote forms in their case. So this zygote is also known as oxospore. It's a Ames PYQ. Okay, it's a Ames PYQ. So oxospore will form, which is also referred to as rejuvenating spore, which is also referred to as rejuvenating spore, which help it to regain its size. Is that clear? Which help it to regain its size. Is that clear? So you people can see this here, right? So silicified cell wall, which we are calling as frustule, right? या फिर shell, it is indestructible. Why? Because it is having the cilia, right? So it forms diatomite या diatomaceous earth या Kysel girl. So it is having that gritty nature. So it is used in polishing or filtration of oils and syrups. Okay, so main points I have already mentioned. So cell wall mein it is thin overlapping shells beta. So this fit together like a soap box. So we have apitheca and hypotheca, chlorophyll A, C, beta carotene, diatoxanthin and dinoxanthins are present here. So reserve food material is oil or chrysolaminarian. Asexual reproduction by binary fission. So gametic fusion, the sexual reproduction also occurs here. But the point here is, right, that will help to rejuvenate the size. So it is the oxospore, which is the zygote, yeah, rejuvenating spore. So they have different shapes, circular, right, that pinnate diatoms are there. So that's how they look under the microscope. You people can see that, right? So oxospore question was there in the AIMS exam. So zygote formed during sexual reproduction in diatoms is the oxospore. So next is the, for desmids. In the case of desmids, you remember, they only found in fresh water. Desmids. Only in clean fresh water you will see. If there will be pollution, they will not clean water. So they will not be able to grow in the pollution. They will only grow in the clean water. Is that clear? Is that clear, beta? Right, so desmids they are, right beta? So in fresh water only, in the clean water only you will get. So they are also referred to as symmetric algae, right? Done. So they will have two symmetric halves. Clean fresh water you will get it, right? So they have cell wall which is having the cellulose. So that is it. They don't have much detail. So yes, they grow only in the clean water, in the fresh water. Done, bache? Right? So they are symmetrical. They are symmetrical. So under the microscope, when you will see, you'll see the proper symmetry there. So yes, they do have cell wall, right? They do have cell wall that will be cellulosic. So this is all that you need to know about them. So they will found in fresh, clean water. If the water is dirty, they will not grow. Okay? They will not grow. Sure? Done, beta? Sure? So they are desmids, right? So yes, the next to it is the dinoflagellates. Next to it is the dinoflagellates, which are also referred as golden brown photosynthetic protista. They are obviously unicellular, but they are motile, mostly marine, but some are in fresh water. So here also you will see that the cell wall is made up of theca, but here this is what you have to remember. Just let's say here, yes beta, here also we will discuss this thing that they are apitheca, hypotheca, but it is not made up of silica, right? Here you will see the cell wall is cellulosic, right? Just stiff plates, stiff cellulosic plates are there. That is written now. So cellulosic cell wall. So there will be a common point from where the flagella will arise in both the direction. So there will be a transverse flagella and longitudinal one, right beta? These are the, so this flagella is not same, it is heterocont. Are you getting my point? Heterocont. They will arise from a common point, but they will move in the two opposite direction. Is that clear? Is that clear? So we call them as whirling whips as well, right? Whirling. We also call them as whirling whips because of their, spinning and rotation type of movement. So from a common point, the flagella arises, from that grooves it will pass. So as I said, the cell wall is called theca, have stiff cellulosic plates. Flagella are heterocont, meaning two different type of flagella. One is longitudinal and it is smooth as well. The transverse one is ribbon-like, right? Hairy as well. Okay, hairy as well. So yes, you need to know that, but they arise from the common point. So they are oriented at right angle to each other. So they produce spinning movements. So we are calling them as whirling whips. Okay, beta. So this part is not that important, but yeah, they do have the different, these are xanthophyll, which will give it the different color. Reproduction mainly asexual, cell division is also there, and carbohydrate and oil is the reserve food material. Is that clear? Right? So they have some special features as well, but okay, you remember this, okay? Done, beta? So just focus on these main points, okay? Done? So there is one unusual thing about the dinoflagellates. Just their sexual reproduction is generally absent, reported in some. The marine dinoflagellates, some they will show the bioluminescence, just Noctiluca. Okay, Noctiluca. But let me tell you that in dinoflagellate there is a special feature. In dinoflagellates, their chromosomes are always condensed. मतलब they are, जैसे normally we say ना, chromosomes they get condensed during the division. Chromosomes are always condensed. That is, they are throughout the, मतलब their DNA is always condensed, right? DNA is always condensed. I would say throughout the cell cycle it is condensed. Is that clear? Throughout the cell cycle it is condensed. That's what you have to remember. Okay. Okay, that is what you have to remember. So chromosomes, they are attached to the nuclear envelope. They are always condensed, they are intact. Okay. Done. So this is the structure, this cingulum, this part, you can see that this is sulcus, right? The groove-like structure from where the flagella is coming out, okay? Right? So that's the diagram. Just keep it in your mind. So some dinoflagellates, this is again a PYQ, like Goniolix beta, right? So this is very important, please pay attention. So some dinoflagellates, right? Like what? Like Goniolix. They produce toxin, which is also referred to as sexy toxin, into the seawater. This toxin is poisonous to vertebrates. It is not killing invertebrates. In invertebrates, it is not getting metabolized. It is just getting accumulated there. Right? It is just getting accumulated there. It is not getting metabolized in that. So marine shellfish, they consume these dinoflagellates and accumulate the poison, which is not harmful to shellfish, but upon being consumed by the vertebrates can cause paralytic shellfish poisoning. So this is Goniolix. One thing about Goniolix, the toxin thing, then you will also see that they will form the red tide. They excessively grow and their hypergrowth will result in the red coloration. So yes, this is important. This is important, not extra, but yeah, this is important, okay? So some dinoflagellates, right, they proliferate in large number, cause red tides of the sea. Again, example is Goniolix. Done? Next is euglenoids beta. So euglenoids may the largest, yes, so euglenoids may that largest genera is of Euglena. So what is this Euglena? It's spindle-shaped organism. So it doesn't have one flagella. It is actually having two flagellas, right? So this is the longest one, right? So this is flagella, but overall in Euglena, so Euglena, which we call as spindle organism. What do we call it? We call it as spindle organism. Spindle shape mane tapering at both the ends, wide in the center. So these are the structure from where the flagella arises, right? This is the basal body. It is also known as blepharoplast. Right, this is that mouth-like structure, cytostome, cytopharynx, right? So there they have the speciality, their outer covering is pellicle, right? So their outer covering is pellicle. This is proteinaceous, right? This is proteinaceous. This is providing it the flexibility. It will help it to show the creeping movement. You will also see parallelly arranged, right? Myonemes are also present there. Here you will see the paraflagellar body also. So overall they have two flagella, one is short, one is long, right? One is short, one is long, and overall you find it in fresh stagnant water, right? The stationary water, in that water you will see this. So it is also having the chloroplast, right? So it can conduct the photosynthesis, and when photosynthesis is not there, right, it can also eat the food. So it is showing what? It is showing the mixotropic nutrition. Both the things are there. So what are we talking? We are talking about the euglenoids, right? We are talking about the euglenoids. So they are group of chlorophyllous and non-chlorophyllous flagellated organisms, right? So largest genera is Euglena. Where do you see fresh water and found in stagnant water? Again a PYQ. So it is having the pellicle, also known as periplast, instead of cell wall. So this is made up of proteins, make the body flexible. So due to which they can undergo the creeping movement. Contraction expansion is there, that is metabolic. So they have parallelly and obliquely arranged myonemes in that pellicle that will help them. Right? Right? That will help it, okay? So two flagellas are there, one is long. So tinsel type mane hairy, right? Hairy flagella is there. So each arises from the basal body, blepharoplast, where both the flagellas, they join. Just let's suppose this. So here you will see a photosynthetic paraflagellar body will form. So let's suppose both the flagella, they arise from the basal body. This is one long flagella, one short. The point where they are attaching, you will find a paraflagellar body there. What will you find there? A paraflagellar body. Is that clear? Paraflagellar body will there. So they show this creeping type of movement. So apical end mein, you have seen, right, when you see their mouth. So dekho, mouth is cytostome. That canal-like structure is gullet or cytopharynx. Then comes the reservoir. So it helps in ingestion of solid food particles beta. So there is one more structure in euglenoids. This is extra stigma eye spot, which is attached to the reservoir at the level of paraflagellar body. That is it. So contractile vacuoles, not in this only, it is in fresh water, it is having the contractile vacuole. So what is the role of this contractile vacuole? It plays role in salt and water balance as well as in excretion. So nutrition is mixotropic. They can show saprophytic or holozoic, ya fir photoautotrophs when the light is there, right? When the light is there. So they have chlorophyll A, B, xanthophylls and carotenoids. Am I clear? So next is the slime mold beta, which is compared to what? Fungi, because it is also the decomposer. Very interesting organism, right? Very interesting organism. Let's finish this first and then let's have a 5 minutes ka break and then we'll go for the fungi. Okay, done. Any doubts? Any doubts? Okay, so when you talk about this, you see they are the consumer, decomposer or the protestant fungi, right? So it is showing the character of both animals and fungi as well. So we also call it as fungus animal, okay? So they are saprophytic, they show the absorption, they do not have chlorophyll. So when there is the decaying twigs, they will grow around it. They will grow around it. Do you know in their case what will happen? In their case, when the conditions are suitable, this is important, this is a PYQ as well. When the conditions are suitable, right, they'll form the aggregates, plasmodium. Matlab, jaise, let's suppose these slime molds, they will come closer, they will come closer, they will come closer, they will come closer, right? Their common walls will get dissolved. Are you getting my point? Right, then their common walls in between them, they will get dissolved. So if their common walls in between will get dissolved, what will happen? All the nuclei will be together. You will see a multi-nucleated condition. You will see what? Multi-nucleated condition. Yes or no? Right? Yes or no? So you are using the word plasmodium for it. So this is not that plasmodium which cause malaria. Nah. Here you are talking about this aggregation. This is the important thing to understand, that when under suitable conditions, right, the conditions are suitable, then they form an aggregate called Plasmodium, which may grow up to several heights. So yes, it's a PYQ that whether they grow under suitable condition या unsuitable condition to form plasmodium. Suitable condition, right? So common walls will dissolve. It will form a multinucleated structure, right? The syncytium is the word that we use. So they are surrounded only by plasma membrane. Matlab normally when you see the slime mold, it's outer covering, right? Normally it is having the plasma membrane only, right? Normally it doesn't have the cell wall. It is without a cell wall. So vegetative parts may only plasma membrane. That's a very important point here. Vegetative parts may only plasma membrane. So next is when the conditions are unfavorable. When the conditions are unfavorable, then this plasmodium will further grow and it will form a kind of fruiting bodies, right? Just fruiting bodies, this is a feature of fungi as well. So it will show the similarity with all the three. So when conditions are favorable, plasmodium will form, but when the conditions are unfavorable, it will start making a fruiting body in which it can form the spores, right? But spores are having the cell wall. So vegetative part is without cell wall. This is very important. Vegetative part is without cell wall, but spore is having cell wall, right? So vegetative part, it is without a cell wall, but the spore is having cell wall. Done? Done? So they are extremely resistant and survive for many years and under adverse condition. So at one stage of life cycle, they have amoeboid structures, amoeba-like, irregular structure, right? And non-cellulosic, but spores have cellulosic wall. Matlab, because they are just creeping, they have that amoeba-like structure without cell wall, but spores are having cell wall. So these are the characters that they are showing. So plasmodium is the term that we are using here, right? Right? So vegetative phase resemble animal, pseudopodia-like, no cell wall, reproductive phase resemble animal, and plus because it is forming the sporangium, a fruiting body. So basically they are forming the fruiting body, so it is also showing the fungi-like nature, right? So spore forming nature is just like fungi. So fungi-like feature may spore forming या fruiting bodies, plant-like may they have cell wall around spores, animal-like may, which is the plasmodium, without cell wall. So yes, they do show the characters of all three. Okay? Okay? Right? So next is, sometimes these slime molds, they are cellular, sometimes acellular, right? Sometimes what happened, beta, these slime molds, they come closer, right? They come closer. Their common walls, they do not dissolve. Then it is pseudoplasmodium. When their common walls do not dissolve, they form aggregates like this. Okay, so next is beta protozoans. So again, they are compared with the animals. All of them are heterotroph, some are predator or parasitic. So they are believed to be the primitive relatives of animals. Understood? So on the basis of their locomotory organelles, we are dividing it. Just let's suppose if it is amoeboid, so it is having pseudopodia, right? From class 9th, this is what we are reading. This is what we are reading, right? That when you are talking about the amoeba, which is a freshwater protozoan, it is having what? It is having pseudopodia. So pseudopodia is what? Pseudopodia is the false feed. What's that, guys? It is false feed. So pseudopodia needs actin filaments, या microfilaments for its formation. Again a PYQ. Done? So it is freshwater organism. It lives in, right? They can live in seawater also, moist soil mostly, right? Free living, a few are parasitic. So they have false feet. Marine forms may you will also find silica shells. Okay, so amoeba, which is in the freshwater, amoeba you see in the freshwater. Amoeba is also having the contractile vacuole. So this one, entamoeba histolytica, which cause amoebiasis or dysentery, you know that it lives in human large intestine. So guys, when you will read this chapter from NCERT, in the bacteria section, in the Monera section, you will find some diseases name as well. So do read it and do find their causative agent. Done? So obviously flagellated protozoan, which will have flagella, right? Trypanosoma, which causes the sleeping sickness. Just say you have Trypanosoma gambiense. Trypanosoma cruzi is the another species which causes Chagas disease. So in case of Trypanosoma, the vector is CC fly. And in this Trypanosoma cruzi, it is redubit bug. So Leishmania, Kala-Azar, dum-dum fever, black fever. So here the vector is sandfly. Flea-bottomous species, है ना? So ciliated protozoa, obviously they have cilia for the locomotion, right? So here you see the slippers organism, if you remember, right? That is your paramecium. You remember? So they have cilia all over the body, which will help in its locomotion, and moreover it will move the food laden water into this mouth as well. So in paramecium you will see they also show the dimorphism in nucleus, large and smaller one. Smaller one will control the reproductive function, larger one will control the vegetative function. In some strains you also see in paramecium, in some strain you also find the kappa particles beta, which are basically the toxins. So yes, they are freshwater or marine, few are parasitic. Cilia is for the locomotion. They have the gullet for ingestion and egestion. So cilia movements, they cause the water laden with food, matlab water which is having food to pass into the gullet. So as I told you, this is again a PYQ, guys. So nucleus here will show the dimorphism, micronucleus which will control the reproductive part, and macronucleus is there. Is that clear? So paramecium is the example. So paramecium is also known as slippers organism. It looks like a slipper. Then comes the sporozoan. So they just form spores. They do not have locomotory organ. So they all are endoparasite. So locomotory structures are absent. They are the infectious spore-like stage they have. That's why sporozoan. So example is plasmodium, which is basically the malarial parasite. Is that clear? So you know that here the vector is female Anopheles mosquito. Okay, female Anopheles mosquito. Okay, baki amoeboid, they are also known as sarcodines, right? We just discussed these protozoans now. So they are also referred to as sarcodines. So they are also the sarcodines. So that's what you have to remember. This is all about kingdom Protista. So next is the kingdom fungi that we have to discuss, okay? Next is the kingdom fungi. So ready? So next is what? Next is the kingdom fungi. Okay, so multicellular. We have the example of yeast, which is unicellular. So they are achlorophyllous. That is, they do not have chlorophyll, right? So we will see they either show saprotrophic nutrition, या फिर parasitic. Cell wall is basically chitinous in majority, right? So this is what we have to discuss. But let's have a 5 minutes ka break. Okay, let's have 5 minutes break. Done? So we are about to finish it. Stay tuned. let's start again. Okay, let's start again. So the next thing that we have to discuss is kingdom fungi. Okay, so here okay, let's start. So these are the general points here. Okay, so in the case of kingdom fungi, you have to remember pineapple, banana, dates. What you have to remember? You have to remember pineapple, banana, dates. That's what you have to remember. So P for Phycomycetes, A for Ascomycetes, these are the classes there, right? B for Basidiomycetes, and D here is Deuteromycetes. So that's what you have to discuss here. Are you getting my point? So Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. So when you are talking about the Ascomycetes, जैसे Phycomycetes is referred to as what? Phycomycetes is basically the algal fungi, right? No, it is not given in NCERT, but yeah, under this particular Phycomycetes, which is referred to as algal fungi, we also discuss, right, Oomycetes and Zygomycetes. So it is also the, just in some books, Oomycetes is actually referred to as algal fungi. So it is also called as egg fungi. And this one is the conjugation fungi. So that's what we have to discuss here. So Ascomycetes is basically the sac fungi, Basidiomycetes is the club fungi, and Deuteromycetes is the fungi imperfecti. Why? Because here there is no sexual stage. So when you look at this structure here, what we have to discuss, see here basically they are multicellular, but your yeast is actually unicellular. So mycelium, we will start with its basic structure. We will talk about the mycelium, the thread-like structure. But before that, what is there? Dekho. First of all, here in the case of fungi, you will talk about such thin structure. We are calling them as hyphae. What do we call them? We call them as hyphae. Now, network of these hyphae, right? Network of hyphae, what's that? Network of hyphae is referred to as mycelium. So these are the thread-like structures which are making the fungi. Okay. Now, when you are talking about these hyphae, if let's suppose if they have no central, no wall in between, if they have so many nuclei, so if this is multinucleated, so you know that for multinucleated we also use the word cenocytic. Yes, we also use the word cenocytic. So these type of mycelium are aseptate, right? You will see that in the case of Phycomycetes, right? You will see such type of mycelium in Phycomycetes. Now, if they have this sort of partition in between, right? If they have this partition in between, the septum, plural is septa in between, then it is septate. Okay, so obviously now here nucleus will be present in each part here. Now, where will you see this? You will see this in Ascomycetes, Basidiomycetes, Deuteromycetes. Now, when it is the Ascomycetes in the hyphae here, you see a pore-like structure. If beta, it is a simple pore, right? Just you will see it in Asco and Deuteromycetes. If it is a simple pore like this, right? Nucleus will not pass through it. Other things will pass through it, right? It is the simple pore. And in the case of Basidiomycetes, around this pore, you will see the kind of swelling, right? You will see the kind of swelling structure here, bean-like structure here. Then you will call it as dolipore septum, right? And this is something that you see in the case of Basidiomycetes. Where do you see this? You will see this in the case of Basidiomycetes. Got that? Yes, please. So here you people can see this very clearly. So cenocytic mycelium, where you will see it's multinucleated aseptate, Phycomycetes. Septate will be there in Ascomycetes, Basidiomycetes, Deuteromycetes. So network of hyphae is called mycelium. So if these septum, they have the simple central pore, just say Asco, even in Deutero it is like that. And in higher fungi, like your Basidiomycetes, dolipore septum is there. Matlab that opening, that pore-like structure is having the swelling, this kind of structure we have. So this is the dolipore septum. Only in the rust and smut fungi it will not be there. Otherwise it is present. Understood? Then next to it is what? Right, beta, we will discuss the habitat as well. Please stick to the topic. Do not, right, do not break the flow, or else please do not break the flow here. So next is what? The cell wall. The cell is eukaryotic. What sort of cell wall do they have? Please pay attention. No doubt it is the chitin, which is also referred to as fungal cellulose. This chitin we also see in the exoskeleton of, today itself we discussed it. We also see it in the exoskeleton, right? We also see this in the exoskeleton of insects. Exoskeleton of insects may we also see this chitin, if you remember, and this chitin is nothing but the homopolysaccharide. Homo means same, like same types of sugar are going to make it, that is N-acetyl glucosamine. So chitin is also the second most abundant organic molecule on this earth. It is also the second most abundant organic molecule on the earth. So in some fungi, this is important, in some fungi, the cell wall, beta, it is made up of cellulose only. So that is Phytophora and Phythium. So they are the members of the Phycomycetes. So reserve food material is oil and glycogen. So if somebody will ask you, without any exception, all the members of the fungi, right, without any exception, all the members of the fungi, do they have the chitinous cell wall? No. There are some, right, there are some which will show you what? Which will show you the cell wall made up of cellulose. Is that clear? Is that clear? And overall, beta, we know it very well that fungi, they grow in the warm, humid condition. There are certain fungi which can also grow on dung. If they are growing on dung, you can use the word coprophilous for it. Right? They are the coprophilous fungi if they are growing on the dung. So they need the warm areas, right? Right? They need the warm conditions for their growth. Is that clear? Is that clear? So on wood they can grow, on dung they can grow. So they have hyphae, network of that hyphae, because one slide is deleted here. That's why I'm telling you, network of hyphae is known as mycelium. So nutrition is ultimately heterotrophic. They are saprophytes as well, right? So fungi will always release. Just say, let's suppose on food, right, it will release the enzymes on food, right? This food will get digested, of course, and then this digested food is absorbed by fungi. So in case of fungi, you will see extracellular digestion is there, right? You will see extracellular digestion is there. Like fungi absorb pre-digested food, right? The food which is digested, pre-digested food is absorbed by fungi. So digestion will take place outside the body. So this is the extracellular digestion here. So parasites, right, many, they've developed a structure called haustoria, which will penetrate into the host and will take nutrition from that. And symbiotic association may, we know that lichens are there, where you will talk about the algae plus fungi, and mycorrhiza, where we discussed in the case of roots of higher plants plus fungal hyphae, that is Pinus is the example. So do not forget about it. Is that clear? Do not forget about it. Is that clear? Right. So next is the vegetative reproduction. So when we talk about the vegetative reproduction, fragmentation is there. Part of mycelium will break, will grow into new. Fission can also occur, but it will be in unicellular fungus like your yeast. Right. Budding is also there. Right. Outgrowth will develop. That is also you will see in the case of yeast. Otherwise, you will see what? You will see the fragmentation in multicellular fungi. So here also beta spore formation occurs. Asexual reproduction may be or sexual may be. You will see the spore formation. So when it comes to the asexual spores, because see, say algae you have seen na. So if you talk about the fungi, their main body is actually haploid, right? So it is haploid. It has to form the haploid spores. It will go for what? For mitosis. So here you will see mitospores are there. That is by mitosis spores will form. So spores are what? They are single-celled reproductive structures. They can be motile, they can be immotile, they can be thick-walled, they can be thin-walled as well. So in the paper, they can ask you the question, which one is the thin-walled spore, ya thick-walled spore, ya asexual spores, ya sexual spore. So zoospore, which we also discussed in the case of fungi, algae. So zoospore, first of all, it is endogenous in nature. What is the meaning of endogenous? Just say, let's suppose fungi is having the hyphae, right? On one of its branch, a structure will form, sporangium, in which spore will form. So when do I call it zoosporangium? When inside it is the zoospore that form. So they are motile, they are thin-walled, already told you about this, right? So they can have single flagella, they can be biflagellated, they can be pear-shaped, that is pyriform, they can be kidney-shaped. So zoospore is the motile one, is the thin wall. Then comes, so it is endogenous in nature because it forms, just say this branch is sporangiophore, this is having the sporangium, spore form inside is the endogenous, right? So these spores, they germinate into hyphae. So next is beta, the sporangiospore. Again, if it is sporangiospore, it forms within a structure. It forms within a sporangia. Got it? It forms within a sporangia. So yes, it is thin-walled. It is non-motile spore. It is having thin walls. It is not showing the movement. It is also produced endogenously in a sporangium. So it forms under favorable conditions. Just in Rhizopus, in Makar, you will see this, right? But aplanospore, this is also the term, but aplanospore term for the sporangiospore, you also use the term aplanospore because it is non-motile, it is thin-walled. But this term is more common in the case of algae. We have just discussed it today. Okay. Then comes the chlamydospore, right? So aplano या फिर sporangiospore or zoospore forms in favorable condition when the conditions are favoring the growth of the fungi. But when they are not favoring, anything can be there, temperature, pH, food availability, anything can be there. So under unfavorable conditions, you people will see thick-walled spores will form. And when they are thick-walled, they'll be resistant, right? They will be resistant. They later get separated from this and they help in the perennation. So this is again non-motile. So you can say that when the favorable conditions will come, they'll give rise to new individuals. Is that clear? Is that clear? So chlamydospores are there. Then oidea, when let's suppose wherever you are growing your fungus, if it is having too much of sugar medium, right, then the parent cell will get separated, right, and they form the oidea. Okay, so Rhizopus and Makar can show that. Now, again, asexual spore, the very important one is Conidia. Beta, this is not an endogenous spore, this is exogenous. Just there will be the branch of hyphae, which you are calling as conidiophore. So that conidiophore, on that conidiophore, you people will see the spore formation will be there in the form of chains in basipetal arrangement. Okay, so it forms exogenously. This is your conidia. So they are non-motile spores, exogenous spores, and form in a basipetal manner. Is that clear? They form in a basipetal manner. Is that clear? Is that clear? So it means, yes, please. Basi mane base, petal mane movement. So movement is from top to bottom. Is that clear? Is that clear? So movement is from top to bottom. That's how you can remember. It means that the young spore, you can see that young spore is at base, right? Young spore is at base. The older one is at the top. That's what you have to remember this. So these are the conidia. In the Ascomycetes, we see that. So it is the asexual spore, the question can come. So sexual reproduction mein beta 3 steps are there. Very first thing is, there is the, jaise ye dekho. Here you can see. Let's suppose these are 2 gametes, या 2 hyphae. First of all, their protoplasm will fuse. Their nuclei will not fuse. Their protoplasm. You will call it as plasmogamy. What will you call it? You will call it as plasmogamy. Why? Because their protoplasm is fused, not their nuclei. Now, if their protoplasm is fused, so this is the haplophase. Why haplo? Because it is still haploid. Now, because here the nuclei, they are not fused. So can I not call it as dikaryophase? Yes, I can. So in your Phycomycetes, these nuclei fuse immediately. Fuse immediately in your Phycomycetes. But in Ascomycetes and Basidiomycetes, it takes time. So if it takes time, majorly in basidiomycetes, if it takes time, right? If it takes time to fuse, so yes, they will have a prolonged dikaryophase. So after that, there is the second step, karyogamy. Karyon, you know that nuclear fusion. So don't you think a diploid nuclei will form? So I will call it as a diplophase, है ना? But here you know that main plant is haploid, main thallus is haploid rather. So meiosis will occur. In diplophase, meiosis will occur, right? So this is the diplophase. So after that again, meiosis will occur, again haploid structure, again they will grow. So first of all, fusion of protoplasm, either it between two motile and non-motile gametes, plasmogamy. So then the nuclear fusion will be there, karyogamy. Then meiosis and the zygote will be there. So we can also call the zygote as zygospore because it is undergoing the meiosis, and haploid spores will form that will develop into that hyphae again. So when a fungus reproduces sexually, beta, right, two hyphae of compatible mating type, जैसे in some fungi, right, जैसे some fungi are your homothallic fungi. Homo माने same, मतलब they can fuse, right, the same, मतलब they don't need the different strains to get fused. Some are heterothallic, they are two different strains of fungi. So some fungi, they show, right, they only fuse when they are of different strains. So one is considered as plus, other as minus. Here one is considered as male, yeah, another is female. But some can fuse with the same strain, some cannot. So that's why we are saying, so it depends what is their compatibility. So on the basis of their compatible type, they'll come close, they'll fuse. So when they fuse, you know that if these two nuclei stage, if it persists, so we call it as dikaryophase. So this dikaryophase we see in Ascomycetes as well in Basidiomycetes, mainly in Basidiomycetes, beta. You people will see this phase. Is that clear? Is that clear? So, right, so karyogamy occurred just before meiosis. So life cycle has three phases. One is beta haplophase, another is the dikaryophase, another is the diplophase. Got that? Done? So dikaryophase is a feature of Ascomycetes and Basidiomycetes, particularly this, right? Particularly this. So because karyogamy is not immediate after plasmogamy, that's why dikaryophase is there. So one by one we will read it. So sexual spores are beta zygospore. It is diploid because this is the one which will undergo meiosis. Oospore is also the diploid spore. Then Ascomycetes will have ascospore. Basidiomycetes will have basidiospore. So it is endogenous spore. Important PYQ. This one is exogenous spore. Done, beta? So where the spores will form? The spore will form in the structure which we are referred to as fruiting bodies. So in fungi, yes, you are going to see the fruiting bodies. So sexual reproduction is absent in Deuteromycetes. That's why you call it as fungi imperfecti. So how do you classify the fungi? Again, a PYQ, all right? Firstly, we will see the morphology of their mycelium, whether aseptate, septate, then how the spore forms, and on the basis of fruiting bodies. So this is a PYQ, this is a previous year question. So what sort of morphology mycelium is having? What is the mode of spore formation? What is the fruiting type of fruiting body is there? On that basis, it will separate. So you have Phycomycetes. Right. So yes, beta, in some literatures, let me tell you that in some literatures, yes, they genuinely have mentioned this Oomycetes as the algal fungi, right? But just to avoid any sort of confusion, I'll simply say Oomycetes. You can also call it as egg fungi, right? So algal fungi it is because it is very primitive. Zygomycetes is the conjugation fungi. So here, remember, there is a trick for the examples. RAMP. So RAMP means R stands for Rhizopus, which is also the bread mold. This is the one that grows on the bed. A is albugo. It causes the white rust of crucifers. M is mucker. This is the dung mold. And from P, you can remember Phycomycetes as well as Phytophora. So which causes the late blight of potato. Late blight of potato. So this is the trick to remember the examples because question will come from the examples. So class Ascomycetes found in aquatic habitats on the decaying wood in moist as well as the damp places, or it is the obligate parasite on plants. So you take the example of Albugo candida here, white rust of crucifers. So oomycetes, as I said, what do you have to call it, right? Oomycetes, what do you have to call it, right? Just to avoid the confusion, वैसे in some literature it is given like that, so you can call it as egg fungi. ठीक है? So here what you people will see, the mycelium is cenocytic, mycelium is aseptate. Already you have discussed it. So in oomycetes actually, right, which comes under the phycomycetes, the cell wall is cellulosic. Asexual spores में motile is zoospore and the aplanospore is there given in NCERT. So sexual spore which is diploid, it is 2N. Oospore, so Albugo candida which causes the white rust of crucifer. Phytophora infestans, late blight of potato या फिर Irish potato famine. Do remember this, right? This one is important. So early blight of potato will be caused by Alternaria class Deuteromycetes. It is the late blight of potato. Late blight of potato. Then zygomycetes, conjugation fungi, chitinous cell wall, aseptate, cenocytic. Here you will see sporangiospores beta. Sexual spore mein zygospore. Zygomycetes, zygospore, because here in it meiosis will occur, no motile stage. So just say the one which grows on bread, right? That is the bread mold. Makar is the dung mold. That's it. Then comes Ascomycetes, sac fungi. Why do we call it as the sac fungi? Because during its sexual reproduction, it will have the formation of these sac-like structures which we call as ascus, whose plural is asci. So many asci will asci together, they form the fruiting body, ascocarp. So this is what you discuss in the sexual reproduction. Okay? Okay? Right? Done? So it includes saprophytic, even the decomposer, even the parasitic and coprophilous. So coprophilous, I told you already, they can grow on dung. Then they are multicellular. One exception is there, yeast, and you know that yeast will show budding, yeast will show fragmentation. Yeast will show budding, yeast will show fragmentation. Mycelium is branched, septate, haploid. Here cell wall is chitinous. Asexual spores, very important, conidia. So conidia, already told you, conidia is thin-walled, it's non-motile, it is exogenous spore. It is thin-walled, non-motile, exogenous spore. Thin-walled, non-motile, exogenous spore. So motile structure do not occur in its life cycle. Important information, that's why I have underlined it. Next to it is what? Next to is the sexual reproduction. So male sex organ या male gametangium is antheridium. Female one is escogonium. Right? The female one is escogonium. So here what will you see? Except the yeast. Right? Here plasmogamy occurs, but karyogamy is delayed. So when karyogamy is delayed, we know that dikaryotic phase will stay. Just say for an example, let's suppose if these two hyphae, they are fusing, right? So karyogamy is delayed. So this is the thing that you will see. So some dikaryotic cells here, beta, they will behave like ascus mother cell here, right? They will show karyogamy, and then this diploid nucleus having ascus mother cell. So it will grow into the young ascus. It will grow into the young ascus. So in the case of Ascomycetes, there is young ascus which is having 2N, which will undergo meiosis. And imagine, just say, because this is Ascomycetes, ascus it is having. Just say this is the ascus. Let's say it is having the 2N nucleus after this karyogamy. Now, after this, you know that meiosis will occur because that is fixed. So how much spores you will have? You will have 4 spores. They all will be haploid, right? But immediately after this, you will also see that mitosis will also occur. Right? So after mitosis, after meiosis, this is 2N, it will undergo meiosis, 4 haploid spores. Then immediately mitosis will occur. So this nuclei, obviously because it is mitosis, right? You know that it is the equational division. So from these 4 haploid spore, 8 ascospores will develop. 8 ascospores will develop later. So in one ascus, you will see 8 ascospores. So there are different types of fruiting body. So as I said, so many ascus, they together form ascocarps, which are of different type. So the fruiting body here is the ascocarp. Fruiting body here is the ascocarp. So each ascus will show me 8 spores. So ascus is actually the site where the karyogamy and meiosis will occur. So which one is the complex fruiting body here? Ascocarp. So examples में जैसे yeast है बेटा, you know that Saccharomyces cerevisiae. So it is used in it is also the baker's yeast or the brewing yeast. It is used. So you have penicillium, blue-green mold, from where we got the first antibiotic, penicillin. Aspergillus niger, it is basically the lab weed, yeah, guinea pig of plant kingdom. So when I'm specifically talking about Aspergillus niger, what are we getting from that? Citric acid. What are we getting from it? We are getting the citric acid from it. Then Claviceps purpura, ergot of rye, source of LSD, that is your lysergic acid diethylamide. Not given in NCERT, but a lysergic acid diethylamide, right? So lysergic acid, so this is a powerful hallucinogenic agent. So Moschella esculenta, morels, which is basically edible. You have the truffles, tuber estivum, which is again edible. Neurospora species, Drosophila of plant kingdom, right? Because you know that the genetic based experiments are on this. So there are other examples also. The Gibberella that you will study, that also belongs to this. So yes, you have to remember their economic importance as well. So dekho, yeast is only a unicellular member, saprotrophic it is, sometimes parasitic. So it is having zymase, so it is facultative anaerobe. Facultative anaerobe, right? So then next to it is the penicillium, blue-green mold, antibiotic is obtained. So Aspergillus, right? So Aspergillus, there is one species of that, Aspergillus flavus. Beta, this is again a PYQ, this is important. So it grows on the stored grain and ground nuts and produces carcinogens. Okay? So it can contaminate lab culture, various foodstuffs, right? So flavus can grow on the stored grain, so it can produce the toxin, aflatoxin. Done. So Claviceps ergot of rye, source of lysergic acid. So it is used for preparing LSD, a hallucinogenic drug. Is that clear? A hallucinogenic drug. So Moschella esculenta, morels, edible ascocarp is there, beta. So tuber estivum, truffles, this one is also edible. Jaise morels, this is also referred as Gucci. This is very expensive to expensive fungi, but it is having medicinal properties. So Neurospora crassa is actually the pink bread mold. Okay, Neurospora crassa is actually the pink bread mold. So Gibberella also belongs to this. Is that clear? Done. Sure. So they are poisonous mostly. Do remember that, right? So one is done. So next is the Basidiomycetes, the best decomposer, the club fungi. This is having the Basidium. Plural is Basidia, right? And these Basidia together form Basidiocarp. This is the fruiting body here, right? This is the fruiting body here. But these spores, they form exogenously, not endogenously. Bullet points. So you can see that it is the most advanced fungi, most commonly seen fungi, your mushroom, right? They belongs to this. So they are the best decomposer. They secrete the pectin. So mushrooms are there, toadstools are there, puffballs are there, even the bracket fungi. You have seen it. Do check it. Do check the images, right? Do check the images. You have seen it. Is that clear? Is that clear? Sure. So next to it is they can grow on the soil logs, on tree stumps, right? They are parasitic also, just rust and smut. Very common example. A repeatedly asked, right? repeatedly asked question. So they can decompose cellulose as well as lignin, which is not metabolized by other fungi and even bacteria, but Basidiomycetes can do that. So beta, branched and septate dolipore septum is there. So nucleus will not pass through it, but other thing, so motile structures you are not going to see in the Basidiomycetes. Very important it is. So asexual spores are generally not found. So these the points, they are from NCERT, they are very important. So they have vegetative reproduction by fragmentation is very common. It is very common. Okay. Then these 2 points are very important here. Understood? So mycelium, 2 types of mycelium are there. So secondary mycelium is the one which is having the dikaryotic phase, right? So you can see this part beta. This is also multicellular, but it is having single nuclei, right? It is having single nuclei, right? So many multiply by oidea, conidia like spore, but you know that asexual spores are not present. So they are generally short-lived. So that's why we say asexual spores are generally absent here. So secondary mycelium mane, the one which is having these 2 nuclei. So there is a very interesting thing that you see in Basidiomycetes. You see a kind of tubular connection, clamp connection that is basically done when we have to convert this monokaryotic mycelium into dikaryotic na. So this tubular connection, clamp connection helps in that. So yes, there is nothing like, I will not discuss it in detail, but yes, you can remember that this clamp connection thing is associated with converting this monokaryotic mycelium into dikaryotic. It is in Basidiomycetes. Because directly from the dolipore septum, nuclei cannot cross. So in between these 2 sections, this type of tubular structure will form. So when 2 nuclei will form here, one will go to this will divide into 2. So that's how monokaryotic will convert it to dikaryotic. So I will not emphasize it. I'll be very clear here, right? So somatogamy is there. Jaise ye dekho, 2 hyphae, they are fusing. You are having dikaryon stage. Now here, others, you are single, they are still having 1 nuclei. So they will form the clamp connection. These nuclei, they will divide. One will pass to the nearby cell. So this clamp connection, these tubular connection actually help in the proper division. They are converting this monokaryotic mycelium into dikaryotic. So once beta, this dikaryotic mycelium form na, so on it, this club-like structure form. So then what will happen inside it? This nuclei will move, they will fuse, and then they undergo meiosis, and over it you will see these spores will form. So here you can see, right, these nuclei, they will move here, they will show karyogamy, then meiosis. So basidiospores, they form exogenously. Ascospores, they form endogenously. So these are the point where you have to give your attention. Okay, okay. Okay, so yes, they multiply by different types of spore, but mainly you know that sexual spores are present, right? So it is the basidium in which karyogamy and meiosis occur. So basidium is having 4 meiospores. Why? Because they are the result of meiosis and they form exogenously. So ascospores are endogenous spore, but they are exogenous spores. Understood? So that's it. So basidiocarp is the fruiting body. Sex organs are also absent here. So do revise it from the NCERT as well. So it is the plasmogamy or somatogamy due to which the fusion of this hyphae will be there. So this is already I have explained beta, even the clamp connection also. So I will jump to the this part, because these points, right, you already know that this fusion will occur, basidiocarp will form. So let's jump to this example part. So bracket or shelf fungi, trust me, you guys have seen it. Do check the images on the Google, you have seen it. So they are similar to, right, they are similar to brackets, that's why they are known as the bracket fungi. Then comes the puff balls. So fruiting bodies are puff like beta. Only young basidiocarp is edible, right? So there is one more, so these species not required, that's it. Next, mushrooms in which we eat the basidiocarp, they are umbrella like, right? Some are edible, not all mushrooms are edible. So when it comes to the delicious mushrooms, Agaricus campestris, Agaricus bisporus is there. So Agaricus campestris is the field mushroom. This one is the guild mushroom. So some species of Agaricus are also poisonous. So we cannot say that they all are edible, right? They all are not. And toadstools are not at all edible. They are the most poisonous mushrooms, right? So Amanita phalloides, Amanita muscaria. So muscaria is having the hallucinogenic agent, and Amanita phalloides, it's very deadly, right? It's death cup, and it's very deadly. It's very deadly. Okay. Okay. Done. So this is, from this now we get a toxin, and that can stop RNA polymerase 2. So if it can stop RNA polymerase 2, no mRNA will form. Then obviously how come the protein will form? So it is having the inhibitory act on it. So they produces the toxins that can block RNA polymerase 2, and obviously the protein synthesis will stop because cell cannot function and die. So there are the toxins which are available. So this, they can stop this. So it is amatoxin which can do that. This is important. So it is highly dangerous, this particular mushroom. So smut fungi and rust fungi are there. So smut, S-M-U-T, you have to remember Astelago, right? So they cause the smut disease on plant. They infect the seed and form black kind of spores, right? So loose smut of wheat is called by it. Then rust beta, from rust you have to remember Puccinia. Puccinia graminis is going to cause that. So it is loose smut of wheat, but this is the rust disease, right? So here you will see rusty, some pustules like structure will be there. Here you will have black kind of spores, right? So they infect the wheat. So one is causing smut, one is causing this. Okay, done. Done. So Puccinia, but that's it. So Deuteromycetes is the next one, beta. Deuteromycetes is the next one. So it is the fungi imperfecti. Why is it so? Because it doesn't have the sexual stages, only asexual stages are there. So here you have tricks, जैसे taco is there, triangle and circle, जैसे taco, trichophyton, trichoderma, alternaria, coletotrichum is there. So here there is no sexual stage, no perfect stage, right? So the fungi in which sexual stage is absent, they are kept in this part. And let's suppose if in near future their sexual stage is found, then they are placed in the respective fungi. Okay, then they are not kept there. They will be placed in Esco and Basidio wherever they match. Okay, so only fungi with asexual stages are kept here. They are only kept here. So some members are saprophytic or parasitic beta. So they are also the decomposers. Jesse we have Trichoderma species, and Trichoderma polysporum you guys have even read for the cyclosporin part. So mycelium is septate and branched. Only asexual reproduction is there with the help of conidia. With the help of conidia. Okay, so examples mein techo, so Trichoderma species, Trichophyton which can cause the ringworms, so Coletotrichum which can cause the red rot of sugarcane. So and Alternaria, early blight of potato. So coat, taco, triangle and circle, whatever trick you want to keep in your mind you can. So Alternaria cause early blight of potato, but the Phytophora cause late blight of potato. So it is having cellulosic cell wall, and it belongs to what? It belongs to your Phycomycetes. So do not make the mistake here. Which one causes early, which one causes late blight of potato? You should know it, okay? Okay. So that's it. So next is beta viruses, exception to cell theory. They are the connecting link between living and non-living organism. So virus is a Latin word which means venom. So who gave the term virus? Who gave that term? M.J. Vezrenik. He was the one who gave it. But the credit for this discovery goes to Russian botanist Dmitry Ivanovsky, 1892. Year is also very important. So he was the one who recognized that there are certain microbes which are causing mosaic disease of tobacco, but they can cross the bacteria-proof filter. Bacteria-proof filters are the one which cannot be crossed by bacteria, right? They have certain dimensions below which bacteria is, like they'll keep the size of that bacteria proof filter right below their last dimension ki, okay, the smallest bacteria is of this size, but they observed that this causative agent can even cross that bacteria proof filter, right, and it can cause the disease to the tobacco plant, right? So M.W. Wezrenik, he called them Contagium vivum fluidum. Right, so Dmitry got the credit for the discovery, 1892. He observed that tobacco plants are getting affected. So M.W. Wezrenik, 1898, he called them Contagium vivum fluidum, that is infectious living fluid. He demonstrated that the extract of infected plant of tobacco can also cause the infection in the healthy plant. And then W.M. Stanley in 1935 beta, he has showed that the viruses could be crystallized, and crystal consists largely of proteins, right? Viruses could be crystallized and crystal contains largely of proteins. So they are inert outside the body. Outside the body, they are not reactive, right? And he got the Nobel Prize for its discovery. So he crystallized TMV virus, tobacco mosaic virus. Okay, so viruses, you know that non-cellular, right? They are ultra microscopic nucleoprotein entities. So they have nucleic acid covered by the protein coat, which we are calling as capsid. So they contain either DNA or RNA. No virus contains both. Beta, there is now, though the data is not available. If you will solve the PYQ, this example is given that there is a leucovirus having both, right? Having both DNA and RNA, but when we try to dig in more, we don't get the proper information regarding this, and it is also considered as a retrovirus only. So you better don't focus there. But if by any chance this leucovirus thing comes, so that's what you have to mention. You can put this question on the Google. In the question bank, you will find this. So they are inert outside the specific host, and viruses are the obligate parasite. They are they are specific for their host. Okay, so let's suppose this is their genetic material. So they have the protein coat outside, that is basically called as capsid, and that capsid is made up of what? That capsid is made up of capsomeres. Is that clear? That is actually made up of capsomere. So viruses may beta, and some viruses outside this capsid, they also have the envelope, lipid envelope. So if they have lipid envelope, they are the enveloped viruses. If they do not have, they are the naked viruses. So the protein coat of the virus, of this virus, right, the capsid is made by virus itself, but its lipid envelope is made by host, right? So when viruses, they are outside the body, when they are inert, so inert virus ke liye, inert virus which is just a crystalline protein, you use the term virion. It's not viroids, it is virions. So they are inert outside the specific host, they are in crystalline form, then you are calling it as virions, not viroids. Do not confuse it with viroids. So they have nucleic acid, that can be DNA or RNA, right? So they are the obligate intracellular parasites, right? So plant viruses beta, generally they have RNA, right? Exceptions are there. They say plant viruses are generally single-stranded RNA, but you have cauliflower mosaic virus, which is double-stranded DNA virus, and there is Gemini virus as well, curly top virus, which is single-stranded DNA. So exceptions are there. Animal virus is generally double-stranded DNA, but you know that RNA virus is also there. So if that RNA virus, if that RNA is double-stranded RNA, then you will use the term reovirus. If it is the single-stranded RNA, you will use the term retrovirus. Okay, you will use the term retrovirus. So proteinaceous coats are there, which are made up of capsomeres, guys. So envelope is actually what? Envelope is the loose membranous covering that occur in some animal viruses, rarely plant and bacterial virus. So virus without envelope is naked. So it is made up of protein. So protein part of the virus is made by virus itself, but lipid and carbohydrate is by host. Okay, so various enzymes like lysozyme, RNA polymerase, reverse transcriptase, they are present in them. So that is about its structure. So TMV virus, you know it very well that your TMV virus is what? It's elongated, rod-like, having single-stranded RNA. Okay, so its capsid has 2130 capsomeres. So viruses have a different structure. We know that bacteriophage, the virus that infects bacteria. It's the virus that infects bacteria. Got it? So usually double-stranded, but we have examples. Jैसे QB bacteriophage. It is given in your NCERT. So this is basically having RNA as its genetic material. So T4 bacteriophage, beta tadpole-like structure is there, polyhedral head connected to the tail. There is a collar in between. Tail fibers are there for the attachment. So this is TMV, RNA virus and the, right, this is the RNA virus and the capsid. So capsid is having what? Capsomere. So tail fibers are the arrangement, sheath, collar and the head. Is that clear? So TMV, single-stranded RNA, Gemini virus, single-stranded DNA virus, cauliflower mosaic virus, double-stranded DNA virus. Okay, so animal viruses may you have adeno, herpes, retro, rio or mumps, right beta? So you can see that this is adenoviruses. That's how it is. Baki QB bacteriophage, single-stranded RNA, 5 into 174 single-stranded DNA. So other than that, the discoveries, so you know that viroids, right? We have the viroids. So Teodiner, right? Teodiner in 1971 discovered that there is something which is even smaller than the virus. So it is not having the protein coat. It is just the infectious RNA. Infectious causing RNA, which is without any capsid. That is the viroid beta. So their molecular weight is lower and they cause disease in plants. They are infecting what? Plants. So PSTD, potato spindle tuber disease, chrysanthemum stunt is there, citrus exocotis is there. So PSTD is important. So next is prion. So P for protein. So they are abnormally folded proteins. What are they, beta? They are abnormally folded proteins, right? So they have only capsid. So basically, protein is actually abnormally folded, no genetic material. So Kuru's disease, laughing death in human, mad cow disease, this is bovine spongiform encephalopathy, and CJD disease in the humans. So this mad cow disease, in which is affecting the cattle, in our case, that is actually CJD. Right? That is, in our case, this is actually believed to be CJD. Is that clear? So muscle coordination and the brain functions are damaged, result in the mental deterioration. Very dangerous disease it is. So when proteins are abnormally folded, they can do that. Okay, when proteins are abnormally folded, they can do that, beta. So scalpy disease in sheep is also there. So this is all about the chapter. So next is the living word, or the last chapter for today's class. So quite happy, right? Chaliye, so let's go for this. So you have bio. So here, what is living is the part of your syllabus. It is not in new NCERT, but it is in old NCERT. So you have this thing. So when you define something as living, na, there are certain properties that we have to consider. Dekho beta, overall, when you talk about the living beings, okay, right, overall living beings mein, there are certain characteristic features, right? So characteristic feature matlab that is what we see in the case of living beings. So jaise growth hai, reproduction is there, metabolism is there, right? So you have consciousness, cellular organization. So they all comes under the characteristic feature. Matlab, you know that when we say something is living, we will see the growth is there, we will see they are reproducing, we will see they are eating, they are moving, they are respiring and all. But there is one more word that we use, and that is basically the defining properties. That is basically what? Defining properties. So when we talk about the defining properties, so they are the one They are present in all living beings without any exception. Without any exception. They are present in all living beings without any exception and are not shared with the non-living beings. So when you talk about the defining properties, so these three are the defining properties. So characteristic features are all the features, but when it comes to the defining features, only these three are. Why is it so? Because growth is basically what? Growth is basically the increase in cell number, increase in mass. That is what the growth is. Growth, we know that it is the result of anabolism. When in our body, when the buildup is more than the breakdown, then the growth occurs. When the buildup is more than the breakdown, then the growth occurs. So when you people talk about the growth, right? When you people talk about the growth, so it is the irreversible permanent change. It is the irreversible permanent change. And we know that there are two twin properties of growth. And that twin properties are increase in cell number or organism number plus increase in mass. So this is what we say when we see the growth. Okay. Now, in the case of non-living beings, just say, let's suppose if there is a mountain, if there is a sand dune, yeah, if there is a mountain, you know that in their case also, we see their size increases, right? Their mass increases. Why? Because we see the external growth. So external growth, right, which occurs due to accumulation, right, which occurs due to deposition, because we see the increase in their size with time. So this type of growth is accretion. So yes, we do see accretion, right? We do see accretion, that is the accumulation, which the growth where the mass is increasing, right? Growth where the mass is increasing due to the accumulation, we call it as accretion. And when students, you talk about the internal growth, then you are talking about what? You are talking about intussusception. Intussusception means internal growth. So intussusception you see in the living being, but this accretion you see in the non-living beings. Even the secondary cell wall in the plants, because there also the layers are basically occurring, so that is also the accretion. So growth is not just there in the living being, growth is also there in the non-living being. So that's why it is the characteristic feature. We are not putting it in the defining feature category. Moreover, you know that when you talk about a single-celled organism, when these single-celled organism, they grow, their cytoplasmic increases there. So when this ratio disturbs, they're divided into two. So in unicellular organism, we cannot differentiate whether they are growing or they are dividing. So in unicellular organisms, growth and reproduction are what? Growth and reproduction are what? They are mutually inclusive events. Right? What is the meaning of mutually inclusive events? They are occurring simultaneously. They are not occurring separately. Rather, they are occurring simultaneously. So because they are occurring simultaneously, we cannot differentiate them. Right? That is also the thing that you have to remember. But in higher organisms, right? But in higher organisms, these two events are not taking place simultaneously, are not taking place simultaneously, so are mutually exclusive. Done, beta? So that is also the term that you need to know. Okay? That you need to know. Okay? So reproduction, means again, production means to produce. So already existing individuals, they are giving birth to young ones. And we know that not all not all living beings they reproduce, right? We have the sterile organism, sterile worker bees, mule is there, the hybrid, right? Hinny is there, right? Even the infertile human couple. So reproduction is also not shown by all the living beings. Metabolism, then let's talk about the metabolism. So this part, because rest everything is easy. So metabolism, I would like to highlight it from the NCERT. So why all living beings grow? Done. So reproduction is also a characteristic feature, not a defining feature. So yes, characteristics features, defining features are definitely the characteristic feature, but not all characteristic features are the defining feature. So let's talk about the next part, that is the metabolism. Beta, metabolism indeed, told you already, is the defining feature. It is what? It is the defining feature. What? Metabolism. So metabolism is made up of two things. One is catabolism, another is anabolism. One is catabolism, another is anabolism. Cata mane cut, mane you are talking about the breakdown here. Then anabolism mane build up. You're talking about the build up here. So when in your body there are the reactions which is breaking down the complex things into the simplest one, that is the catabolism. And when these small things they join, right, they are forming the big things, they are the anabolism. So metabolism mane chemical reactions which are keeping us alive. So here you will see catabolism as well as anabolism, breakdown as well as build up. Because of that we are living. So definitely if there is any living being, any living entity, be it a single cell or a multicellular organism, without chemical reactions we cannot live, right? Now can these chemical reaction occur outside the body in the lab? Yes, in the lab they can occur, but they will not be self-driven. So if in the lab they are taking place, you will call them as living reaction. Just say, let's suppose in this test tube, if a reaction of living body is occurring here, so it doesn't mean that test tube will become living. No, it is the reactions which are taking place outside the body in the lab. They are neither living nor non-living. They are not making your petri dish living, है ना, if they are taking place there. So you are not putting them, you are not calling them neither living nor non-living. You can simply say that it is a living reaction. Just to mention this thing, right? Just to mention this thing that they are just the living reaction. They occur in the living body and they are taking place outside the living body also. So yes, we can refer them by saying they are the living reaction, but otherwise they are not keeping this test tube live. They are not giving it the life. So let's see this. So all living organisms made up of chemicals. So they will constantly made and change into another form. These are the metabolic reactions, beta. So all plants, animals, fungi, microbes, they exhibit that. So the sum total of all these chemical reactions, it is the metabolism. So no non-living being can exhibit metabolism. So metabolic reactions, we can demonstrate them outside the body in a cell-free system. Outside the body in a cell-free system, in the test tube, in the petri dish. Yes, definitely you can demonstrate them. You can show them. So these isolated metabolic reactions which are performing outside the body in a test tube, beta, you are calling them neither living nor non-living. You cannot say that the test tube is living. Right? So you are putting them in no category, neither living nor non-living, but definitely I can say this is the living reaction. When I'm saying living reaction, which is taking place in a test tube, I only want to signify that it is the reaction of a living body which is taking place outside in the test tube. So metabolism is a defining feature without exception. So isolated metabolic reaction in vitro are not living things, but surely the living reaction. So do read it. And hence, every living body is made up of cells. So cellular organization is definitely the defining feature. So then comes the consciousness. Consciousness means how do we respond to our surrounding? All the living beings, they respond, be it a unicellular organism or it is a multicellular organism. So it is the most obvious but technically complicated feature. So all the living organisms, they have the ability to sense their surrounding or environment and respond to that. Like even a unicellular organism is aware that this water, its pH, temperature is not favoring me. They show the changes to that. So environmental stimuli can be anything. It can be physical, chemical, or biological. right? So we can sense our environment through our sense organs. Plants also respond to external factors. They also respond to external factors like light, like water, like temperature, other organisms, pollutants, etc. So all organisms from prokaryotes to even the most complex eukaryotes like we chordates, we all respond to the environmental cues. So photo period, that is light period, you know that it affects the seasonal breeder, right? It affects both plants and animals. Plants, they also need the light period. Accordingly, you know, in their case, the flowering is also affected with that, right? So seasonal breeder animals are also affected with it. So all organisms, they handle the chemicals entering their body. So we all are aware of their surrounding, but we humans are the special. We are also having a property of self-consciousness. We are not we are aware of ourselves as well. We know when we are lying, we know we are not feeling good inside, we feel the things. So we are having the property of self-consciousness as well. So I know here the question comes from that comma patient thing. So in comma patient things, because machines are taking care of the body things, so what do we say in that case? That person is not conscious. How do we say that? But if we give a medicine to that, if a person is responding like some sort of reaction we are getting na. So in case of humans, you cannot just define it. You cannot just define it, right? That whether matlab, you cannot just keep one criteria to say whether it is living or not, right? So the comma patient, right beta, in the case of comma patient, they are virtually supported by machine, right? Otherwise the patient will be brain dead, and brain dead patient mane dead only, right? So patient is not having the self consciousness, but you cannot say that the patient is not having consciousness. Self conscious is not there, patient is not aware of himself, but we cannot say that there is no self consciousness. Okay, so overall in living body, the interactions are there. Cell will interact with tissue, tissue will form organ, organ will form organ system. So baki beta, diversity is there, you know that. Species, especially you are talking about the eukaryotes here, 1.7 to 1.8 million. In class 12th NCERT in biodiversity, more than 1.5 million is given. So obviously taxonomy is required, right? So that we can arrange them properly, right? So here also one more thing I would like to highlight. जैसे ये देखो, international code for botanical nomenclature is there, international code of zoological nomenclature is there. But if you will read this ICBN from authentic site, from its own site, then at the place of for off is given. So these are the things that I would like to highlight. So baki beta, when you talk about the basis of classification, what do you see? When you talk about the basis of classification, what do you see? The trick there is chicken, CHICN, right? So CH mane characterization. The description is important. Then you have to identify the organism. So CH means what? Characterization. I means what? Identification. C means what? Classification. N means what? Nomenclature. So firstly, the description of organism, then we identify this, classify it, its name is given. So you know that binomial nomenclature is followed, given by Carolus Linnaeus, right? So binomial nomenclature is followed, given by Carolus Linnaeus, right? And two names are given. There is the generic name, which is the species name, and there is the genus name and the specific epithet. So when we have to write it, there are certain rules. So when we are typing it, we have to go for the italics, which will show it Latin origin. The first letter here needs to be capital in the genus, rest all needs to be small. So in and for species, all need to be in small case. So when you are handwriting, when it is handwritten, so genus name, species name, we have to underline it separately. If there will be a same line there, then it will be incorrect. So the genus, the first letter needs to be capital, rest all small. So Mangifera indica. So if I have to mention the author here, so that should be in the abbreviated form. Just let's suppose P. How do we write it? Pisum sativum. So when it is handwritten, it has to be like this, otherwise in italics. So when you are writing it in italics, then there is no need to underline it. Okay? Okay, so because we are running out of time, so that's why I'm trying to elaborate it quickly to you. So here you can see, just biological names. right? The first name is the genus, second component denotes the specific epithet or species. Both when handwritten are separately underlined. If they are underlined together, it is incorrect. When they are printed in italics, italics will show their Latin origin. Then beta, first word denotes the genus start with capital letter and specific epithet starts with small letter. So here you can see, right, if we have to mention, just Linnaeus is mentioned. So this is showing L-I-double-L-N, that this is the author who described this species for the first time. Understood? Understood? So this classification is a process by which we group them into convenient category, right? So when it comes to the category, the texons, just we have 7 broad categories. What do we have? We have 7 broad categories. So already I have given you a trick for that. That is keep pot clean or family gets sick. Keep pot clean or family gets sick. So you will talk about the kingdom, phylum, class, order, family, genus and species. So we know that species is the basic or the smallest unit of classification, and phylum may you also use division. Division we use in the case of plants. So these are 7 broad category, right? The broad category where the highest one is kingdom beta and the smallest one is species. Just we all have seen the animal kingdom. You know that we all are in kingdom Animalia. So basically when you move from kingdom to species, general character decreases.
I found this on the web for you know that we are in kingdom when you move from kingdom to species.
My Siri is just, I don't know doing what. Chalo. So general character decreases and common, or I would say specific character increases. Done? That's what you have to remember, okay? So general character, when you go down, right? Kingdom to species, general character decreases, right? We go more for the specificity because you know that individuals of 2 same species, human, they will have more similarities. Just say for an example, if I'll give you the example here, let's suppose Panthera is the genus. So Panthera leo and Panthera leo will show, 2 Panthera leo will show the maximum similarities, right? Then Panthera leo and the Tigris, yeah, Panthera leo and the Pardus, right? So that is why I am saying that when you move from kingdom to species, general character decreases and specific character increases. But when we move from species to kingdom, general character increases and specific characters, yeah, common characters, they decreases. Got that? They decreases. Understood? Right? So kingdom is basically the group of related phylums. So phylum mein you will see group of related classes. Classes mein you will see group of related order. Order will have group of related families. Families will have group of related genus. Genus will have group of related species. Yeah, species. So genus is having group of related species, family is having group of related genus, order is having group of related families, class is having group of related orders, phylum or division is having group of related classes, and kingdom is having group of related phylums. So here the species will show the maximum similarity. At the level of species you will show maximum similarity, then they will keep on decreasing when you will go up. And when you move to the order or something, then you see the aggregation of characters. Then what do you see beta? You